So we are live now, I think, but we're gonna wait a little bit for people to join us. Yeah, welcome everybody who is joining today to this session 38, room 4, in case anybody is confused. Thanks for joining us. We are waiting a little bit, like 1 minute or 1 minute or two to allow more attendees to join, and we are starting very, very soon. All right, I think because we are a bit tight with time, we're going to start. So hello, everybody. My name is Alex Hernandez. I'm speaking to you from Madrid, and I will be the moderator of this session. This is session 38, room 4, Neuromatch 3. And I want to thank Ariana, who is helping us in the back end, doing a lot of work. And yeah, we have a great lineup of speakers today. They are Emily Crow, Noor Seidel, and Karthik Mahesh Paradarajan. And they're going to talk about visual perception of complex scenes, vision and action, and affordances. And before starting, I just want to remind everybody that you are welcome to tweet about the session, use the hashtag NMC3, and of course, feel free to ask any questions. Keep this session as interactive as you like, and we'll have a better time altogether. All right, so I'm going to introduce the first speaker. She's Emily Crow. She's currently a postdoctoral researcher at the Vrije Universiteit Amsterdam with Ellie Brenner and Jeroen Smeets, where she studies sensory motor control. So welcome, Emily, and the stage is yours.
Thank you. Yeah. Can we see this okay? Perfect. So thank you. And today what I'm going to do is characterize a hand stabilization mechanism to explain the hand's response to abrupt background motion. So if I give you the task of intercepting this green ball as it moves across the screen, and then suddenly I move the background abruptly, what your hand will do is it will follow the direction of that abrupt background motion. And this is called the manual following response, and it's a very robust and well-documented effect. So basically, if the background moves leftward, my hand will follow that background leftward. And if the background moves rightward, my hand will follow that background rightward. So this has led to people asking, well, what causes this manual following response and why do we do it? And the most compelling explanation so far is that it is a compensatory response to inferred self-motion. So the idea that is, if the background moved leftward, I infer that I myself have moved rightward, and therefore I need to correct accordingly and compensate by moving my hand leftward in the direction of that background motion. And it's proposed that we use the instantaneous optic flow to estimate how much the magnitude to which I've moved leftward or rightward. There are, however, some experimental characteristics that do not support this suggestion. So first of all, the finding that it's primarily based on background motion close to the target. So this suggests that there is a bias to which motion drives the largest manual following response, and therefore it doesn't appear to be reliant upon estimating self-motion from the whole optic flow. And then second, this manual following response isn't present after illusory self-motion is induced by vestibular stimulation. So this doesn't really make sense, because if it is a response to inferred self-motion, it shouldn't matter how that self-motion is induced. So what we want to do here is propose a new hypothesis called the hand stabilization hypothesis. And according to this idea, people do not actually analyze the instantaneous optic flow to estimate their self-motion, but we assume that the endpoint of our action is stable. Any abrupt motion that appears near the desired endpoint of my action is therefore attributed to that position having moved relative to myself. So during my ongoing movement, I follow any motion within that region to stabilize that movement. And what I'm going to do here is present 6 experiments that we believe provide support for this hypothesis. So the first experiments I will talk about were completed by participants stood in front of a very large screen, and they perform an interceptive action using the index finger of their dominant hand, and they were free to move however felt most comfortable. And we basically tracked the position of the finger throughout the entire trial. This is what the experiment task looked like, and this did vary slightly across the experiments I will present. But basically, to start a given trial, participants have to bring their finger to the black starting circle on the top left panel. After a random period of time, a target item will appear, which is the black circle, and that will start rolling rightward across the screen. And participants are instructed that they need to intercept that black target when it enters the grey circle, which is what we call the interception zone. At a fixed point during the target's motion, the background will move abruptly. This will last for 100 milliseconds and then stop. So it's a very abrupt motion. And this can occur either leftward or rightward, so is always horizontal. The target then continues moving, and the trial will end when a participant hits the screen such that they have performed their interception. For all the experiments I'm going to present, I will also use the same data analysis. So what we first do is, for each participant, we take the average lateral position of the finger from the time of the abrupt background motion. So for each participant, this gives us an average rightward response and an average leftward response, which you can see plotted here for individuals, and then the thicker lines show the average across all participants. We then convert this into a velocity, and the final step is we use this to quantify what we call a response. So what I do here is I do the average rightward velocity, minus the average leftward velocity. And this shows us whether there was a difference in behavior when the background moved leftward compared with when it moved rightward, which we define as a response to the background motion. So importantly here, if there is this positive response, then it shows the classic manual following response where the hand has indeed followed the direction of the background motion. And this is the analysis that I will present throughout the next experiments. So in experiment 1, we were interested in whether we're less likely to respond to abrupt background motion if the background is unstable. So an assumption of the manual following response is that because we typically assume the background to be stable, any background motion, any retinal motion of the background is more likely to reflect self motion than background motion. So we asked the question of what would happen if we constantly changed the background such that it wasn't a very reliable source of visual information from which to infer self-motion. So we used 3 conditions. In a high stability condition, there was only the abrupt background motion on 20% of trials, such that overall you were viewing a very stable background. In a medium stability condition, there was always abrupt background motion, either leftward or rightward. And in the low stability condition, we used asynchronous limited lifetime dots such that they looked a bit like this with flickering. So the background was constantly changing and then underwent the abrupt background motion, such that it isn't a very reliable background. And these are the results. So for each figure, I will plot the average response of participants in the main figure that you can see here. And in the insert on the top left, I present the individual initial responses to the perturbations. And what we define as the initial response is the average magnitude of the response between 150 and 200 milliseconds. So the gray bar that you can see on the x-axis. What I want to draw your attention to here is even in that low stability condition, so in the purple curve where the background was constantly changing, we still clearly display a manual following response. So this shows that any abrupt background motion is sufficient to drive this response, and it doesn't depend on us inferring how stable the background might be prior to the onset of our action. Instead, we just assume the endpoint is stable such that any abrupt background motion leads to the manual following response. In the second experiment, we wanted to ask whether abrupt background motion of any structures is sufficient to elicit the manual following response, or do all structures need to move? So specifically, we were interested in what if we only move 1/3 of these background dots and the rest of them remain static. So it looks something like this. We were interested in whether we would still get a response in the direction of background motion. And these are our results. You will also see in the legend something about colour, but I'm not going to talk about that in this talk. And instead, I just want to draw your attention to the fact that in both conditions, clearly there is a manual following response. So even when the majority of background structures are static, any abrupt background motion requires us to stabilize that ongoing movement. In the third experiment, we really wanted to test just how little abrupt motion was needed to drive the manual following response. So here we had a similar idea, and only 3 out of 600 dots, so not accurately represented here, moved. So a very, very small proportion of the dots actually underwent motion. And this was either at the location of the interception zone or at the location of the target. So it looks something like this, where the majority of your background is completely stable, but 3 dots did shift leftward or rightward on any given trial. And these are the results. So again, the important take-home message here is that in both conditions, so whether the 3 dots that moved were at the location of the target or the 3 dots that moved were at the location of the interception zone, we still get clear evidence for a manual following response. So taken together, these last 2 experiments really showed that only moving dots are considered and any motion seems to drive this response, and the static surrounding background doesn't seem to be playing a role. After this, we wanted to look at trying to refine the spatial constraints of the manual following response, of our hand stabilization hypothesis, sorry. So the next question we asked was, does only motion that occurs very close to the target's path contribute to the hand's response? So what we did here was we basically split the screen in half, although the horizontal line wasn't there. And the 2 halves of the screens moved in opposite directions, like this. And we were interested in what would happen in this condition, whether participants would respond at all, and whether they would follow the motion presented in the top half or the bottom half of the screen. So the first thing to note here is the golden curve, and this is our control condition basically. So here, both the top and the bottom halves of the screens moved in the same direction, and you can see that standard manual following response. The turquoise curves shows the response of participants when the 2 backgrounds moved in opposite directions. And this looks very different. So although there is this tendency to respond in the direction of the background motion presented in the top half of the screen, where the interception zone and the target were presented, the bottom half of the screen is clearly not irrelevant because this manual following response looks very different to the standard golden curve. For the last 2 experiments, we used a different setup. So here we were interested in the role of depth information. So we had 2 screens, and background dots were presented on both screens, and the target and interception zone were always presented on one of these 2 screens. Experiment 5 assessed the question of whether the magnitude of the manual following response depends on where the target and background were presented relative to each other in depth. So if we take our setup, we say we have the near screen and the far screen, and in this experiment, only background dots on one of those screens would move. The target on some trials was presented on the same screen as where the background motion occurred, and at some trials was presented on the opposite screen to where the background motion occurred, such that we have a 2x2 design like this. We were interested in whether these kind of manipulations changed the magnitude of the manual following response. And what we can see here is it clearly does. So the main result that I want to draw your attention to here is the 2 thick curves that show the largest magnitude of the manual following response. And this occurs when the target is presented on the same screen as where the background motion occurs. And what this suggests is that depth information is important, and the relative location of the target to the background matters in terms of leading to a larger manual following response when they're in the same plane. For the final experiment, we used the same setup. And this time, there was always background motion on both screens. In one condition, the background motion moved in the same direction on both screens. And in another condition, they moved in opposite directions. And in line with the previous experiment, we were interested in what direction the hand moves in here. The first thing to draw your attention to is the turquoise and golden curves. So these are our baselines here, where both backgrounds moved in the same direction, and we can see the standard manual following response. The red curve shows the response when the 2 backgrounds moved in opposite directions and the target was presented on the near screen, And what you see is that in line with what we would expect from experiment 5, the hand follows the background motion on which the target is presented. The purple curve, however, is somewhat more difficult to interpret because it seems like the background, we first follow the screen on which the target is not presented, and then there's this delayed latency to follow the background on the motion of the screen where the target is presented, which is a somewhat more confusing result. But overall, to conclude, we'd like to introduce the hand stabilization hypothesis to explain the manual following response. We think that the manual following response does not rely on an analysis of the global optic flow to directly estimate self-motion, but instead that we assume the endpoint of my action is stable, such that any abrupt motion near where I am aiming is attributed to that position having moved relative to myself and requires me to perform a manual following response to stabilize my ongoing movement. Thank you for listening. I'm pleased to ask any questions.
Thank you very much, Emily, for the presentation. That was really, really interesting. Unfortunately, we don't have much time for questions. If there is one quick question from someone in the audience, we can do it. But otherwise, I'm afraid we will have to move on. That was really interesting, Emily. And maybe if people have questions for Emily, feel free to ask in the chat. I'm sure she'll be happy to reply in the chat. Right?
Yeah.
Cool. Thanks very much. We're gonna move to the next speaker. Noor Seidel. Noor is also postdoc at the Vrije Universiteit Amsterdam, working with Martijn Meta, and she combines neuroimaging, behavioral experiments, and machine learning to study vision and vision in the brain and machine vision. So I'm really looking forward to her presentation. Welcome, Noor.
Thank you. Can you see my screen? Good. Okay. Yeah, so thanks for the introduction. My name is Noor, and today I will present a study in which we investigated the need for recurrent processing during visual object recognition. And today I will present data from behavior, EEG, and deep neural networks to argue that this depends on the need for scene segmentation. So object recognition has been studied for a long time, and because people are so fast and efficient at, for example, recognizing this dog, it has been argued that a first feed-forward sweep of visual information might be enough for successful recognition. And while this might be true for this dog, recently a multitude of studies using more naturalistic scenes have suggested that recurrent processes might be employed more adaptively, so depending on the visual input. So while feed-forward activity might be enough for very simple scenes with isolated objects, such as this dog over here, for more complex scenes or more challenging conditions, so for example, objects that are partially occluded or degraded, additional visual operations that require recurrent computations might be needed. So for simple scenes, rapid recognition may thus rely on this coarse and unsegmented feed-forward representation, while for more cluttered images, recognition may require extra visual operations to, for example, group parts of the object together and to segment this object from the background. And several studies have already shown more indirectly that the segmentability of a natural scene might influence the degree of recurrent processing. So for example, in this study from 2014 by Mika Koivisto and colleagues, they reported that masking, which is a technique shown to affect mainly recurrent but not feed-forward processing, was more effective for objects that were rated as being difficult to segregate. And also in a more recent study, we showed that natural scene complexity indexed by 2 natural scene statistics modulates the degree of feedback activity. And interestingly, these 2 parameters, they provide information about the segmentability of a scene. So in this complexity space that you see over here, simple images containing one or a few easily segmentable objects are on the lower left, while images with a high degree of fragmentation end up on the upper right. So these 2 studies seem to indicate that recurrent processing plays an important role during figure ground segmentation. However, both these studies, they did not test for effects of segmentation explicitly, and they used natural scenes that were uncontrolled, so in which complexity could correlate with other contextual factors in the scene. So therefore, in the current study, we wanted to systematically investigate whether scene complexity influences the extent of recurrent processing during object recognition. So to do this, we went for a more hybrid approach, in which we showed objects in isolation or superimposed on 3 categories of phase-scrambled versions of the real-world scenes, still corresponding to the complexity properties of the naturalistic scenes that we used in the previous study. So by generating the stimuli in this fashion, we combined relevant features of objects in natural scenes, embeddings with different levels of complexity, with a design that is more controlled. And importantly, each object was presented in all conditions, allowing us to attribute the effect of complexity or segmentability of each trial and rule out any object-specific effects. So basically, our aim was to see whether we need more extensive or recurrent processing specifically for these more complex images. So to this end, participants performed on a 5-choice categorization task in which they had to differentiate images containing cats, birds, fire hydrants, frisbees, and suitcases as accurately as possible. And in half the trials, we impaired feedback activity with visual backward masking. So in addition to behavioral measures, we also measured EEG responses to examine the time course of visually evoked activity. We used a setup in which we split the data into an exploratory and a confirmatory data set so that we could use this first data set to explore, but also to define our electrode selection, pre-processing steps, and time windows for further ERP analysis. So then in this confirmatory data set, we looked whether we could replicate these results both in behavior and in the ERPs. So I will first show you the behavior. So basically, results indicated that apart from main effects of both scene complexity and masking, there was an interaction effect, indicating that masking impaired performance for objects presented on more complex backgrounds stronger than for less complex backgrounds. So this is what we expected. So if you need recurrent processing for these complex images, you expect it to suffer most from backward masking. And if we look at the confirmatory set, we see again the same pattern of results. So then to the ERPs. To investigate the time course of figure ground segmentation in visual cortex, we pooled the evoked responses to the masked and unmasked conditions across occipital and perioccipital electrodes. And as you can see over here. And what you can already see is that here you see the 4 different conditions. What you can see is that they start to diverge at some point. This is easy to see for the unmasked condition, and for the masked condition, it's more difficult also because we see a very clear effect of the backward masking. So what we did is we generated difference waves by subtracting the signal of each condition from the segmented condition. And doing so enabled us to eliminate the effect of masking and to only look at the differences between the low, medium, and high complexity scenes. So then, because this was our exploratory set, we used this data to define different time windows. So what we did is we performed t-tests on every time point to see whether the signal was different from 0. And then, based on these significant time points, we defined 5 time windows. You can see over here. So we have a very early window, around 100 milliseconds, and then 4 later time windows. So then we repeated this analysis for the confirmatory set, but now in the confirmatory set, we could look at these different time windows to see whether there were different effects of complexity and masking. So here we see that the data looks quite similar, but now if we look at the average ERPs in these different time windows, we see different patterns emerge. So crucially, what we see is that main effects of complexity only start to emerge in time windows 2 and 3, indicated with an asterisk below the time windows. And importantly, we only see an interaction effect, indicated with an X or a cross, appear in time window 4, indicating that masking impairs performance ERPs differently for different complexity conditions only late in time. So then we presented the same images to deep convolutional neural networks with different architectures to more directly test whether different feed-forward or recurrent architectures perform differently on these stimuli. So first, to investigate the effect of recurrent connections, we tested different architectures from the CORNET family. And in the CORNET family, we tested three architectures: CORNET Z, which is a feed-forward architecture, CORNET R, which is a recurrent version, and CORNET S, which is recurrent with an additional skip connection. And then, to further evaluate the influence of network depth on scene segmentation, we also showed these images to three different ResNets, or residual networks, with increasing number of layers. And here we see the results. So in line with human performance, results indicated a higher degree of impairment in recognition for complex backgrounds for feed-forward, as you can see for the CORNETs, and slightly less clear for more shallow networks, as indicated by the ResNet performance. So we have three lines of evidence showing that recurrent computations are critical for recognition of objects embedded in more complex backgrounds. So first of all, we have behavioral results indicating poorer recognition performance for objects with more complex background, but only when feedback activity was disrupted. Secondly, we see EEG measurements showing clear differences between complexity conditions in the ERPs quite late in time after this first feed-forward visual sweep of activity. And finally, we see that deep convolutional neural network performance confirms this, showing that feed-forward networks show a higher degree of impairment in recognition for objects in complex backgrounds compared to recurrent networks. So together, these results support the notion that recurrent computations drive figure ground segmentation of objects in complex scenes. So for this dog, when presented in isolation, perhaps a feed-forward sweep might be enough, but when it's presented in a naturalistic background, that could be different. Thanks, and also to my colleagues, and for Neuromatch for organizing this event. I'm happy to take any questions.
Thank you very much, Nora. That was a really nice presentation. I really enjoyed it. We don't have questions from the audience yet. I encourage everybody in the audience to ask questions either on the chat or on the Q&A tool, and I will ask it for Nora. In the meantime, so I've been following all this personally, all these new results showing evidence like you did also very, very neatly with behavioral and electrophysiology and so on, showing evidence that the recurrence is necessary. So how would you explain this fact? So on a higher level, why do you think that recurrent processing is especially necessary for processing complex scenes, complex backgrounds in the case of
yes. So I think that for these very simple scenes or yeah, I don't know, isolated objects, it might not be important that we know exactly where every part of the object is. So there, just this first very coarse representation is already clear enough or distinctive enough for your brain or for you to recognize what is shown. And for these more complex images, yeah, this might be a too noisy representation, or there might be conflicting features. And there, it might become more important that you know exactly which parts of the image you need for the task at hand. And yeah, we know that recurrent computations are involved in encoding these more fine-grained, detailed spatial relationships. So I think that's why we see this difference.
Mm-hmm. Cool. Thank you. And I mean, my intuition is as well that, so in general, we don't normally see a lot of we don't get a lot of visual input without much complex background. So we are constantly processing complex scenes. Do you think that it will be able it will be possible to show evidence of recurrent processing as well, to show to understand complex backgrounds with more natural images and distinguish?
Yeah, so I think this research really shows that it's important to also use natural scenes in your research, because the results can be quite different from all these previous studies showing, for example, these isolated objects on gray or uniform backgrounds. I don't know what your question was exactly, but
yeah, I was a bit distracted.
That's okay.
No, I wanted to ask that. So in your experiments, you are using this kind of artificial synthetic background, I would say.
Yes.
So I'm wondering if you have ideas how to reproduce this sort of experiments, which I find great, with natural images, for example, with.
Yeah, so So in a study before, we did use naturalistic scenes, and there, we noticed that it is quite difficult to attribute the effects only to the difference in background, for example, because of course, with naturalistic scenes, there are just many other factors that correlate or vary along with the complexity of a scene. So I don't know, what you could do is maybe try to generate more, I don't know, synthetic, or generate scenes that are naturalistic, but in which you have still control over, for example, what other objects are seen, are invisible, or you should, then you have to very carefully select your stimuli, and
yeah, exactly. Yeah, it's a complex.
Maybe using virtual reality scenes or virtual environments, something like that, then you would be able to better control this.
Yeah. Okay, I think we need to move on. Thank you very much, Noor, for your presentation. I'll keep an eye on your future work, and I'm gonna present our next speaker. He is Karthik Mahesh Varadarayan. He's a computer vision engineer at KCL and TU Vienna, and over in the US. And he is founder of a 3D reconstruction company, if I'm not mistaken. And today he is going to talk about electrophysiology of affordances. So welcome, Karthik, on stage. All right, we can see your screen.
Okay, can you hear me? Perfect. Okay, so today my talk is going to be on electrophysiology of affordances. This is work done at KCL as well as at TU Vienna. And so we're going to be focusing on a very specific definition of affordances coming from Gibsonian literature. There are a number of different variants to what an affordance is. It could be anything from a functional form fit to user interfaces to user experience and so on. But we'll be focusing primarily on the functional aspect of an object. So we'll be looking at visual primitives that help you identify the functionality of the object and how that influences recognition and related task processing in humans, essentially. So we're going to be looking at the sensory motor control codes that can be embedded as affordance features. So going further, as I mentioned, we'll be talking about the functionality of objects as the key underlying principle using which we define it. So if you look at a mug, then it has at least two functional parts, one being the handle of the mug, which is graspable, and the other convex shape, which actually holds the liquid or any solid that you put into it. So that part provides you with the containability affordance, and this is key to defining what the object is. The functionality of the object essentially defines the category of the object, the type of the object, and so this can be helpful in recognition. So in the case of the objects seen at the bottom of the screen, you have things like weirdly shaped mugs. One looks like a cow, the other looks like a shoe. So how does a human actually classify these? So this depends upon again the functionality of it and using some kind of inference to identify how these functional parts link with each other and how they can be used. So we'll be talking about these types of affordances. And specifically, we're going to be categorizing affordances into structural affordances, material affordances, and grasp affordances. And we will be looking at one of the various different aspects of it. The aspects being vision, robotics, psychology, psychophysics, and neuroscience. We'll be focusing on the neurobiological principles and the electrophysiology of affordances in this talk. So, oops. So this is kind of related to the previous talk as well. There is a lot of semantic understanding which helps improve recognition performance in humans. So essentially, if an object is out of context or simpler or in a different color or texture, it reduces the accuracy with which the object is recognized. It also increases response times in the sense that the subject would need to spend a larger amount of time in order to identify the object, and classification rates also go down in such situations. So we are talking about a gradient of changes with different types of objects. So starting with something that looks like a cartoon, something that is more or less the actual shape, and then in C, you actually put it in context of an affordance, how an object is grasped or how an object moves in the environment in a semantic context. And D and E are like are novel variants of the object. So this kind of reduces the classification. So C is typically when you obtain the highest accuracies, and on either side of the spectrum you get lower accuracies. So we'll be talking more about how affordances exactly help with these with improvements in recognition and response times. So yeah, we claim that all of this is grounded in common coding theory, in the sense that there are sensory motor codes, and these sensory motor codes enable the mixing of both visual percepts as well as how an object can be interacted with in terms of motor primitives. And we essentially try to fuse these functionalities to define the recognition of an object, the features that are required to recognize an object. So we looked at like the MNS pathway as well as AMNS pathway, the mirror neuron system, anti-mirror neuron system, and how they might play a role in visual processes linked to affordance-based recognition. So these are links from both the how pathway and what pathway as well. So we came up with a framework for this, which was presented a little while back, on various types of linkages between the how pathway and what pathway, going from bottom-up affordance detection, saliency type detections, to high-level top-down saliency, as well as equivalence class recognition and novelty detection. I won't go into detail, but we'll discuss more about the electrophysiology aspects of it. So this is again some of the results that we obtained from the affordance test. So if an object is directly used in the context of recognition, it results we get recall rates of like 34%. With the aid of affordance, we are able to improve the recognition rates all the way up to 62%. And if we put it in a negative affordance construct, something like behaves like a distractor, then the recall rate goes down to 21%. So clearly, affordance plays a significant role. This could be ego affordance, in the sense that the user of the system is actually performing the action associated with the affordance, or it could just be an observation from an external like demonstrator. So we built a semantic pointer architecture to kind of formulate this logic and test it out, whether affordance codes based on sensory motor inputs can sufficiently model visual primitives. And we obtained quite a bit of promising results, in the sense that the recognition rates with affordances were typically much higher compared to typical shape features. And this was performed across several categories of objects. And going further in there, so we have a clear understanding that affordances can play a significant role, and they can be modeled as sensory motor codes. So we wanted to look into the actual modeling process. So in terms of EEG, EMG, and all the types of electrophysiological measurements that you can make. So we play a quick video of the kind of demonstrations that we did. So essentially this shows a ball being rolled on a table. First a person is demonstrating it to the subject, and then the subject tries it out on his own. And also we kind of simulate the execution of this functional behavior. In the sense, rollability is an affordance here, and we want to kind of simulate it even without having the actual object in place. So and we measure the EEG readings in these experiments. So oops. Let's see if this video plays. Okay, yeah. So pretty straightforward. We roll the actual ball, then we simulate it, and then the subject has an opportunity to actually play the ball, essentially perform the affordance associated with it. So we did these kind of experiments across a number of different affordance categories. We collected the EEG data, looked at the various types of various FFT channels required for the processing. So we looked at the alpha, beta, gamma, sensory motor rhythm, and mu channels. Mu channel is particularly of interest here since it's associated with sensory motor performance. And so we used primarily C3, CZ, and C4 electrodes for this measurement. And what we found was essentially that, which confirmed a lot of previous experiments, in the sense that there was the affordance codes, or the mu channel, or here in this case represented by the alpha, largely had very distinct and unique codes for each affordance category, but within an affordance category, the codes were very similar. So this is what we wanted to kind of demonstrate, in the sense that affordances are unique and they can be used used in visual processes for classification and recognition and so on. And these experiments kind of are in line with our expectations. But then within subcategories of affordances, there is still ambiguity. So we wanted to model further using EMG, and especially in the context of grasp affordances. So for grasp, so we were looking at the KTH database of grasp taxonomy with 33 categories, and yeah, so we modeled this using the EMG sensor, and basically performing grasp actions from the database, something similar to this. And this is specifically a thumb abduction with Palmer 2-5 grasp performance. Again, we looked at the EMG codes. It helped improve the performance, but still was quite ambiguous, especially in the context of pinch grasp. So we went further on and wanted to model this in terms of proprioception modeling. So we looked at accelerometer codes, and also used a data cloud to collect more pulse and velocity information to improve our modeling performance. So with all of these put together, in addition to that, we also used gaze mechanisms to identify point of contact on objects, as well as the gaze location where a person was looking while interacting with the object. So a combination of all these systems, we claim can be used to model all of the affordances that is out there in literature in terms of functional primitives. So we are able to model like 25 structural affordance types, 33 grasp affordance types using this schema of multiple codes. So yeah, essentially, in conclusion, what we found was affordances are useful indeed as sensory motor codes. EEGs do provide a sufficiently distinctive framework for recognizing affordances, but for further granularity, EMG is also helpful. but then again, we need more than that. We need to use gaze and finger perception, etc., to improve the quality of, improve the distinctiveness of these sensory motor reference codes. And yeah, we plan to complete this work in terms of building all these codes and use it in a vision-based system for recognition and task processing, essentially. So yeah, any questions? That's my.
Thank you very much, Karthik, for your presentation. We don't have any question from the audience yet. I am curious. So I suppose this is very hard to study, to build like computer vision models that take into account the affordances of objects. What would you say it's the main challenge, and what would be the most important things to address in future work?
I think coming up with the taxonomy is quite hard. So there are a number of different affordance taxonomies. Bulk of it are very restricted to objects that are that can be clearly defined, right, in terms of CAD models, in terms of what the manufacturer of a certain object would have thought should be the shape of the object in order to determine how it can be interacted with. So there are still quite a few categories of objects, even like household objects, that are outside affordance specifications. I think that is kind of the main challenge. There are a number of different ways to doing this. Machine learning being a big way, like with ConceptNet, and there are affordance nets, and there are some different automatic learning schemes out there. But still, I think in terms of the range of objects that can be covered by such systems, especially in a functional form factor, it's still pretty restricted. So I think that's kind of the main challenge.
I see. Okay, we need to finish because the next session will start in 1 or 2 minutes. I want to finish by thanking again all the speakers, Emily, Noor, and Karthik. Thank you for your presentations. Enjoy the rest of the conference, and thanks everyone who attended the session, and also Ariana, who helped a lot in the background. Thanks very much, and enjoy the.
Thank you for hosting.
Thank you.
Well, perfect.
Yeah.
Thank you.
Hopefully I do not catch fire. That would be unfortunate for everyone.
Well, this season we're having a lot of tropical storms during the fall as well. So I hope everybody's power stays on and everybody's safe.
I'm in the wettest country you could imagine, so I don't have that issue. Anyway, so I will. People are starting to come in. So it is my great pleasure to introduce my first session of a Friday morning of Halloween weekend in Ireland. This is an interactive session. In the background, hopefully you won't see him because things will go smoothly, is Taha Morche. And we have 3 speakers from all over the world, but I actually think only 3 locations. Josefa Zalazar is in Madrid, yeah? He'll be our first speaker. Then it's Thomas Emmer, I assume Germany, but am I wrong? Yep. And then lastly, Pascalina Guida, who is in Madrid as well. So yeah, these are everywhere I've lived. If there was someone in New York as well, we'd hit all my places. So please ask questions in the Q&A, put things in the chat. Also, because it's an interactive session, the other panelists can't put things in the Q&A, but you can ask questions. So that's a great time to interact with each other as well. I'll be posting some things in the chat, and if something comes interesting as well, I'll post up papers that might be pertinent and so forth. So I think we can start the session. So our first speaker, I'd like to introduce Josefa Zalazar.
Perfect.
Is a postdoc researcher in the Center of Integral Neuroscience in Madrid. She's studying selective vulnerability of specific subpopulations of dopaminergic neurons for Parkinson's disease. To advertise my own talk later on, I'll be talking about Parkinson's disease at 5 o'clock today. So it's the two talks there are particular interests for me, but also I'm very interested in neuromorphic architecture. So all three talks will have a great interest for myself. So over to Josefa.
Thank you. I'll start sharing. Wait, sorry. Okay, so thank you very much, John, for the nice presentation. So as John told you, I'm a postdoc at the SINAC in Madrid, and I'm gonna present you the project that we have just started in collaboration with the Roper Lab at Goethe University in Frankfurt. So as many of you may know, Parkinson's disease is the second most common neurodegenerative disorder, affecting approximately 5 million people worldwide, but it's expected to increase up to 10 million by 2030. Classical symptoms are motor features such as rigidity, tremor, bradykinesia, but also it's accompanied with non-motor symptoms at early and late stages, such as cognitive decline. The two main pathological hallmarks are the loss of dopaminergic neurons in the substantia nigra pars compacta, and of course the aggregation of alpha-synuclein that will eventually lead to the formation of the Lewy bodies. It is very important to take into consideration that the neurodegeneration within the dopaminergic system is restricted. So it means that it's been reported that there's a preferential loss of the ventrolateral TIA compared to of the substantia nigra compared to more medial regions and of the BTA. There are many factors that may explain this selective vulnerability, especially regarding genetic or molecular factors such as calcium handling, oxidative stress, mitochondrial failure, etc. But to the date, there's no consideration of the function that these neurons play and the role that these neurons may have in behavior. So it is important to take into consideration that the brain has two major systems of controlling behavior that are mediated by dopamine, which is the goal-directed system and the habitual system. The goal-directed system involves voluntary actions and it engages the associative cortex and the dorsomedial striatum. And on the other hand, the habitual system involves automatic actions and it engages the sensory motor cortex and the DLS. So as I have already told you, the ventrolateral TIA of the substantia nigra projects to the DLS, and it's been shown that in early stages of PD, there's a preferential neuronal loss at this area, and therefore it's leading to dopamine depletion at the DLS. Also, it's been reported that increased corticostriatal activity can be enhanced by a repetitive habitual behavior. This excessive corticostriatal activity increases calcium at the synapses, and it also leads to an increase in the secretion of alpha-synuclein in the striatal extracellular space that then may be uptaken by these dopaminergic axons. So our hypothesis is that the engagement of the ventrolateral TIA of the nigra in habitual behavior can be considered as a new additional vulnerability factor for this selective dopaminergic vulnerability. The habitual behavior will recruit corticostriatal circuits that will be involved in the performance of the automatic task. In our case, we have decided to assess this with the lever press assay. So once this corticostriatal circuit is defined in our specific task, we will also challenge it with injection of alpha-synuclein fibrils at this point. So here's the methodological strategy that we're gonna follow. So we will target the forelimb motor region of the cortex, especially in the right paw region. We will characterize this circuit And then we will inject alpha-synuclein preformed fibrils at these specific coordinates of the cortex. Then we will divide the animals into groups. One will be the control, and the other group will be exposed to enhance habitual behavior by the lever press assay that will specifically affect the right paw. And then we will analyze if indeed alpha-synuclein fibrils are being taken by the dopaminergic terminals, and of course the influence of this enhanced behavior in the ventrolateral tier of the nigra. So very quickly, as we just have started this project, I will show you the preliminary results that we have obtained so far. So first of all, we have started by the characterization of this corticostriatal circuit involved in the task. So as I told you, we are targeting the forelimb motor region of the cortex, and specifically the digit area. So we are focusing on these coordinates. That's why we've selected them. And we have performed tracing experiments in order to fully characterize this circuit. So we have started with classic anterograde labeling using FAL, and we have injected these at these coordinates. Here are the injection sites, and as expected, we've seen that the tracer is being able to transport to the projection, and it does indeed correspond to the DLS region. So in order to confirm this circuit, now we have used retro labeling. So we have used red beads at this, and we have injected them at this defined DLS coordinates. So this is the injection site. And as you can see, we've been able to see the projection at the cortical regions that we are interested, and also at these terminals projection site at the level of the nigra. So right now, our ongoing experiments are the important ones. So right now, what we're doing is we're injecting these alpha-synuclein preformed fibrils in these coordinates that we have defined at the cortex, and we are starting with the behavioral assessment of the lever press assay in order to later on study the degeneration that we will have at the substantia nigra. And finally, I would like to thank Professor Roper and Professor Obeso for giving me the opportunity to start this project as my postdoctoral project. To all of you for your attention, and I'm happy to take any questions that you may have, as this is a very starting project, so there's a lot of things that could be asked and we can comment on. Thank you.
Thank you so much, Josefa. So, well, it's early morning, so there are no initial questions, but there might be questions at the end. So I'm going to ask is, so with this kind of mouse model, how much of a delay, like between the control mice and the sham and the Parkinson mice, like how much is that motor delay obvious in the behavior, you know? And how much do you think this motor symptom, like, because a lot of my work shows that it's no surprise people with motor disease have a motor issue, but how do you think that'll inform the other symptoms as well by learning, trying to address the motor part of the symptom of the disease? Or like, well,
we are trying to with this model, we are trying to mimic more kind of like the early stages. So we are supposed not to get many motor manifestation. We may eventually have some, but right now what we are seeing with the lever press assay is that there's no difference in the motor behavior of the control group and the group that has been exposed to this lever press assay. So right now, what we're most focused in is that if this enhanced habitual behavior will at the end affect the nigra. So we expect to see at the behavioral level no difference between the two groups. That will be the ideal. Maybe, of course, as we are overstressing them with this task, we may eventually see also some changes in behavior, but our initial thoughts it's that there's supposed to be no difference in the motor.
No, it's funny you say with some of the stuff we show on Parkinson's is that we show that people with Parkinson's have similar motor response times, but they're doing it by compensating by recruiting more areas. And then when we tax the system, that's when we see the real behavioral differences. But to see the more subtle differences, you have to look neuronally, you have to look at these kind of things. So yeah, and also early stuff is so hard. Parkinson's being so hard to diagnose and symptoms being the primary course. It's so important to understand that early stuff is vital.
Yeah, totally. So this is,
you can live very long without actually realizing that it's the beginning of a Parkinsonian symptom.
Exactly.
So that.
This project it's focused exactly on that phase, on the prodromal phase. So it's been known that since you start showing symptoms, the neurodegeneration process may have started even to 10 to 12 years before you show any symptom. So we are trying to focus on that period, even though we know that from the patient side, it's still very hard to diagnose that moment. But also, as there are all these biomarkers coming through, we hope that in some years there may be a clue. So that's why we want to focus on that prodromic phase with this project, in order to study more at this moment where we think that therapeutic interventions may have an effect. Because as you said, like when you start showing symptoms, sometimes it's already too late, and any neuroprotective treatment, it would be very hard for it to restore the system back to the beginning. So that's why we are trying to center on this prodromal phase in order to find a therapeutic target that may work at this moment, with the help of all the biomarkers studies going on, in order to be able to translate this into the patients and to be able to give an effective treatment at the right moment. That would be the...
No, it's very true. Like, definitely, because I only do stuff in humans, is that you have someone who's progressed very far in the disease, and still there's subtleties that aren't there, and it's too late. So that is actually a great point to introduce our next speaker. And if there's time, maybe all three speakers and myself will have a chat, because Thomas Emmer will talk about the possibility of using neuromorphic architecture to model this kind of thing. As I said, on my own side, I've done a lot of Parkinson's work, but actually I'm a mathematician, so any modeling, anything like that, especially the neuromorphic, is very exciting stuff. So to introduce Thomas, he has a PhD in biophysics and electrophysiology, and he has been at the Merck Group for 20 years. He is the co-founder of the Neuromorphic Interest Group, which talks about neuromorphic properties and how we can use neuromorphic to model different diseases, and also use it to better understand the brain. And I'm going to include a link to a podcast which you can listen to about neuromorphic architecture from the work he's doing. So, Thomas.
Thanks for the introduction, John. I guess it's really complicated what we are doing. So first of all, maybe let me, I decided to share screen nevertheless. So let me just maybe share the screen. So you said you should see now my PowerPoint, is it? So I'm working for Merck. So maybe a first disclaimer here. So we have two different Mercks which are completely unrelated. So there is the what in the US is called Merck Sharp and Dome. Yeah, those are those guys, and we are the company Merck KGaA or Merck Group, which we have three different sectors. Yeah, and we are a science-driven company, and we have healthcare company, we have performance materials, and we are also known as Millipore Sigma. So we distribute all the stuff you need in the lab. So that's us. And in this we have in our semiconductor branch, we have presented at the CES Consumer Electronics Show 2020. We have identified neuromorphic technology as one of the groundbreaking novel paradigms. And of course, many people from the semiconductor are rather looking what you can do, how you can improve in-memory computing for faster image recognition, efficiency, etc. And I myself am stupid enough to be a troublemaker. And as a troublemaker, we are currently investigating how we can use neuromorphic technology for developing novel tools for the research community and also for the sake for the benefits of patients. And in there currently we have 3 working hypotheses, so that's under discussion. So I'm just really under discussion. So it's work in progress. So therefore we have our coming out and ask for feedback if it's nonsense or not, or looking also for partnerships. So there is of course one thing which could be a different kind of biomarker. So the pharma industry is very much focused on wet biomarkers, so everything that is wet. Eventually, we have novel ideas, variables-driven infrastructure using neuromorphic infrastructure to really monitor clinical outcomes that we hope to have from treatments. This could be drug treatments, but it could also be therapy, other therapies. And also maybe interesting also for Parkinson and also for multiple sclerosis, that we have early identification of an onset of a hidden disease. So the earlier we get in, specifically for MS, and we give one of the drugs that are available that prolong the disease progression. So the earlier we stop the disaster going on, the higher is the disease, the overall survival time. I mean, that's the ethical viewpoint of it. And currently there is not much that we can do to detect the onset of MS. I mean, we can do NMR, but we need to do this really, I mean, it's painful to do that, or lumbar puncture, etc. So that's one thing. If we could neuromorphic infrastructure, what does this mean? We don't know yet, but definitely it might be variables with in-memory computing, etc. So the next step would be maybe here jumping to the third one in sequence, that we have neuromorphic prosthetics and implants, which is we have the ability to use spiking networks, which record, I don't know, the signal from the lumbar spine or whatever in a normal person. And then if I get a fit or whatever, I can have an implant that can intelligently and autonomously adapt to this switch. I don't know Parkinson too much, but maybe, we don't know. And have something like a complement, like a heart pacemaker, something like a neuro pacemaker, if this makes sense. So this is another direction we want to investigate. And something which is close to my heart, and this is really far out, we have been discussing with lots of people in the neuromorphic infrastructure here in Heidelberg. We have a very fantastic infrastructure called brain scales. So they really build with analog chip technology, they really build from the synapse up to the cortical network. Then we have with Steve Ferber and his Spinnaker, we have another approach. They start from the synapse and the functional synapse up to mouse brain. I think in Dresden they are now working on the second generation of this. We have lots of approaches, and my idea is that we could, and this is for debate, that we could have really multi-scales, so that we work on the membrane level with ion channels to check drug interaction on the ion channel level, and not do and then rebuild the whole neural infrastructure based on the anatomy atlas that we have, for example, in eBrains. I mean, we have all this data, and bring this really together as a novel research instrument to do the following: that first of all, we test if our hypothesis works. If we say we know how MS works, so let's just invent a drug that mimics the disease progression, or that mimics the inhibition of the disease progression. And I think the challenge is that, or the hope is that these things are more or less that I'm emulating the disease. So this is not I'm simulating it, but I'm really emulating it. And the difference is that I'm just doing another thing. So instead of having a human in carbon-based, I have an artificial human or organ or cell, depending on the scale, in silico. And it's just behaving as is. I mean, it's really behaving as is. And there come, of course, lots of problems. For example, if we look at fatigue, I need to, first of all, train the baby and then grow it and then feed it and then check if it's fatigued. I think that might be not only an ethical problem, but also a technical problem. but the proposal, I think we should investigate as a whole community, from neuro researchers, rehabilitation, drug development, performance materials guys. Are we able to bring such a thing to life? Maybe just to avoid the typical case that I have an idea from a disease. I build my animal model, I build my knockout mouse, I've put my soup on top of it, and then I go into human and I just see it doesn't work, because the human is different. It's not that I'm hitting out the knockout mouse, but I have a real human. And yeah, I mean, that's my hope. And I know that it's very far away. And nevertheless, I think we, as intelligent community for neuroscience researchers and performance materials, just starting the discussion. If you from a research say, if I would have something that mimics the, I don't know what, L-type potassium channel, L-type calcium channel here with my something, eventually we could go together with neuromorphic infrastructure developers and say, we have this material, we have intermolecular is a part of our ecosystem, and we really are chip developers. Yeah, so we know what to do. So it not alone, but we might have compounds that are interesting for others who then build something for you. So that's the network approach. And I think also looking at the time, this is where I stop presenting, and really look forward for questions or destruction criteria and say this is fully nonsense, it doesn't work, or also just asking for help or further input.
Thanks so much, Thomas. I've had a very similar but not thought through opinion like that about the neuromorphic architecture, is that I know, and I've only read periphery around it, but it does make sense to think of it as developing this from modeling these kind of diseases and being the bridge between animal models to primate to non-human primate. So it makes sense to me. And like as both you and Josefa said, that early period is so difficult to understand. It's a period that I'll talk about myself because maybe I'll be disregarding that. But a lot of people ignore the early, like we all want to get to that stage, but the reality is to see differences between groups, you have to look at a late stage as opposed to an early stage where it's subtle and it's hard to see. And if you have models like mouse models or the neuromorphic architecture, you can actually do development and you can see many strands of changing something, see how things develop. So like, yeah. And also neuromorphic, like, I don't know how easy it is to work with, but it's not so hard, is it? Or how are they to work with? Like, I have a colleague.
There are, of course, different technologies. So I have not programmed it yet myself, everything. But I mean, it's like programming a computer. So there is, of course, this main difference of spiking neural networks, which everybody thinks is interesting. So also from an energy efficiency. So you have equations, and then you feed them equations in a so you can do neuromorphic modeling on a in silico classical computer. So this is what things are running at. And of course you can mimic really the physical architecture of the synapses at least, and give them a functional memory, or you give them a function how they react to input channels, and then you let it. You build the network. And I think that the real challenge is that most of the interesting things are unfortunately or fortunately emergent problems. even if we understand them in theory, and then we let them just run and not predict, but we really let them run. And then for sudden things, we have life, or the leeches capture something and they build a whole new organism from emergent, and we just don't know. And this is my hope, that we could use these kinds of tools to emulate things and detect stuff we have not been able to foresee, and specifically putting patient safety first. I mean, that's what we as a drug, the drug company, part of our company, is doing. Yeah, you have something, and to the very best knowledge, you assume each and everything works fine, then we let the thing run, and we detect after 10 years, ooh, something happened. I mean, definitely it's not only the drug and the neuro, so it's also the environment influencing, but there is this feedback loop, which is, I guess, unfortunately, extremely complex to model, but at least we could identify the players and say, we have no data and we let it run and we see it doesn't fit. So there must be something on top, and then we can identify novel levers.
And that complexity also comes with, for example, Parkinson's. It's not a single genetic disorder, it's many different, and that makes it so hard because many different pathways gets to the same behavior, but there are very different routes, and therefore modeling of those different routes is so important, because for different people, the turning point, and even within MS, for example, like different people relapsing, remitting, you know, a long time they don't have anything, and then they someone who has the same diagnosis is, you know, affected very aggressively early on, and you know, finding out what environmental or genetic differences or what things to look for is so important. So now we'll go on to our final speaker of today, which is Pascalina Guida. Pascalina is studied psychology, and now she's a PhD student in neuroscience in the Autonomous University of Madrid. In the center, they're developing, her thesis is integral neuroscience with movement disorders, specifically Parkinson's disease. And today she's going to characterize motor and cognitive habits in healthy participants and in Parkinson's disease patients. Thank you, Pascal.
Thank you, John, for your presentation. I'm going to share my screen. Can you see the screen? Okay.
Yes.
Okay, perfect.
Yep, perfect.
Okay. So, as John said, I'm a PhD student in neuroscience, and the main theme of my thesis is the behavioral and functional characterization of the natural motor and cognitive habits in healthy subjects and Parkinson's disease patients. As my colleague Josefa explained very well before, the brain has two major systems to control movement and behavior. One is the goal-directed system, and the other one is the habitual system. And these two systems has been linked to two different corticostriatal loops, specifically the goal-directed behavior, the goal-directed system is connected to the associative corticostriatal loop, while the habitual system is connected with the corticostriatal sensory motor loops. And as Joseph explained too, this part of striatum is the part that is more affected in Parkinson's disease. From the early stages of the disease. So when we approach to realize this study, we realized that we have three important challenges in the study of habits in human that we have to front of. One is the need to connect the real life habits with one studied and created artificially in the lab. The second is understand if there are any differences in how automaticity develops in motor versus cognitive tasks. And finally, very important point for our center is understand the role of motor and cognitive habitual behavior in Parkinson's disease. So the aim of this project is the evaluation of motor and cognitive natural habits to understand the differences in motor versus cognitive tasks and the influence of the early dopaminergic cell loss on habitual behavior. So to reach this aim, we have developed a behavioral protocol composed of these three parts: the neuropsychological evaluation and two computerized tasks. One to assess the cognitive component, and the other one to assess the motor component. So we have started this project, and these are the first results that we obtained. And for the neuropsychological evaluation, we can see that there are no significant differences between groups. So patients and healthy control are pretty similar in this part. For the cognitive component, so to assess a natural cognitive habit, we developed a go-no-go association task to measure an implicit automatic bias as a tendency to associate something familiar with positive concept, with positive words, and something that is unfamiliar with negative concept. We know that a bias can be defined as association between concepts in memories, and that these associations are automatically activated in presence of trigger stimulus. So we thought that a measurement of an implicit bias could be considered as a social cognitive habit. So we developed this task. It has 4 go conditions, and as the measurement of the implicit bias, we expected a tendency to respond more quickly on blocks that are congruent with bias compared with the ones that are incongruent with our bias. These are the results, and we can see that there aren't significant differences between groups, but there is a congruency significant effect, which means that independently from the Parkinson's disease group or for the healthy control group, the subject performed more quickly in the congruent block that in the incongruent ones, and the same happened for the mean of errors. They had less errors in the congruent block than in the incongruent ones. And last, we thought that a natural motor habits could be measured using handwriting. So a series of writing tests were collected from the more automatic option, like signature and writing in Spanish, to the more complex and goal-directed condition, like writing in other languages, like Polish and Greek. We recorded these variables, and specifically the last 2 are the more important to evaluate automatism in handwriting. And specifically, lower value of these 2 variables means greater level of automaticity. And these are the results of this task. For the duration, horizontal amplitude, and vertical amplitude, we can see significant differences between groups, but we can see a language effect. But when we move to the variable more connected with the automatism, we can see that there are significant differences between groups, specifically between the variables, the conditions that are more habitual, like signature and Spanish. In this case, this means that healthy patients with Parkinson's disease showed less automaticity in these variables, in these conditions. So finally, from these results, we can conclude that at neuropsychological evaluation, there is no significant difference between patient and healthy control in habitual components. In the cognitive task, we can see that in the C group, in the 2 groups, there are the same pattern for which subjects are faster in congruent condition than in congruent one. So there is no significant difference between patients and healthy control in the natural cognitive task. And finally, we can conclude that PD patients show less automatism in the execution of habitual condition, of habitual motor condition like signature and Spanish in our task. So a preliminary conclusion of this study is that in the comparison between patients with Parkinson's disease and healthy control, there are more differences in habitual motor behavior than in cognitive one. And this is all. Thank you.
Thanks so much, Gasolina. That was a very interesting talk. And once again, if you have any questions, put them in the chat or Q&A. But that's a lot of work at the beginning of your PhD. And I have to say that once again, this just sounds like me talking about my talk later. In our own work in people with Parkinson's, we've had very similar results. Is that simple behavioral tasks like go, no go, we see similar response times. It is when we get them walking and doing tasks, is where we start seeing divergence, where there's more stress upon the system. And we have the talk later on, but I published this earlier as well. Even with the simple go, no go task, while they're maintaining the same level of reaction time, they're recruiting a lot more of their motor cortex to compensate for that loss of automation, is what you're describing there. And so that's like, it's such a simple thing to do, handwriting. It's such an elegant solution to illustrate it. I really love that. Like, there's a paper recently saying, defending the that people should do behavior. Behavior is what drives forward, not electrophysiology. And I think that's a perfect example of it. You can do, like I had, for my own, I had a VR, I had to do all these things. And instead I could have just got them to write their name to see if there was a difference between automation and non-automation. It's such an elegant way to approach. But once again, it kind of bring it back is, how far along are these patients in the progression of their disease?
No more than 2 years. So they are
still
really
quite early.
Yeah.
Yeah. But once again, they are showing symptoms, which means, as Josefa said, like, that could be 12 years they've had the disease.
Totally.
And it's a very indirect measure, is years since diagnosis, because you could ask, you can, and I know with the neurologists I work with, there's also years since you believe symptoms onset and things like that. And it's a very fuzzy time period, you know?
Yeah. Actually, there are some patients that have a diagnosis or 1 month or 2 months, but when we talk with them and ask for the onset of the symptoms, it could be years before. So.
Yeah, absolutely. You know, sometimes you don't notice a subtle thing. Like, some people don't realize they have a tremor because it's a slow progression, you know? So it's these kind of simple tasks, and like a lot of tasks that people do, or you can learn or whatever, but something like your handwriting that you do all the time, like your signature, it doesn't matter how many times you ask the person to do it, because it's meant to be automated, and that's a very difficult thing. But similarly, and once again, like with diseases like Parkinson's or the others, people misrepresent what could be a cognitive issue, but it's not a cognitive issue, it's a motor issue that's impacting cognitive response. And so it's these subtle differences that are so important. So yeah, and then as Thomas was saying, like, and we'll have that progression between onset symptoms and how to know what point it is yeah, no, well, I've thoroughly enjoyed all 3 talks for 3 very different perspectives, but actually all kind of addressing now the guy named Dan Goodman, who did the algorithm to match things, he'd be delighted to hear that this works. So maybe it's random chance. Don't tell him I said that. This isn't being recorded, is it? But things like that. So,
I would have a question, John, if I might.
Yeah, of course.
Is there evidence that if you do, I don't know, motor coordination training, that you can strengthen the coping mechanism of the mental decline, or?
There is some evidence, I think, that if patients do some kind of motor training, it can help to the cognitive decline too. Yeah, I think that there is some evidence of this.
That's cool. I mean, it's a whole new argument for doing sports.
Oh no, like I always, once again, I'm a mathematician who works with neurologists, so don't take my word for it. I'm definitely not that kind of doctor, but I would argue that people like yoga and things like that, because what happens with a lot of freezing a gait, for example, or a lot of these things, is the feedback loop isn't working as well as it should. And if you walk around in your shoes, you get very little feedback. And therefore, it's better to train that. And even having that motor feedback is definitely going to benefit other areas. Like, the brain is super connected, you know? If you can stop the decline in one area, it's going to impact the other areas as well. Because that's like the one thing, shoes are great, but And I'm not saying people shouldn't wear shoes, but you do lose a lot of somatic sensory. So a lot of my work is self-motion perception, but you lose a huge amount of somatic sensory and proprioceptive information by wearing shoes. So
I would need to talk offline later with you or John on this one, because my little son has Down syndrome and also a lack of proprioceptors. So I think that there's a very strong correlation between.
Oh yeah, and a lot of disorders have this early, like with Parkinson's, we were showing I did some work on multi-sense integration in Parkinson's, and you know, you have this thing is that it's not the sensory system that you think is the issue, it's the other part that's the issue, and how they interact and those long distance connections. But it's same with and my work with autism, you know, sensory processing, proprioceptive, it's more prominent because you can see when someone's not feeling something. You can't see when someone's not seeing something. Sometimes it's, you know, well, we're going to have to finish. So the next session can start. I'd very much like to thank our panelists, Josefa, Thomas, and Pascalina. And of course, I'd like to thank Taha, who by coincidence as well, he also does PD research. So we could have just called this a PD session. So thanks so much to Taha. Thanks to everyone for coming.
Thank you.
Bye. Thanks so much. Bye.
Thanks. Bye.
Thanks, everyone.
Bye.
I will end the session in 5 seconds.
Okay.
I can start.
Okay, so let's get going. I introduce the speakers. It's a session that is quite interesting. Well, we're missing the first speaker. Let's see if she joins us in a few minutes, but we will go with the second talk. And the session, in theory, at the beginning was going to cover several topics on memory and cognitive control, as well as learning. So it's quite a large and wide interrelated topics that I think it's going to be a fun session. So we have a first talk, which is the interaction between associative learning that mediate initiation and inhibition of responses that we are going to miss or maybe not. She may join us. And then the second session is the neural signatures of amygdala hippocampal interactions during emotional memory formation in humans, which relates quite nicely with the third last talk, which is a learning replay mechanism that is happening while we are learning actions using MEG in humans as well. So anyway, I would love to get as many questions as you guys want. Please put them in the chat. Any question is welcome. Interrupt. There are no silly questions, so anything is welcome. And that will make a more interactive and fun session. So let me introduce, and let's start with Dr. Manuela Costa. She is a postdoc in the clinical neuroscience laboratory of the University Polytechnica in Madrid, Spain, and she will speak us about amygdala hippocampal interaction during memory formation. So off to you.
Can you see my screen? No. Now should be fine. Okay. Hi, everyone. I'm Manuela Costa, postdoc at the Laboratory for Clinical Neuroscience in Madrid. I'm very glad to be here today at this conference for presenting the last results about memory formation in humans. Sorry. Can you see the screen?
No.
Sorry, it's just the slide didn't change. Is it fine now? All right. Okay, so as we all have experienced, memory of emotionally arousing events tend to be more vivid and to persist longer than neutral ones. But why this happens? Well, we know the amygdala is an important area for emotion regulation, so it's not surprising that the amygdala also play a fundamental role in emotional memory formation. Accordingly to the memory modulation hypothesis, indeed, arousing experience activate noradrenaline release that will modulate memory in other brain regions, such as the hippocampus or the cortex. But which is the mechanism of amygdala and hippocampus communication during emotional memory formation? This is the main topic of our study. Previous findings have suggested that amygdala-hippocampus communication might occur via coordinated oscillatory activity. But today, I will try to show you that other mechanisms, such as the exact phase at which the coupling occurs, can be important for emotional memory formation. For this study, we recorded electrophysiological activity from 15 patients recorded at the Ruber Hospital in Madrid and at the University Hospital in Zurich. All patients had electrode implanted in the amygdala, and 8 of them also had electrode implanted in the ipsilateral hippocampus. Patients perform a memory task. The first day, they saw either unpleasant or neutral scenes, and they were asked to make an indoor and outdoor judgment. After 24 hours, we make the retrieval, and patients were asked to say whether they remember, they were familiar, or they don't remember the picture. We start analyzing the behavioral response, and in line with previous findings, we actually found that recollection performance was higher for emotional stimuli compared to all other types. In other words, patients remember better the emotional items compared to the neutrals. So now we can look at the oscillatory activity in both structures. I want to stress that we focus on the subsequent memory effect, meaning that we divide the trial at the encoding whether they were later remembered or forgotten. And we also collapse familiar and forgotten activity. So looking at the amygdala activity, we actually found a fast gamma oscillation that was associated with successful encoding of unpleasant scenes. This result was consistent when we look at the 8 patients that also had the electrode in the ipsilateral hippocampus. By contrast, when we look at the hippocampal activity, we actually found that the gamma response was associated with subsequent recollection, independently on whether they were emotional or neutral. So just to summarize, in the amygdala, we found faster response starting at around 400 milliseconds, while in the hippocampus, we found a slower and later response that starts around 500 milliseconds and that was not sensitive to the emotional content of the scenes. Now, having characterized the activity in the amygdala and the hippocampus, we moved to study the coupling between these two structures. From previous study, we know that there is, during retrieval, they found amygdala-hippocampus theta synchrony in rodents, and in non-human primates, during an aversive learning task, they found a flow of information going from the amygdala to the cortex, still in the theta range. In our dataset, we didn't find a significant difference in the low frequency across conditions, but as you can see, theta was present in all trial types. So we calculate coherence and Granger causality analysis. I mentioned already that we didn't find any type of coherence, but when we look at the Granger causality, we actually found that the amygdala theta leads the hippocampal theta when patient viewed emotional scenes, with a lag of around 24 milliseconds. Phase amplitude coupling has been proposed as a mechanism for facilitating memory formation. So we reasoned that we could find a difference between successful and unsuccessful encoding of unpleasant scenes. we perform the modulation index between the phase of the amygdala theta and the amplitude of the hippocampal gamma. But actually, what we found is that phase amplitude coupling was evoked to all emotional stimuli, but was unrelated to memory. So we reasoned that modulation index is actually not sensitive to determine differences in the preferred phase at which the gamma coupling occur. Look, for instance, this example. These are two examples of phase amplitude coupling, but in one case, the peaks occur in the peak of the theta oscillation, and in the other one on the trough. So we develop a new matrix that we can call phase amplitude coupling opposition index, which quantify the strength of phase opposition between two conditions. With this approach, we actually found that the magnitude of gamma amplitude was concentrated at different phase bins between successful and unsuccessful encoding of unpleasant scenes. Here on the x-axis, you can see the bins of theta, and this is the amplitude. So you can see that successful encoding of unpleasant scene, that is in red, is more localized in this preferred phase compared to emotional scenes that were not later recollected. So in other words, this result means that the precise amygdala theta phase at which the hippocampal gamma peaks occur is important for successful encoding of unpleasant scenes. Just to summarize, if you remember, we found that amygdala gamma response were selective for successful encoding of unpleasant scenes. And now we are showing a phase-dependent coupling between amygdala theta and hippocampal gamma. I want to stress that if there is phase amplitude coupling opposition, also there should be a delay between the two gamma oscillation. So we explored these relations, and in particular, we calculate a correlation between the peaks envelope of gamma oscillation in the amygdala and the hippocampus. As you can see from the plot, we actually found a significant transient coherence when the amygdala peak precedes the hippocampal ones. And finally, we found that there was a proportional lag during successful encoding of unpleasant scene between transient coherence, that you can see here, and the preferred phase delay, that means the result from the phase amplitude coupling opposition converted in the delay, showing, as I said, that there was a proportional lag between the two structures. So I hope that I convince you that emotional memory formation is actually phase dependent, and particularly that the specific phase at which the coupling occurs is fundamental for this process. Moreover, with our data, we show how the amygdala is influencing hippocampal oscillation during emotional memory formation. Before ending, I want to thank my colleagues, my supervisor, Brian Strange, Diego Lozano Soldevilla, that is also first author in the paper, all my colleagues, the hospital, and the patients that took part in our experiment. And thank you for watching.
Okay, thank you very much, Manuela. So we don't see many questions, but while people are making up their minds, well, it's been a wonderful talk, and I have a curiosity really. I'm trying to know a bit more of these mechanisms that may be altered in some pathologies that have over-representations of, for example, bad memories, let's say, anxious people, or I don't know, different types of diseases that may have too much of an enhancement of a bad memory. How would this mechanism work? Would the hippocampus compensate, or what would be the way out?
Yes, thanks, Ignacio, for your question. Actually, yes, we think that our result can be relevant for some pathologies, such as the post-stress traumatic disorder. I mean, remembering negative experience is important for survival, but as you said, it can become problematic in this type of pathology. So we think that our mechanism can be interesting, especially related with the possible stimulation that can be applied to this type of patient. There are already some cases. So I think, sure, it's relevant for this type of, for the clinical aspects.
Okay. We have, okay, so that's quite interesting. In fact, I see there are no more questions going on, so we have now, we should move on. We have swapped the order of the talks for those that joined later, and Nilosmita Banerjee is with us. Hello, Nilosmita.
Hi. Hi.
Yeah.
Can
Sorry
you,
for the confusion on the time schedule. Are
yeah,
you fine?
that's fine.
Are you fine to start now your talk?
Yeah, I am. I'm fine to start. So shall I just start sharing my screen then?
Yeah, please do that.
Okay, perfect. Right.
So while she's building up her system, Nilosmita Banerjee is a research assistant at the University of Sheffield in the Cognitive Ability and Plasticity Lab. She will talk us about modeling behavioral task, which is an inhibition task, a stop signal task, and how it interacts with associative memory. So please, Nilosmita, whenever you're ready.
Yep. Can you see my screen though?
Yeah.
Perfect. Okay, cool. Right. So, hi everyone. I'm Nilosmita Banerjee. I'm currently an MSc graduate in cognitive computational neuroscience from the Department of Psychology, and I'm going to talk you through my MSc project that I've recently worked on and submitted just about a couple of months ago, and it's titled "Modeling Appetitive Disinhibition in a Modified Stop Signal Task: A Computational Model of Associative Memory Initiated Disinhibition." Right, so today we're going to cover a bit about the introduction and the background, the research gap and the current aims of the study. We get on to looking at the methods, the results, and then just concluded. Right, so we looked at inhibition. So inhibition is one of the most important executive function that enables us to efficiently drive around in our environment, make optimal decisions, and it's important for tackling goal-irrelevant behavior. And response inhibition has been studied for long in cognitive psychology and studied using the putative behavioral paradigm called stop signal task. Many mathematical models have been proposed in the past, but the horse race model happens to be one putative model that's been used long to quantify and predict the performance of participants' inhibitory control power in the stop signal task. So this is a schematic of a typical stop signal task, which is used in most of the cognitive psychology and also neuroscience paradigms. So just to run you through a little bit, every single black box is basically a computer screen that appears to the participants, and the arrows, as you can see, are what we call the neutral stimuli. They point in either of the directions, right or left, and the participant is supposed to press the keys designated to the directions when they're doing the task. In a stop signal trial, the participant will be given a couple of co-stimuli, which is pressing right or left, and will be interspersed with a stop signal like the cross in as shown in the schematic, when the participant is signaled to basically inhibit their prepotent action to the particular task. This is a more descriptive schematic, which also shows the trial and the participant reaction time and what constitutes of it. Right. A large body of research has demonstrated that the ability for appropriately inhibiting some goal-irrelevant response, or that prepotent response, terribly impairs when a participant is asked to do the same in an environment which is called appetitive stimuli or appetitive choices. And this has been captured by a body of over 30 years of research in experimental psychology studies that have used a modified version of the stop signal task, but with appetitive pictures instead of the neutral sort of the arrows that we saw in the schematic before. So this is a schematic which shows that it exactly is the same. The task is not different in its structure. The only thing that changes perhaps is the stimuli that is used instead of the neutral stimuli. They've used the appetitive stimuli. In this case, we've shown chocolates. This is actually a paradigm we've picked from, the schematic is picked from a paradigm which was used in some actual experimental psychology studies done. Right. Now, the research gap that we found was that every single model, mathematical model that was developed so far to explain the behavior was descriptive in its nature. However, the standard sort of the process models that explains the mechanisms of inhibition that is involved in a stop signal task was proposed only very recently by Bouchard and colleagues in 2007. The problem was that although this particular interactive race model explained really well what was going on in a stop signal paradigm in terms of explaining the inhibitory behavior, they did not really capture, or the parameters of the model did not really capture or explain what was happening in the impaired response inhibition phenomena that we saw in these modified stop signal tasks. So in simple words, it did not really explain what was going on when the participants were presented with an appetitive cue in a modified version of stop signal task. They were showing this inhibition, but the model did not have any parameters that could capture why this was happening or how this was happening. So that's where the aim of the current study came in. So the current study aimed to build a process mathematical model similar to the interactive race model, or what we did in this study was build on the interactive race model of appetitive cue initiated disinhibition. So we were trying to capture the disinhibition element that was observed in the modified stop signal task with the appetitive cues. And theoretical frameworks, a little bit of, because this was a very theoretical project, we tried to base all our augmentation processes on what the theory had to suggest, and the literature showed that the theoretical frameworks have indicated that such a disinhibited response or a going behavior or approach behavior towards these appetitive cues is usually an outcome of recalling these approach behaviors from one's associative memory. There's a lot of research even in neuroscience that focuses on the elevation in the level of dopamine, for example, that sort of happens when appetitive cues are presented, which has been considered to be an instantiation of associative memory recall initiated behavior or going approach. So in order to therefore address this, we built an augmented version of the interactive race model of action suppression with an associative recall free parameter, which we called lambda. Lambda instantiated the associative recall of the approach behavior in our current model. So the methods we used, we tested 3 different parameter variations for augmenting the associative recall parameter lambda in the interactive race model of action suppression. And we formalized 2 different lambda parameters, one of which was the lambda go, which instantiated the approaching behavior or the disinhibition that we observed in the participants for the modified stop signal task, and then the lambda stop, which instantiates the inhibition behavior, which is usually what is expected. So before starting to test the variations, we started with the actual model. So this is the neutral we called it the neutral variant because it was well simulating the behavior that we observe in a non sort of non-appetitive condition. So it was because it was explaining that, we started off with it and we called it the neutral variant. So just to walk you through this original model a little bit, the first equation models, the parameters of the first equation models the the going behavior, and DA-Go basically stands for an accumulator. So we have formalized the going process as an accumulator that starts from, that races from a particular starting point, and it races towards a threshold theta. So every single parameter in the first as well as the second equation actually conceptualizes and formalizes that sort of racing between the two processes of pressing a button or not pressing a button, for example, in the case of a stop signal task, so going or stopping. So DA stop, the equation 2 basically formalizes the stopping process. And I just want to draw your attention to the most important part of this particular equation, which is the part of the beta parameters. If you can see the beta stop and the beta go. These are the important parameters in the equation specifically because they talk about the interaction that the two processes have. So for example, if there is a go sort of a trial where there is no stop signal presented, then the participant will have no interaction of the stop unit. So the beta go will not be interacting with the beta stop. So there will be no buddy sort of actually stopping the process. However, if there is a stop process, there is an interaction. So the beta go will interact with the beta stop, and that will sort of stop the process, and it will not let the going process reach the threshold theta. So those interaction parameters were the most key sort of parameters to focus on in the process of the augmentation. We do have model schematics in the further slides that will make this more clear. So this was the parameter variation 1. We call this the model 1, and we augmented the lambda parameters such that they actually affected only the part of the model of the interaction bit. So you can see that the mutual inhibitory interaction between the go unit, which is the disinhibition behavior, and the stop unit, which is the stopping behavior, those accumulators interaction, mutual inhibitory interaction was affected and or biased in a way with the parameter values of lambda stop and lambda go. So this was the first model. The second one, we tried to, because we were trying to rigorously test and see every single combination and see what was actually giving out the dynamics of the reaction time that we observed in the modified stop signal task, we tried to affect the mean threshold. So basically the growth of these accumulators towards the threshold and not the interaction in this case in the parameter variation 2. In the parameter variation 3, we tested and we tried to affect both the mutual inhibitory interaction as well as the mean growth rate. And these were the parameter constraints we had. The results led to this. This was the first architecture. So in here, you can see that the associative recall units are grayed out. So they're not playing a role and they're not biasing the processes. And this was the first parameter variation. Here they are green and they're affecting the mutual interaction. So you can see that there's a bigger purple circle in correspondence to the lambda go, which means that the go is actually in a way inhibiting the stopping process, and the stop process does not have a lot of impact on the go process. We had the parameters affecting the mean growth rate in this architecture. And we hoped to see the dynamics where the lambda go would have affected stop process more. And then we had both together. So the simulations ran for the first parameter variation in the parameter constraint settings, which are presented here, were the only model which actually led to the behavior that we were observing. The simulations resulted in the dynamics. So we had, this is a probability of signal response, which basically means the probability of responding on a stop signal trial was the highest for the first parameter variation model. And this was another comparison. This is only a subset of the simulations results, because there are a lot of other results, simulations of other dynamics that we looked at as well. But the red bar in here basically shows that the SSRT stands for the stop signal reaction time, and it was exactly the same. The simulations showed exactly similar ranges of reaction times for the first model augmentation. So in conclusion, we can say that we've actually been able to successfully build a model that showed the interaction between these two elements of inhibition behavior and associative memory. And in future directions, we are now currently working on, so we have collected some experimental data with modified stop signal tasking and psychology paradigm, and we're now performing some fitting of this particular data with the first model for the future directions, which is what we're working on currently. Yeah. Thank you.
Thank you very much, Smita. This was a nice topic. We are kind of running late for the next speaker due to the initial technical difficulties. So we don't have questions. I would pose you a few because it is kind of very much related to my research of interest. So I may email you, okay?
Yeah, sure, definitely.
And if any other person as well would like to email her, but we need to move on to the next speaker. Yeah. So if you can start sharing, Yunsel Liu is a postdoctoral fellow in UCL, University College of London, and he will speak us about the experience replay on mechanisms of learning. So Yunsel, whenever you're ready.
Okay, great. Thank you for the introduction. Can you guys hear me? Okay, cool. So thank you very much. So my name is Yangzi, and I'm going to talk about a recent work about linking replay to model-based learning. Cool. So now let's imagine if you are this rodent, and the only thing you want is this piece of cheese, okay? And things might get a bit complicated in a real-life situation. For example, you might start from all 3 locations, and again, the goal of your life is to get this piece of cheese. Now imagine in one situation, you start from the blue and you choose the left option and you get the cheese. That's great, right? However, if you want to be more efficient, you should also know what you should do when you are being put in the yellow or the green location, right? So that's saying I should know whether the other road is also leading to the cheese or not. And this does create a problem, right? Because the state and action and then the outcome are separated by both time and space. And this is a well-known problem called credit assignment in the reinforcement learning. And today I want to show you how in the human brain could we implement a model-based learning algorithm that's trying to solve this problem. And I think one potential mechanism is replay. So just a little bit background on replay. So replay is a well-documented phenomenon in rodents. So the play cells that are normally only going to fire at a given location will fire sequentially during rest, that actually recapitulating past or future experience. That's what we call replay. It's a suitable mechanism because it is internally generated. So it can quickly, and it's also time compressed, so it can very quickly bridge action and space that are, the action and outcome that are separated by time and space. All right, so how we do that? So the first thing we need to do, we need to design tasks that dissociate local and non-local learning. And here is what I mean. So let's say, again, I'm in the blue path and take this one, and then the path I just chosen is what I call local learning, right? You can just do that through try and error. And there are 2 other paths, I would call that non-local learning. It's non-local because you have the lead by passing the local outcome to the non-local actions. And the way to do it is what I termed non-local learning. And if this, and upon this problem, there is a second order question, i.e., with so many non-local experience in our life, how should we prioritize? How should we prioritize the memory access? Right? Should I replay more of first to the path lead to yellow versus the green, or vice versa? Luckily, this problem has already been solved by Marcello Marta and Nathaniel Dahl 2 years ago, based on reinforcement learning. So they figured out a lumping model by saying the way to decide which memory should I have access to first, depending on a product of 2 factors, One is called need, another called gain. And need determines how often did I encounter this experience again. And the gain determines how much actual reward do I get by replay the experience. And with close collaboration with Marciano and Nathaniel, also with Tim Barons and Redonen, we're trying to do that. We're trying to do that in humans. So in humans, this is the task they're doing. So we manipulate lead by gain separately. The lead is manipulated by the probability they're going to start at each of the arms. So at each trial, the participant be put in one of those arms based on those starting probability. And then the job of the participants is choose the path leading to the outcome, i.e. X and Y. And the reward probability of X and Y going to change independently, follow a random walk. So this is going to manipulate the gain. And the aim of this study, and then we can study priority. And then the aim of this study is to look at those triangles, i.e. priority, replay, and behavior. All right, so first let's talk about the model-based behavior. So we can define model-based behavior in a model agnostic way. So again, we already separate local and non-local learning. Let's say again, on the last trial, we start from the blue. And then in the current trial, if I have been putting in a different starting arm, if I am model-based, i.e. if I know the transition structure, I should more likely to choose this path versus this one, right? Because I already get a reward on this path. So this is what we can use as index of model-based behavior, while we can using some model agnostic analysis to detect that. And indeed, this is what we find. So the same means starting at the same arm as previous trial, diff means starting from different arm. So the reward means get a reward last trial, unarg means didn't get a reward. So this contrast is a textbook of model-free learning. So you do that through trial and error. And this would be an index of model-based learning, i.e. if I'm starting from a different arm, what is the probability do I going to choose the path that leading to the same outcome as the last trial? And you would imagine if I'm model-based, I would more likely to do so if I get a reward on the last trial. And this is indeed the case. And the difference between model-free and model-based in this task are indifferentiable, which means the participants indeed are really model-based in this task. Then we ask a second question. In the model-based behavior, do we see prioritization? We can further separate each of the non-local paths as high lead versus low lead or high gain versus low gain. If we compare the learning rate of the high high priority versus the low priority, we see indeed a significant higher learning rate for the high priority path versus no priority. So this establishes the first line of this triangle, i.e. the behavior, i.e. there is prioritization in behavior. All right, so let's now turn to the neural data. So again, so this task, we want to look at the neural mechanism. So first thing first, we want to say, do we actually can decode all those pictures? And this is not a trivial task because there are 16 states in this task, right? So because there are 18 pictures in this task because there are 6 sequences. Each sequence have 3 distinct stimuli. So first, we can show that we can decode all the picture pretty well with decoding accuracy peaked around 50%, while the chance level is below 10%. And again, peaked around 200 milliseconds, consistent with our pre-study. And then the second thing we want to look at is only look at the time when the reward has been received, because this is only time in this task where new learning can happen. And if we look at those time, we can say in general, we have 2 types of sequence. So the x-axis here are the time lag. The smaller the time lag means the faster the replay is. And the y-axis means the strength of the replay. And the dotted line is the permutation threshold. Anything that past that would be significant. So you can see we can identify 2 types of replay, one in forward direction. So forward indicating as the same direction as experience, while backward means in the opposite direction as experience. And it's interesting because this forward thing will replicate our previous study doing REST that we show a dominant forward thing at 40 or 50 millisecond time lag. So it's really, really fast. But this backward one is intriguing. It's slower, it's backward. It potentially may be related to non-local learning. Now, again, in this task, we separate local and non-local learning. So the first thing we want to say is, do we see a representation content difference in those two replay with respect to non-local and local representation? And indeed, we see this 30 millisecond forward replay mostly encoding the local experience, i.e. the experience you just go through. So it's indeed literally replayed, while it doesn't represent the non-local experience. However, the 106 milliseconds show a distinct pattern. It only encodes non-local experience and doesn't care about the local one. And it's interesting. Then we can look at the physiological difference, right? Again, we show the 30 millisecond thing is associated with a shape wave ripple power increase at 120 to 180. And this is consistent with our previous studies for the similar kind of replay. But we didn't see that for the 160 millisecond. And a few of the beamforming results, which I haven't shown here, suggest this 160 millisecond have a more cortical engagement as compared to the 30 millisecond. So this all of those indicating the 160 millisecond replay is another type of replay and is potentially suitable for non-local learning. And we're going to test that formally through a computational model. The idea is to say, does the inclusion of sequence going to significantly increase the learning, i.e. the learning rate we put on the replay, while already taking into account of the baseline that doesn't care about replay. And now we can say, do we say 160 millisecond increase non-local learning or 31 increase? So the answer is 160 millisecond sequence. The inclusion of that does facilitate learning. And that establishes another line of this triangle, i.e. replay does support and facilitate non-local learning. Okay, great. So the last question is replay actually prioritized so that it's going to most likely to replay the most policy informative behavior? Again, this can be translated in the analysis, i.e. do I replay more to the yellow path or to the green path, depending as a function of the product of lead and gain, right? And again, just to remind you, the lead is manipulated by the starting probability of the arm, and the gain is manipulated by the reward probability of each outcome. And then at each of trial, we can separate the high priority with low priority. And when we do that and collapsing all trials, we can detect a difference in the replay that's stronger in high priority paths versus low priority paths. And so we can say that, again, picked at 160 milliseconds, while this 30 millisecond thing, which is dotted line here indicated by this blue line here, is not significant at all. So this establishes the last line of this triangle, i.e. replay is indeed prioritized. It prioritized so that it first replay the most policy informative experience. All right, with that, I want to conclude. So we find non-local learning is prioritized to support model-based behavior, and we find reverse non-local replay at 160 millisecond lag help model-based learning. And it's both functional and physiologically different compared to this shape wave ripple faster 30 millisecond replay. And this 160 millisecond replay is prioritized for the most policy informative experience. And with that, thank you, and thank you all my collaborators, Kim, Nathaniel, Marciano, and Ray, and thank you.
Thank you, Yancy. Such a nice piece of evidence on reward and non-local replay. I'm very curious, but let's see if there is some of the questions. Yeah, there is a question by Lawrence Hunt. So I'll just read it aloud. He's saying, he's trying to map your forward reverse results onto what is found during SPW and theta sequences in the rodent. Are there any example of rodent backward replay during theta that you could relate to your 160 millisecond sequence? Is that clear?
Yeah, indeed. This is a really good question. So this is a question that buff us a lot. So the question is, so what Lawrence is trying to ask is, what is the correspondence of those human replay to the rodent one? And those 160 millisecond is intriguing because the 160 millisecond indicating a 6 hertz, right? So the speed is 6 hertz and is in the theta range. And intriguingly, in rodents, we also have those theta sequence. But normally, theta sequence is forward-looking, right? It's going in right in the direction of where you are, and it's continuous while the rodent is like moving around. It's not like going to be initiated by the reward. So so what we have found is more like the shape-wave ripples in rodents because it's initiated by the reward, and it seems to have a function of backpropagating the value. But indeed, because it's
okay, live stream's working. So we have like 1 minute till the start of hour. Don't forget to start slowly until people join.
Yeah, since it's I think it might be worth waiting if there are not enough people or not many people, because you don't have super long talks where you'll have to rush a lot to tell us everything.
All right, let me start now and have fun. Take care.
Thanks, Harash. All right, welcome everybody who is joining the session. This is session 3012, room 4, to make sure that everybody is joining what they want to join. We're gonna wait a few seconds to allow everybody to join. And yeah, we'll start very soon. Okay, so again, welcome everyone. My name is Alex Hernandez. I'm speaking to you from Madrid, Spain, and I will be the moderator of this session. Again, session 3012, room 4 of NeuroMatch 3. And I want to thank Arash, who is doing a lot of back-end work behind scenes. And yeah, we have here today a great line of speakers, young speakers. They are Thijs van der Plas, Mathilde Tristani, and Dennis Tuoku. And they're going to talk about neural networks, biologically inspired neural networks, neuroscience. So super interesting. I'm looking forward. Remember that if you like, you can tweet about this talks or NeuroMatch in general on using the hashtag NMC3. And of course, questions from the audience are really welcome and appreciated. This is an interactive session. That means that speakers will speak for about 5 minutes or so, and then we will have a longer time for discussion. So please use the Q&A tool and yeah, have fun, enjoy, and we're going to introduce straight away the first speaker, Thijs van der Plas. He did his bachelor's and master's degrees at the Radboud University in Nijmegen. And actually his work, his talk today is about that work, if I'm correct. But he's now doing his PhD at the University of Oxford with Adam Packer, studying the dynamics of artificial and biological systems. So welcome, Thijs. The stage is yours.
Thank you. Thanks very much for the introduction. Okay, I think you should see my screen now. Yeah, thanks very much. So actually, I'll talk about work that I did in Oxford with Tim Vogels, who is now at IST in Vienna, and Sanjay Manohar, who's still in Oxford. So main question of this talk is, how can networks learn to solve difficult tasks? So as we all know, both biological and artificial networks continuously try to learn to solve new tasks. And some tasks can be more difficult than others. And for artificial networks, various learning strategies have been developed to enhance learning. For example, things like dropout or different gradient descent rules all can improve learning. But these are very technical improvements that don't readily translate to the biological counterparts. So today I will talk about one of our most recent results, which is a different learning strategy, which we hope might relate more closely to biological systems. So I'll talk about two tasks today, which use the same trial structure. So each trial will consist of 4 stimuli, A, B, C, and D, where A, B, and C can be of one of 2 types, which I will indicate with the 2 colors green and purple here. And so on 75% of trials, C will have the same color, the same type as A and B, while on 25% of trials, it will have the opposite colors to A and B. And so our first task for the network is to predict at each point in time the next upcoming stimulus. So for this purpose, we use regular recurrent neural networks. And so they receive an input, which is the current stimulus, which is then, can you see my cursor? I think so. Okay, so if so networks receive the current input and then project to output neurons that predict the next stimulus. And we train these networks with backpropagation through time, which updates all 3 weight matrices of the network. And so this turns out to be an easy task for networks to learn, and they all like if we run this over like 100 networks with different initial conditions, then they all converge very, very easily. And so on the right, we see a plot of the convergence of the loss function versus the number of epochs. So secondly, let's consider a different task, which we will call a match-non-match task. And so this task is different. Here the network needs to report the trial type at the end of each trial. And so a trial can either be a match if C has the same color as A and B, or can be a non-match if the color has switched. And for this, we use 2 new output neurons that also include a feedback loop to allow the neurons to accumulate evidence over the course of the trial. And it turns out that this is actually a very difficult task to learn for most networks. So again, if we evaluate 100 different initial conditions, then we see that only some networks converge, but actually most don't converge at all. And so the average performance is pretty bad, and there's a large variability across networks. So this is a difficult task to learn for these networks. So how could we enhance learning of this difficult task? Well, as it turns out, if we add the prediction task to the loss function of the match-non-match task, so we get this combined loss function, we can actually enhance learning of the match-non-match task. So in this case, we use all output neurons, and we have increased the loss function to cover both of these subtasks. And if we now evaluate the loss function in 2 parts, firstly, if we look at the prediction part of this combined loss function, we can see that it converges at exactly the same rate of the prediction-only task. So the match-non-match task doesn't interfere with our prediction task. But if we now look at the match-non-match task component of this combined task, then we see that this converges much better than before. So in fact, all networks now converge for this task. So concluding, we see that by adding an easy task to a difficult task, the network can learn in a completely unsupervised way to solve this loss function in parts, and it will solve the easy part first at the same rate as before. And then once this has converged, it will start learning the difficult task, which can now also converge. And so we think it's interesting. I'm very curious to hear what your comments and suggestions are. And I think we hope that this could be applicable to a variety of tasks and systems in the future. So thank you very much for your attention.
Thank you very much, Thijs, for the presentation. Very interesting, very neat, nice slides also. We have actually one first question from the audience. Jake Stroud. I will read it for you.
Thanks.
Or Jake, do you want to go on stage and read it yourself? Hard to get feedback in real time, so I'll read it myself. Yes, that's fine. meaning I don't know if that means Arash, if you can. Okay, cool. Arash, can you turn the mic on? So I'm trying to wait for Arash, our backend host, to do this. I'm gonna try myself. Okay, I'm not sure how to do this easily, so I'm gonna read it myself in the interest of time. Sorry. So he says, I would guess that the poor performance on match non-match is because you have a vanishing gradient problem using backprop, or maybe the network isn't using your nonlinearity enough. The match non-match requires a nonlinearity. Also, the length of the delay period will make the task harder to learn using backprop.
Right. Yeah, I agree. I think this is the issue, right? That because the network only needs to report the trial type at the end of the trial, and therefore I think you get indeed a vanishing gradient throughout the task. But and we use a nonlinearity here, just like a restricted linear unit on the output weights. So I'm not exactly sure what the question is. It was a more comment, but yeah, I think I agree. But I guess this is one way to overcome it, essentially, right? By using, so learning is difficult. So by adding an easy task, we already sort of prime the network to recognize the stimuli, which makes then learning easier. But yeah, I know. Would you have a like another suggestion to overcome the vanishing gradient in this case?
Mm-hmm. So we're gonna try to turn Jake's mic on for better discussion. But in the meantime, there's another question by Raunak Basu, and they're asking, have you analyzed how the weights in the RNN change when the match non-match loss is added?
So I see. So I don't think so. So
yeah,
I mean,
I guess he's referring to basically how the weights of the model are changing during learning.
Yeah, right. It's no, I haven't. Yeah, I'm
okay.
I'm not sure. No.
So now I think
just to be clear, we start off with both functions, right? But I guess you mean the onset of learning of the match non-match task.
Yeah. I think we have now Jake on.
Okay.
And
yeah, can you hear me?
Yeah, perfect.
Yes,
perfect. Yeah, sorry, my question is complicated to ask in the text, but yeah, great talk. Thanks. I used to work with Tim and No Say. So I guess my comment is really that the problem is with backprop, right? That's really what the crux of my question is. So, for example, reward-based learning might allow you to learn the task better. Maybe having a stronger nonlinearity may allow you to learn the task better. And so, for example, LSTM units, won't have a problem at all with the task. And also the length of the delay period might matter, right? If you have a really long delay period, you're going to have a much worse problem with vanishing gradient. Whereas if you have a very short delay period, you're going to have basically no vanishing gradient problem. So I guess my comment is less that is more that it's a problem with backprop really, and that maybe not utilizing the nonlinearity as much. I don't know if you can sort of comment about that, or what your thoughts are.
Yeah, that's okay. Let's see. Yeah, that's a great suggestion. Okay, so I've tried a few other nonlinearities that all didn't work. Like for the yellow loss function, so to say, for the dimensional match only. But I haven't tried the LSTM. But yeah, but I think it's right that this is the issue, right?
Yeah.
But so I guess what I'm suggesting is a way to overcome it is to just add another task instead of changing the network architecture to be an LSTM, for example, or to change it this way.
Yeah, sure. And I think more realistically, these networks have already, learned many tasks, so they already have these stable modes. So they probably don't have as much of a vanishing gradient problem. But another part to my question is, have you tried reward-based learning? Because that will be probably more robust. It doesn't have a vanishing gradient problem. Have you tried that?
No, I haven't. Okay, that's a nice suggestion. So what would you expect in this case?
I think other people have studied this a bit, but it shouldn't have a problem learning a matched and non-matched task, basically. As long as your delay period isn't so long that all your neural activity just decays to 0, then obviously you can't really do anything. I don't know. It would take longer to learn, but it might not have that problem that it can't learn.
Okay. Yeah, I guess all these comments go in the direction of it would be interesting to make sure that the problem is so you're saying the problem is the type of loss, and we added these other loss combined and then it worked, but it would be interesting to make sure that actually adding the other loss is what is making the network work rather than any other potential issues with the optimization.
Yeah, I think it's also, it's also I think a good suggestion to instead of doing this match no match task, to maybe find a task that is really impossible to solve by itself, right? Rather than
right, yeah,
being dependent on the backtrack.
Yeah, that would be a good approach as well. Yeah. Yep. So, is there any other question from the audience? You have one more minute or so. So I was curious along these lines that I was commenting, like if you are planning to extend this kind of experiments to more complex tasks, actually, in the same paradigm, like.
Yeah, I mean, we're planning to. I haven't implemented more difficult tasks. So it's
just as future work. Like I'm curious about
exactly.
the future directions. Not.
Yeah.
Okay, cool.
Yeah.
All right. Since there are no more questions, I think we can move on to the next speaker. But I want to thank you again for the nice presentation, and also wish you luck with your PhD. And thanks the participants for asking questions. Also, Matthias Hennig commented on the chat that it would be good to try LSTM as well.
Yes, I can see the chat.
There are some other, maybe you can take a look at the chat. Great. There are some other comments for you. That's great. Thanks everyone for participating. Okay, so our next speaker is Matilde Tristani Farinha. Matilde is at the ETH Zurich doing her PhD in deep learning, neuroscience, and artificial intelligence. And today she's going to present her recent paper, Equilibrium Propagation for Complete Directed Neural Networks. Sounds really nice. So welcome, Matilde, on stage.
Thank you. Let me just share my screen. You can hear me well?
Yeah, very well.
Okay.
And we can see your screen now.
Yeah. Hello, everyone. Thank you for coming to my talk, actually attending my talk. So I'm here to talk about the research I've done during my master thesis, which was done under the supervision of Professor Sérgio Paquito, Pedro Santos, and Mário Figueiredo. In one sentence, what we did is basically propose a biologically plausible learning algorithm for recurrent neural networks which have asymmetric feedback connections. Before going further into the subject, I wanted to, sorry, I'm actually in the wrong slide, sorry. I wanted to motivate why these type of algorithms matter. So first of all, brains are great because they are actually the best learning machine that we know of. And we can kind of suppose that if we try to bridge this gap between neuroscience and machine learning, we could actually get better learning algorithms. In fact, looking into the history of artificial neural networks, that's actually what we can see. Second of all, the Moore's law has been failing for a few years now, and scientists have been looking for new types of hardware that could surpass this threshold. And these new types of hardware require new algorithms, and one of these type of hardware is neuromorphic hardware, which is inspired by the structure that we have in our nervous system. It tries to mimic it. And these type of algorithms, inspired by that work on energy functions or vector fields, are thought to be actually efficient for this type of neuromorphic hardware. Okay, so when we talk about biologically plausible, I want to explain a bit better what we mean with this. And we refer to certain characteristics that the model can have. For instance, a local learning rule for the synaptic weights, meaning each weight is updated based on the activity of the neurons it connects, having the single type of computation for the feed-forward and feedback passes of information, and also having asymmetric feedback connections. Then we can also consider that there is no like a strict restriction on the architecture, so no strict layer-wise architecture. And we should also be able to consider that we have sparse representations of information in the network and not just those usual dense representations that we observe. This model was inspired, or is actually a variant of equilibrium propagation with its in its version of asymmetric feedback weights, but it differs a lot from this previous algorithm because we introduce a new neuronal dynamics, which accounts for the weighted incoming stimulus to each neuron and also the outgoing stimulus of each neuron. And this has a neat biological property, which is that it preserves the grand mean firing rates in the network when there's no external stimulus. And this has been observed in vivo for the hippocampal neurons. We also drive some interesting theoretical sufficient conditions to guarantee that we have convergence in the inference phase, and that you can see better on the paper. And we also propose a new learning rule that accounts for the fact that we have these asymmetric feedback weights. And it's based on a numerical integration of biologically plausible weight dynamics, which has been previously proposed in the past. And it is biologically plausible because each update, the update of a weight depends only on the activity of the presynaptic neuron and the rate of change of the postsynaptic neuron. We consider that the architecture, the initial architecture, is a complete directed graph, so an unrestricted architecture. And because this is so unrestricted, depending on the task at hand, many of these connections are actually not necessary. So we also introduce a sparsity inducing method that removes these irrelevant connections. In this method, basically a certain weight can be removed with a probability taken from a Boltzmann distribution to find across the incoming connections of every neuron. So the idea is that the strength of a certain weight is proportional to its importance in the network at the moment. So throughout training, we are able to remove these less relevant connections. Okay, so we claim that this could possibly be a better learning algorithm because contrary on these simple kind of proof of concept tasks, such as learning the XOR, it is able to learn contrary to its previous asymmetric formulation. Down on the down plot, you can see the mean squared error. So basically the full green line, the full dashed line is the mean squared error for our model. And you can see that it goes to 0. I refer to the green line because that's the inference. The red one is not inference. And then for the previous model, you have the sorry, our model is the dashed line, and you can see that it goes to 0. And then for the previous model, we have the full green line, and you can see that it doesn't converge to 0. And we tried a lot of tuning on this. On the right, we have the decision maps that we obtain for our models, for simple logical operations when learning these operations. And the x-axis represent the inputs, and the color represents the value of the output. So dark blue is 0 and dark red is actually 1. Yeah. We say that it generalizes well because throughout training, we only give samples of 0s and 1s. But then in inference, after training, if we give samples in between 0 and 1, the model is actually able to classify these samples based on their proximity to the previous samples that it saw during training. So it's a very simple way to show that it generalizes. Below, we have the prunes optimized networks that we obtained when we train the network with our sparsity inducing method. And we can see that for some tasks, simpler tasks such as the AND and the OR, the model is actually able to capture the essential connections between the neurons. I forgot to say that the red neurons are the inputs, the green neuron is the output, and the bias neuron is the blue one. For more complex tasks, that does not happen. So regarding future implications, as I said, because this model can be seen as a continuous time dynamical system, it is well suited to be efficiently implemented in this new type of hardware called neuromorphic hardware. We can also look at this sparsity inducing method that we introduce as a network design tool, because these kind of optimized and possibly minimalistic network architectures could be used as an initial architecture for other learning algorithms, for example. And I think that's it. So basically, as Alex said, I'm currently a PhD at ETH, working as a researcher at the Institute of Neuroinformatics. And on the right, you can have the QR code to the paper for more information, to the GitHub code, and my Twitter tag name if you want further updates on my future research. Thank you. Let me know if you have questions.
Thank you very much, Matilde. That's really nice. I'm waiting for, I've actually, by the way, I put the link to your paper on the chat, and I right away copied the GitHub link. Are there questions for Matilde from the audience? Remember, you can ask on the Q&A tool or directly in the chat if you wish. In the meantime, so
can I ask a question? I can't type in the Q&A for some reason.
That's right. Yeah. This is a funny feature of Zoom. You can go ahead.
Yeah. Okay. I was wondering about your how do you sparsify the networks that you use? What pruning mechanism you use, or how does that work?
Okay. So at start you have this complete directed graph, and then at each epoch you define a Boltzmann distribution for every neuron, and this Boltzmann distribution is defined across the incoming connections of that neuron. So each weight will have a certain probability of being removed, inversely proportional to its strength. So the stronger the weight, the less likely it is to be removed, and then you kind of randomly remove a few weights at every epoch, and that gives us this type of. So it can reach this type of minimalistic network where it only has the essential connections. Yeah.
I see. So effectively you maintain the same performance or take a very, very tiny hit in performance while removing those extra connections.
Exactly, yeah.
Okay, thank you.
Yeah, the same performance. So this is all 100% performance. Like these are very simple operations, learning logical task, simple tasks, learning logical operations, but this is all results for 100% accuracy. Yeah.
Itamar Landau from the audience is asking if you can say again how the learning rule works.
Learning rule.
I think it would be good for.
Yeah. Okay. So the learning rule, maybe you're familiar with STDP, spiking time dependent plasticity rules. And the basic idea is that you update the weight based on the presynaptic activity of a neuron and the postsynaptic, the change on the postsynaptic activity of the neuron. So the change in the rate activity of this neuron. And in this case, a simple step would be to take the first state that you have in these neuronal dynamics. So there are actually two time lapses here. So in one side you have the neuronal dynamics evolving, and on another time scale you have actually the epochs, so the weight dynamics, the updates on the weights. And to do so, to do the updates on the weights, you actually require information about the neuronal dynamics, because that's the whole point, that you have these local learning rules dependent on the activity of the neurons. And you can, a simple way of doing this would be to take the first stable state on the neuronal dynamics and the second stable state. So this is the thing where I talked about that we have two phases of training. So one is a free phase where you have no information about the output, and the second is a clamped or semi-clamped phase where you actually have some information about the output. And usually these learning rules take these two states and use these two states as the change in the postsynaptic neuron, meaning you only need the free state for the presynaptic neuron, and then you need these two states for the postsynaptic neuron to have this direction of change on the activity of the neuron. But yeah, that's basically it. And if you want to be more accurate in the integration throughout this time evolution of the neuronal dynamics, then you can do like numerical integration, where you consider like every subsequent pair of different activities throughout the neuronal dynamics. I don't know if that was very clear.
Yeah, I think it's much clearer now, and I hope that answers Itamar's question. So probably, I'm sure you're aware of these discussions going on about whether backpropagation is biologically plausible or not. Do you take a strong stand on a side? Would you argue?
I didn't know. Okay, I didn't know there was actually a discussion on whether it was biologically plausible or not. I thought it was well established that it was not, because
it seemed to be
several
very well
reasons point
established.
to.
Yeah, it seemed to be very well established, but there are some people arguing arguments to.
Okay.
Well,
I think
maybe I can send you some links.
Yeah.
Yeah. Anyway, like that's. I mean, it's a fair
there are
point.
several reasons, but for example, there are several arguments that, I mean, backpropagation maybe approximates something, or it's a way to, but even, yeah. So two neurons are not symmetric, right? You have dendrites, you have synapses, it's not the same. And one problem will always be the fact that you need symmetric connections to do backpropagation, right? You need two neurons communicating both ways with the same weight. You can't have this kind of asymmetric, when I say asymmetric feedback weight, is that you have some feed forward neuron weight that has a certain value, and you allow for the feedback weight to have a different value, even if you are connecting the same two neurons. And in backpropagation, you have to assume that this is the same weight that goes both ways. And when you think about neurons, that's not what you observe, right? I mean, this is a simple reason that could rule out backpropagation in terms of biological plausibility. But it's very nice, it's very efficient.
Cool, thank you very much. Out of curiosity, so this is work you did in your previous position in Portugal?
Master thesis.
Master
thesis in Portugal.
And you, in your PhD, you keep on working on these topics, biological learning rules?
Yeah. Not exactly the same algorithm, but yes, it's the same topic.
Great. So for the audience, if you want to learn more about all this biological learning rules and biological plausibility in your networks, keep track of Matilda's work in the future. And yeah, with this, I thank you again for your talk, for presenting us your recent paper. And we'll move on to our next speaker, Dennis Turcu. Thanks, Matilda. So Dennis Turcu is a third-year PhD student in theoretical neuroscience at Columbia University in New York, and he's working with Larry Abbott. And today he will talk about using RNNs for dynamic binary classification. Welcome, Dennis, on stage.
Okay. All right, good. Can you hear me? Okay. So yeah, thank you. Thank you for being here to listen to the talk. So thank you all for tuning in. I will describe an alternative solution to the classical binary classification task, an alternative which is more realistic than typical models. The perceptron well studied for this task is effectively a set of n neurons, each of which receives some direct input and then project to an output neuron whose job is to decide the class of the input. Typical daily binary decision tasks include things like, do I do my yoga today or skip it, or do I eat the Halloween candy or pass it this time? And these decisions require sensory perception, cognition, language, motor commands, and even spatial reasoning. So these brain functions are highly linked with the neocortex, a brain area which is highly recurrent and very sparse. And unlike the neocortex, as seen in this diagram, the perceptron is not recurrent, is fully connected, and also has a highly convergent output neuron. So with this in mind, I used a new architecture to solve this relatively simple task. I maintained a similar structure for the input as well as for the output. The only exception is that the output is now very sparse, and I replaced the whole perceptron architecture with a highly sparse recurrent neural network to better model the neocortex. This RNN is truly very sparse, and connections are not symmetrical as suggested in this graph, where some connections are missing, but not all. And I will shortly describe this argument of the final or output sigmoidal or thresholding function to understand this RNN's computation. But before, I want to show that this network can achieve maximum performance with the backpropagation through time. So here, input is shown to the network for a period of 10 time steps, and then it is turned off. And this plot shows performance of the network at each time point, at each stage during training, where one can observe that as training advances, it learns slower and slower until it eventually hits 100% accuracy. So to understand how this network computes, I'm showing the output activity of the RNN for inputs from both classes, red and blue, as depicted here. It clearly shows that this RNN differentially amplifies the signal from one class and suppresses the signal from the other. An additional thing about this architecture is that it can successfully classify approximately as many random inputs as it has learnable synapses. And learnable synapses in this case is sparsity times n squared, the number of connections in between neurons which are learned with the backpropagation algorithm. And here I'm showing the mean in blue and the extremes in orange and green of accuracy as a function of memory capacity across multiple simulations. Just to summarize what the memory capacity of this network is, and to demonstrate that training is stable and reliable and avoids catastrophic issues such as catastrophic forgetting. So with high sparsity that I'm using in this network, the recurrent network will eventually disconnect according to the Erdős-Rényi random graph theory. And here in gray, I'm showing the probability that a graph of size n for different values of n is connected. And simulation results for an RNN of size 100 are shown in blue. So I want you to observe here that performance for this RNN is starting to be affected right about at the intersection in between the blue curve and the n equals 100 gray line. And the orange line is similar, but for an RNN of size 200. How I studied this slightly further is basically when I take an RNN above percolation threshold, so I generate a random graph whose adjacency matrix is connected above percolation. And then I manually disconnect it by building effectively five smaller random networks of the same overall size. Performance drops, as you can see here with this color-coded symbols. So then, interestingly, when I take an RNN below percolation threshold, so this adjacency graph here is not fully connected. And and then manually connected in these two highly structured ways, performance also drops, which this was surprising because the expectation was that a connected network would perform better, but structure in the connectivity is very important as well. And I'm showing that by taking a different RNN, also below percolation, but then I manually connected, preserving as much as the graph's random structure as possible. And in this case, performance increases. So it is not clear what random structure is important for high performance, but graph connectivity proves to be very relevant and allows the RNN to distribute information through all neurons despite the high sparsity. Here I introduced two separate Gaussian noises to the RNN just to show robustness of the architecture. Dynamical noise, which is geared towards the recurrent dynamics, and input noise. And I'm showing that RNNs of different sizes, different memory capacities, and different sparsity values exhibit lots of noise robustness. So in this yellow region here, basically you can have some noise in the network and it doesn't really affect performance. And also generalization accuracy decays in time slowly outside the training period in which backprop through time operates. And importantly, the two noise types that I'm suggesting here, though maybe a bit obscure, the two noise types sublinearly affect performance. And unsurprisingly, dynamical noise here, you can see it decays slightly faster in the generalization regime. Here I'm showing that this table shows that the architecture is also robust with respect to various activation functions and activation functions in the recurrent layer, and also to various loss functions that backprop is optimizing. It has some favorites, like some of these values are better than others, but we have a slight idea why this happens. But overall, it's quite robust. And finally, when neurons in this network have strict excitatory or inhibitory identity, performance decays rather naturally and not catastrophically. So the symmetry in the task, in the input shown to the task, reflects here in the symmetry of this excitatory and inhibitory plot, in which if you have too much excitatory neurons compared to inhibitory neurons, performance slightly decays. And though this is not perfectly symmetric plot, it shows a slight preference for having more E neurons, and we do not know yet why this is the case. But compared to an unconstrained recurrent network, the performance is effective naturally and quite as estimated. So in here we have a capacity of alpha 1. It's equivalent to a similar network, unconstrained network, which has a capacity of 1.6. Which can be estimated with some simple calculations as well. So to summarize, a highly sparse recurrent neural network can solve binary classification dynamically and distributively, making complete use of all of its neurons. It integrates input and maintains a decision for a long time by differentially amplifying its activity for different input classes. And for top performance, according to the Erdős-Rényi graph random graph theory, each neuron only needs on the order of logarithm of n connections to other neurons, which makes it a very, very sparse recurrent architecture similar to the neocortex. And with that, thank you, and thank you to my advisor, Larry, and all the funding sources. Thank you.
Thank you very much, Dennis. So while we wait for some questions from the audience, there is Friedman Zenke is raising hand. So let's see.
Hello there. Can you hear me?
Yes. Yeah. Go ahead.
Thanks for the talk. I have two questions. So first question would be, did I understand correctly that the topology remains fixed throughout the simulations? And the second question is, did you look into the dynamics of the network, and can you say anything about which mechanism actually amplifies or suppresses the solution?
Yes, to your first question, yes, the topology remains fixed. So basically, there's a highly sparse mask, 0 or 1 mask in the adjacency graph. So we do not modify connections. New connections between neurons cannot appear or disappear in the network. Can you please say again your second question? I'm not sure I understood your.
Yeah, I was wondering whether you analyzed the emergent connectivity, like look at the spectrum of the emergent matrix after training, and whether you looked into what's actually amplifying the correct solution.
Oh, I see. Yes, but only slightly, and it's not really clear what happens. So initially, we would expect to have in this plot, for example, we would have expected to have perhaps kind of two fixed points regime, in which one of the classes kind of goes to one fixed point and the other goes to the other fixed point. But what seems to arise naturally, at least, is you have some sort of slight amplification, and after this time point, basically everything slowly decays, which is reflected in this accuracy plot here. When you see when the decay starts to happen, basically the red curves and the blue curves start overlapping, and then you lose accuracy. So it's not entirely clear, at least to us, exactly what happens.
Cool. Thanks, Ferdinand, for the question. Is it?
Yeah, I can keep asking more questions, but maybe other people have questions too.
Not for now, so go ahead.
Yeah, go ahead.
Yeah, so then one more question about the topology. Did you think about maybe approaches of whether you could optimize the topology, as this is done lately a lot, and also we've seen in the previous talk, and whether this would then maybe naturally lead to a fully connected graph, or?
Yeah, I see. No, the short answer is no, I didn't look at that. The main reason is that for now, at least, the purpose of this work has been to kind of understand if a recurrent network can really use its information like distributively if it is sparse, without assuming a special case of highly convergent neurons. So, you know, if you start optimizing the topology, you may end up having a few neurons that require a lot of connections, and we wanted to keep things rather uniform. So here in this slide, when I say that you only need on the order of logarithm of n connections per neuron, we kind of that's a goal of the project. We don't want to have kind of rare event neurons which are highly connected. So we didn't look into that. We wanted to have kind of a uniform picture of the topology. Yeah.
Right, makes sense. Yeah, I find it very surprising that the dynamics are the way you showed, that they're not diverging to a fixed point, and that you basically have transients built into the dynamics, right? Usually you would imagine if you had a low rank connectivity, you would get a fixed point at high activity and one at low activity, but that clearly doesn't seem to be the case. And I'm trying to parse in my head where this could come from, because it clearly looks more brain-like that way, but yeah, very interesting.
Yeah, that's what we're trying to do as well. To be honest, it may be due to the specific type of loss function and training that I'm doing. So this here I didn't mention during the talk, but these black lines are some targets that are used for training. So the red curves are pushed by backprop to exceed this black line, and they are penalized if they are below this top black line. And likewise for blue curves, they are pushed to be below the lower black line, and they are penalized only if they exceed that. So that's why we kind of are able to create this gap to differentially amplify between those, but this is a purely simulation result, and we don't have an analytic understanding of why that happens yet.
Right, but the stability is then baked into the learning algorithm somehow. Although the red one, I suppose, could still go off to high activity, and you wouldn't prevent it from doing that.
Yes, that wouldn't prevent accuracy going down, but for some reason it doesn't happen in simulations. It doesn't explode off here. they all turn back to decay back to zero.
Okay, thanks, Friedemann, for your questions. We actually have one more question before we finish very soon. We have a question from the YouTube stream. How cool is that? That's by Daniel Hernandez. Thanks, Arash, for passing it over. So they're asking if it would be possible to implement this in real time.
The learning rule takes a long time to train, but once you have the network, it's incredible. It's very fast to make the decision. So learning will take a long time, backpropagation through time in this case. But once you have it, it's effectively instantaneous.
I see. Okay, cool. Okay, so if there are no more questions, I think we can finish here because there's another session starting very soon in 2 minutes. So thanks everybody from the audience for attending, and especially thanks to the speakers, Thijs, Mathilde, and Dennis for your great presentations, and I wish you a lot of success and luck in your future research. And thanks, Arash, as well, for doing a lot of great work in the background. Thanks, everybody. Have a good day.
So, hello, everyone. It's my pleasure to start the session. My name is Maria, and today with Jonathan, we will be your hosts. I'm postdoc from EPFL, Switzerland, and we have 3 exciting talks about single-cell modeling, pyramidal cells, some of them hippocampal, some are not. You will find out later. We have 12 minutes for each talk, followed up by 3 minutes question sessions. Please post your questions in the Q&A. We really need them. They deserve to be answered. Don't hesitate. If something doesn't work with Q&A, just post them in the chat and we will answer. Our first speaker is Jason Moore, a postdoc in Basu's lab at New York Neuroscience Institute. You will find a link of his recent paper, which is quite cool. I checked it out a few months ago, and I encourage you to do the same. Please, Jason, go ahead.
All right, thank you. All right, so the screen should be shared now. Can you get a quick thumbs up? Yes. All right. So thank you, everybody. Good morning. My name is Jason Moore. I'm a postdoc in the Basu lab at NYU. Today, I'm going to be talking to you about dendritic activity in area CA3 of the mouse hippocampus. This is a picture of me, and there have been a lot of meetings this morning, so I'm a little zoomed out here. All right, so I like to start out with the acknowledgements. This is our lab, the Basu lab. This is Jaida Basu. She took me in from across the country, and I'm a computational neuroscientist by training, and so this data that I'm presenting today is the first that I presented data that I've actually recorded. So it's pretty exciting for me. Shannon and Roland, they taught me how to do all the 2P imaging, and a lot of the groundwork was laid by Martial Dufour. So the roadmap here, I'm going to talk to you about the biology behind this project, an algorithm that is integral to some of the results, and then the actual results. So the biology, we'll talk about the hippocampus, dendrites, and calcium. So the hippocampus is a deep brain structure involved in long-term memory and spatial navigation. The famous patient is patient HM, who due to epilepsy had to have his hippocampus removed. And afterwards, he was unable to form new long-term memories. He could still hold old memories, but new memories he would quickly forget. More recently, people study the hippocampus in rodents. This is a video of the sort of canonical activity of a single cell. So we're looking at a top-down view of a rat running around in an environment. There's different cues on the walls, and we're listening into one single cell. And we're seeing that this cell fires spikes, these are these little dots here, only when the rat is in this part of the environment and not in any part of the other environment. We see to speed up the video, and we see that this neuron pretty reliably codes for this position in the environment. We call this a place cell. So you can represent it here. You can see the trajectory and all the spikes. Then you can turn that into a heat map. Different cells will code for different places in an environment. So you get a nice tiling of the environment. So you can represent an entire space. We'll get back to some of the important qualities of these later. So the second part is I want to make the hopefully not horrible claim that neurons are not point sources. Neurons actually maintain large trees of dendrites. This is maybe the model neuron that you might learn in a neurobiology class. It has a big old soma. You have these tiny little dendrites sticking out, and you have a big, thick axon. This is nice conceptually because the soma is where people have been able to record. But a real neuron looks almost nothing like that. The soma is this tiny little point down here. The dendrites extend, hundreds of microns, sometimes millimeters, out in all directions, and the axon's a thin process here. So the dendrite is actually where a lot of the action in the neuron happens. And it's not just the fact that the dendrites are just passively receiving input. They also have active integration. If you inject some current out in a distal dendrite and record farther away, you see nonlinearities. You see sodium spikes, you see calcium spikes, you see NMDA-dependent spikes. This is in vitro. Work from my graduate lab showed that if you record from dendrites in vivo, they actually have higher mean firing rates than soma that are in the same region. So we have a lot of interesting stuff going on in dendrites, but because they're so tiny, we don't really know so much about them. So we want to know in generally, what are dendrites doing in vivo? And how do they contribute to navigation? So to get at this, in the Basu lab, we record calcium signals from hippocampal area CA3 in awake mice. Why CA3? It has some interesting computational properties, but also it has this nice geometric affordance for us. So we aspirate some of cortex and we place this cranial window here. And the nice thing about CA3 is it's oriented such that the cell bodies and the dendrites are all in the same focal plane. So you can see them all at the same time. So we head fix the mouse on top of this treadmill. We have different textures on the treadmill to define position. And this is the kind of field of view we get. Absolutely gorgeous field of view. This is viral injection of GCamp7B into area CA3. We've recorded with 7B, also GCamp6F. You can stare at this for hours, and I do regularly. Here's a video of the dynamics sped up by a factor of 20. You can see it's a very rich data set. Sometimes you have dendrites spiking without the soma. Sometimes you have soma spiking with the dendrites. You see a lot of activity in the basal area, sometimes in the apical area. So it's a huge, rich data set that we are excited to look at. But first, I want to talk about an algorithm that we have been developing to actually deal with this. So the first thing you have to do is called segmentation. And I call it an extreme coloring book. So what do we have to do to make sense of this data? We have to go in and identify, okay, these pixels belong to this neuron. These pixels belong to this neuron. How does that happen? We go in frame by frame, we open it up in ImageJ or whatever your favorite program is, and you draw a bunch of lines. You say, that's that dendrite, that's that dendrite, that's dendrite. This is hard. This is tedious, it's repetitive, it's slow, it's variable. If you've had lunch that day or not, you're gonna have different quality of segmentation. So we wanted to come up with an algorithm to do this. We have a little wish list. I call it, we want to have mud here. We want it to be model free, that's the M. We want it to have few prior assumptions. That's to help avoid parameter search, avoid retraining. Ideally, we'd like to have an algorithm that can deal with 2-photon data such as this. This is an area CA1 where you have lots of circles, lots of same sized cell bodies, and an area CA3 like this, which has cell bodies with very different morphologies than the dendrites that they're connected to. We want this algorithm to be usable. We want it to be time efficient, memory efficient. We don't want to have to wait over the weekend to get the results of our clustering. And we want it to be deterministic. Ideally, the results should not depend on random initializations. And the cute little pun here is that from the mud grows a tree. And I say that because dendrites kind of look like trees. This is our fun acronym, Targeted Reconstruction of Excited Elements. I'll give a brief overview of the way we've tackled this. There's plenty of steps after this, but in general, we go frame by frame. For each frame, we look at this pixel, we call a pixel on if it exceeds some criterion alpha, say greater than 2 standard deviations, 99th percentile, whatever you want. So we get a binary mask saying, hey, that pixel's on. We're going to construct segment cores from pixels of connected components in 3D. we have 2D space by time. So we construct these segment cores. We only keep segments with some size greater than, with a size greater than some minimum value. So that's sort of our cleaned up binary mask. You repeat this across all frames, and then you merge components if they overlap by some value phi. So we've got 3 parameters here, not so bad. And they're all very intuitive parameters. This is an idea of how the algorithm works. So on the left, you're going to see the z-scored pixel values across time, and on the right, you're going to see the identified regions of interest. And you'll see that it's doing a pretty good job of picking out lots of regions of interest of different shapes, different sizes, different morphologies. So we're very excited about this. This is only the first step in the algorithm. You'll notice that there's a lot of overlapping segments, and we invoke rules of constrained non-negative matrix factorization, CNMF, in order to demix the signals from overlapping segments. This is some statistics of how do we do compared to an expert human labeler. We're at a pretty reasonable true positive rate, and we are covering more pixels on average than the human. And this is taking about 15 minutes. It's could be faster, but it's certainly not a whole weekend. So now that we can actually get at this data, we're going to get into some of the results about dendritic processing of hippocampal data. So the first thing that we looked at a couple of individual cells, and I'm going to make the claim that CA3 neurons can hold on average 4 place fields distributed among the dendrites. So I've plotted here, this is a dendrite that we have a nice basal tree and a couple of apical dendrites. And each segment here is color-coded according to what position that segment has its peak activity at. So the soma here is maximally firing at about 50 centimeters. Some of these dendrites are also firing at 50 centimeters, but this dendritic branch is firing at 125 centimeters. This dendritic branch is firing at closer to 0 centimeters. Up here, each row is the tuning curve for each of these segments. I've indicated significant peaks of activity with red dots, and you can see that this is actually not uniformly distributed. We actually have sort of a quantization or a clustering. This is a histogram of where are those peaks, and we can identify about 4 individual peaks in this neuron. This is a histogram across a couple of cells. This is not full scale yet, but it's a histogram of how many, in blue is how many fields an individual soma may have. And in red is the histogram of how many fields the entire neuron can hold. You can see on average in our hands, a soma will hold about 2 fields, whereas its entire dendritic tree has about 4 fields. This is positively correlated. So if a soma tends to have few fields, its whole field tree might have few fields, but it's a weak correlation. So this is a nice example. We quantify this measure called branch-specific tuning, which is if a cell has a dendrite with a place field different than its soma, we say it has branch-specific tuning. If a soma is tuned, about 20% of those somas will have independent dendrites. If a soma is not tuned, we get a whole lot more dendrites that have independent tuning, which is somewhat unsurprising because the soma is not tuned. But it is surprising that somehow the dendrites are tuned, but that information is not getting to the soma. So we want to think, what is this useful? Why spend all this energy? We're thinking this goes to the concept of remapping. So really quickly, if a rat mouse is put into a different environment, the cell will remap. It'll start coding for a different position. That happens if you go to a different environment. It also happens if you change the task within the same environment. So possible mechanisms, we're thinking, okay, here's a neuron. Its 2 dendrites are getting coding for different positions. And maybe in context 1, it's listening to dendrite A. It's not listening to dendrite B. There's some gate here. In context 2, maybe the inputs change. Maybe that's all that changes, and so it's still listening to dendrite A, but it's remapped because the input to dendrite A has changed. Another possible mechanism is you have the same initial situation, and what happens is the inputs might stay the same, but this gating changes, that something changes which dendrite this soma is talking to. So we wanted to do running to the end here, we wanted to do a quick experiment to see, do we see evidence of this rerouting? So we have mice run on one context, And this is a correlation matrix of the soma. Soma is in the top row with all of its dendrites. And we can see this nice correlation structure. You know, there's some branches that the soma is well correlated with, some that it's not so much. And then we switch the mouse. We put him onto a different belt, different textures, different colors and such. And we can construct the same correlation matrix of the same neuron. And we see that there's some similarities, but there are some differences here. So I'm just going to plot the top row of belt A correlation matrix with the top row of belt B correlation matrix. And we see this. There's some dendrites that stay the same, but some dendrites that have increased their correlation in environment B, some in environment A. Make a histogram of this. This is kind of suggestive that there might be 3 different transitions, if you will, but we need to expand the analysis to more cells. So the model, the conceptual model we have here is that dendrites hold multiple possibilities. They're out there. And that specific representations are routed to the soma in a context-specific manner. This might enable learning in parallel, rapid switching, you know, plasticity mechanisms are sometimes slow. This would be a rapid way to change the representation. What controls the routing? Perhaps inhibition, perhaps neuromodulators. That's the next step in this project. So quick conclusions, dendrites are accessible with the right surgical prep, Automatic segmentation is critical to processing this dense data, and we're making good headway on this. Neurons can hold multiple place fields in their dendritic tree, up to on 4 on average, and information can be rerouted through neurons. So I'm happy to take any questions, and thank you for your time.
Thank you very much. This is this amazing stuff. It's amazing work. This branch-specific tuning dendrites, it's amazing, really. And we have a lot of questions.
Great.
So yeah, we will try to keep it short, the answers. We will go for 2 questions. So first one, William Mao asks, what is the trial-by-trial consistency of this dendritic place fields? Sorry.
Yeah, the trial-to-trial consistency is actually very high. I don't have a quantification of it, but it's as stable as the somatic fields are. And our surgical prep is such that we don't have a whole lot of motion artifact in the Z plane. So it's very stable.
Okay, thanks. And the second question from Eleanor, who says that the topic is very interesting. And would you say your results support the contextual gating model of Kate Jeffrey, where contextual inputs select which dendrites will be used to generate an action potential?
Yeah, I think it sounds completely coherent with that. Contextual gating, you know, it's always a question of what generates the context signal. And so we might need to get into more subtle manipulations to see, is it, again, to get to the question of, is it the inputs that are changing because the context has changed, or is the gating, is that structure of the flow within the neuron changing with context?
Okay, thank you. Unfortunately, we have to move on and for the next talk, but please don't hesitate to answer questions.
I will answer questions in the chat.
Yes, there are like 4 questions more, 5, I think. So we move forward, and our second speaker is Carol Upchurch, a grad student from Louisiana State University. And we're looking forward to hearing from you, Carol.
Good morning, everyone. Like she just said, my name is Carol Upchurch. I'm actually in Dr. Canavier's lab, who does a lot more computational work. But we work very closely with Dr. Gasparini that produced a lot of the electrophysiology data that I'm going to show you today. And like Dr. Moore, I'm going to be talking to you about place cells, but I'm talking about place cells in the CA1 region of hippocampus, not CA3. Sorry. So like Dr. Moore, I'm talking about place cells. Place cells, I thought his cartoon was pretty great, but I'm going to go over mine again anyways. So we've got this cartoon with a rat on the right, and as the rat runs through this little maze, there's going to be a cell that specifically fires in each place in that little maze. So as it's running through that purple area, those are purple spikes from a cell that will respond to that little purple area. So that's its place cell, and the purple area is the place field. So why do place cells fire this way? Well, in the CA1 region, the CA1 region receives synaptic input from CA3 in the entorhinal cortex. This synaptic input tends to look like a depolarizing hill, so it goes up and then down again as the animal is entering the field and then leaving. And while the hill might be symmetric, the firing is not. The place cells in the CA1 region tend to fire more on the uphill than the downhill. And that's what we're particularly interested in this collaboration I'm talking about. So this is data from Dr. Gasparini's lab. These are two separate experiments. They either injected a triangular ramp, so the same kind of idea of an uphill and then a downhill that you would have seen in vivo. They injected that ramp either in the soma or in the dendrites. And this is the data from that. And there's a couple of things I want to draw your attention to. So first off, I want you to look at the frequency graphs. This is the instantaneous frequency. So we took the interspike interval here, and whatever current is right in the middle of that, it's current, then that frequency. And what I want you to notice is that the filled circles are on the way up, and the open circles are on the way down. And at each injected current, you're getting higher frequency on the up ramp versus the down ramp. This is the adaptation that I was talking about. And so that's the first thing I want you to notice. The second thing that I want you to notice is that this adaptation is more accentuated further out in the dendrites than it is in the soma. And the third thing that I want you to notice is that there's a decrease in spike height as this depolarizing wave goes on. And that's the phenomena we want to capture in our model in Dr. Canavier's lab. So I did not start from scratch with this model. Dr. Canavier had previously published a model of a CA1 pyramidal hippocampal neuron. It's a multi-compartment morphologically realistic neuron. This is the morphology right here. It's implemented in Neuron. You can download it off of model.eb. The model accession numbers right here. It's a Hodgkin-Huxley type model. So we're looking at the equivalent circuit right here. Each of these resistors is your ion channel, and they're all in parallel with the cell membrane capacitance. We made a couple of changes to this model, and that's how we ended up modeling the adaptation. We replaced this sodium channel in the soma and the apical dendrites. This used to be a Hodgkin-Huxley type sodium model with the 3 activation gates and the inactivation gate. We replaced that with a Markov model. We also added a second potassium delayed rectifier, which I'm going to get to later. This is an updated schematic of the model. So again, we added a second delayed rectifier. This is because I wanted to make a point that experimentally, these currents are not uniformly spread across the entire neuron. So we didn't model it that way because that wouldn't be realistic. Certain channels like IH and IA, the conductance is greater the further you get away from the soma. And some of the properties of these channels also change the further you get from the soma, and I'm going to get to that. So I'm a desserts first kind of person. I wanted to show you that we were successful. We do have the adaptation that we were trying to model. It is accentuated in the dendrites. That's kind of visible in the short ramps. This is 1 second up and 1 second down. It's more visible in the longer ramps. This is 5 seconds up and 5 seconds down. But I'm going to show you how we got to this. The first thing I want to talk to you about is the Markov model. A lot of you are probably more familiar with the Hodgkin-Huxley model of a sodium channel, the one that had 3 activation gates and an inactivation gate. That's technically a Markov model. A Markov model just means that whatever state you're going to next is dependent on your current state and not any state you were in previously. The Hodgkin-Huxley 3 activation gates and an inactivation gate is technically a Markov model. The problem we were having is that all of those gates are independent of each other. So when we were trying to implement slow inactivation, we didn't want the channel to go from open or closed straight into a deeper inactivated state. We wanted it to go from inactivated to a deeper inactivated state. So we moved to this Markov model. this was, sorry, having trouble with the slides here. 1 second. This is these two closed states. If you were trying to think of this as that Hodgkin-Huxley model, you'd probably think of these as the inactivated gates being open, but the one of the 3 activation gates being closed. This open state is when your inactivated gate is open and all of your activation gates are also open. This is one of those inactivation gates, and this is that second deeper inactivated state that we were trying to implement and to get that slow inactivation. There are some people who have tried to do that when you, with the Hodgkin-Huxley model, they just added, instead of M cubed H, they have M cubed HS, with S being the slower inactivation gate. The only reason we didn't like that is because then you could go straight from one of these closed states into that inactivated state, or straight from the open state to that inactivated state. And that gave us some really funky waveforms. So we decided to transition to this model. So why were we so interested in slow inactivation? Well, this is some data from the Respon lab. This is a series of cell-attached patch clamp experiments. These are a couple of sample traces from each. They're from different distances from the soma. And what they did is they held it with a voltage clamp at resting potential, but they delivered a 20 Hertz pulse strain. And then they were looking at the amplitude of this first spike and the amplitude of the last spike so that they could calculate a sort of stable percent inactivation that would happen the further they got into this pulse strain. And what I want you to notice is that that percent inactivation increases the further you get from the soma. And the reason we thought that this might be a mechanism for adaptation is, 1, it kind of explains that decrease in spike height that we saw in experiments. And 2, because it increases the further away you get from the soma, it would make sense that if it was contributing to adaptation, that adaptation would increase further out into the dendrites. So I wanted to model this slow inactivation. So I took the Markov model and I put it in a little patch in Neuron, the software that I was using. And I just increased this transition. I just multiplied it by a scale factor, and I would simulate a pulse strain and calculate a percent inactivation. If you do that, what you end up is the black line over here. I'm trying to get to the black line that is supposed to be that exponential from the last slide over here. So I'm trying to get from there to here. So I just fit that first black line with that purple line. It's a 4-piece linear equation. From there, now I know what kind of percent inactivation I want in each compartment from the distance from the soma. Now I just had to say, okay, so for 50% inactivation, I need to use a little bit more than 2. And that's what this graphic right here is. It's just showing you the distance from the soma, from each distance from the soma, what scale factor I'm trying to use. And the results of my model are this red line. I know there's a little inconsistency there, but it's well within the noise. So it was kind of good enough for my purposes, except for one problem, and that is that I was approaching that steady state percent inactivation a little too quickly. None of these traces look like that trace. That's a pretty simple problem. What happened is that both of these transitions were too fast compared to all of the other transitions in the model. They were too big. So I just scaled them both down by 0.2. As long as my I1 to I2 transition is bigger than my I2 to I1 transition, I'm going to have, as long as that ratio is bigger, I still get the increased inactivation. But if I make both of those transitions smaller, I slow down my time course. And once I did that, it was pretty simple to, refit the equation. And I had a model where I could change the slow inactivation based off of how far from the soma I was. And I took that model and I put it in Dr. Canavier's previously published model. And I got results that looked like this. I did not capture adaptation on my first try. I was really confused by this because we were really thinking, slow inactivation would be the mechanism. So I didn't understand why I got this weird burst. If it wasn't the mechanism, I still wouldn't have expected this very strange result. But when I showed it to Dr. Gasparini, she was thrilled. And why was Dr. Gasparini thrilled? Dr. Gasparini was never totally satisfied with our explanation of potassium delayed rectifiers in our model, in the previously published model. And that's because our previous published model only has one potassium delayed rectifier. In CA1 hippocampal pyramidal cells, there's actually two channels that act as delayed rectifiers. There's KV1 and KV2. And this is what happens when you block KV2 experimentally. This is the same kind of experiment she gave at a ramp. So a ramp up, a ramp down, a triangular current ramp to look like that depolarizing hill that place cells receive. And the red trace is what happens when you block KV2. The black trace is control. So I hadn't replicated adaptation. I replicated this weird bursting that happens when you block KV2. So we just went back to model DB and we found a model of KV2. The model accession number is right here. If you go and put that in model DB, it's going to tell you it's from the Allen Institute's data set of cell types, and it'll say it's a visual primary cortex. But if you look at the mod file itself, it was actually fit from data from this lab, and it's CA1 hippocampal pyramidal cell neurons. So we took this model of KV2, we put it in our model, we adjusted the conductances accordingly, and this is just circling back to the results that I showed you a little bit earlier. So how exactly is that working? KV2, when I graph the currents by themselves, tends to be a little bit more active in that hyperpolarization that happens. So this is a zoom in of those waveforms. And you can see it as it hyperpolarizes, that's where KV2 really kind of shines. And I think you need that after hyperpolarization when we're talking about the Markov model to push the model out of this inactivated state and back to the available pool. So to really show that, I want you to look at the C1 state. So they're not equivalent. It's not equivalent to the Hodgkin-Huxley model, but if I was going to make analogies, this C1 and C2 state, the inactivation gate is open, but one of the activation gates is closed. So it's available for spiking, but it's not currently open. And I think you need that after hyperpolarization that was caused by KV2 to push it out of the inactivated state and back into the available pool. And that's why I graphed C1 here. So this is just a victory lap. We're going to look at the experimental data versus the model, and these are the short ramps. Again, it's a triangular current ramp in the experiment, and it's a triangular current ramp that I simulated a neuron. And we did successfully replicate adaptation. There's definitely more firing on the up ramp versus the down ramp. It is a little bit more emphasized in the dendrites. That's a little bit more visible to see in the long ramps. So this is 5 seconds up and 5 seconds down. This is the experimental data. Over here, we have my model. And the other thing that I want you to see is that we have that change in spike height. And that's part of why we really thought that slow inactivation could be the mechanism for this adaptive firing. So I'm going to wrap up now. We created a Markov model of NAV 1.6 that could implement slow inactivation, and that could implement slow inactivation in a spatially distanced way from the soma. And when we put that model into our previously published model of a CA1 hippocampal neuron, we got bursting, which is exactly what happens when you block KV2. So I didn't replicate adaptation. I replicated the bursting that happens when you block KV2, which showed us what was the problem with our model. So then we put in KV2, and that restored spiking. And with both KV2 and the slow inactivation of NAV 1.6, we were able to capture the adaptation and the accentuation of that adaptation in the dendrites and the decrease in spike height that you see in the dendrites. And I'm ready to take some questions.
Thank you very much, Carol. This is very exciting, and also detective work that you did. My congratulations. Do you have any questions, maybe from the panelists? In this case, I will have quick questions. So it's surprising that in the dendritic response, you can also predict choice by putting KV2 in the soma. Or I misunderstood, we also have KV2 in the dendrite. You don't really.
KV2 is also in the dendrites, yes.
Okay, and how did you get the distribution of the Kv2 in the dendrite?
There's some immunohistochemistry results from the Nusser lab, and it shows Kv2 in the dendrites. It's more in the proximal dendrites and less in the distal ones. So I put it in the first 150 micrometers of the model, and then I multiplied it by a scale factor of 0.1 because you can still see it out in the distal dendrites, but not half as much as in the proximal dendrites. So it's not uniformly distributed.
Yeah, but still gives very promising results. It's nice. Thank you very much. And unfortunately, we have to move forward. These sessions have to be longer, really. And our last speaker is Alessio Buccino, a postdoc from Andres Hillerman lab at ETH. And in the chat, you can also find him on Twitter, find his link on his Twitter and ask him questions further. Please, Alessio, go on.
All right. Can you see my screen? Yes. Okay.
Perfect.
Okay. So thank you very much, Maria. So my name is Alessio Buccino. I'm a postdoc at ETH at Andreas Hirnemann Lab, the bioengineering laboratory. And the work that I'll be presenting today is a collaboration with the Blue Brain project at EPFL. And it's about a multimodal fitting procedure to construct biophysically detailed multi-compartment neural models. So first of all, I would like you to quickly introduce these multi-compartment models. Actually, Carol also had some of these models, but basically these kind of models are biophysically realistic representations of single neurons, and they're mainly built of two different parts. The first part is the morphology, which is usually extracted from fluorescence images of single neurons, and we can get a very nice and fine 3D representations of single cells. And the second part, which is harder to basically construct, are the electrical properties. And these are usually fitted to electrophysiological recordings, mainly from patch clamp data, and they are used to fit the many parameters that these models are made of. And I just want to mention that in the last 5 years or so, maybe 10 years, there's been a huge international effort in building and sharing these detailed and complex models, especially from the Blue Brain project, where they have thousands of cell models from rets, sensory motor cortex, and also hippocampus, and also from the Allen Institute of Brain Science, where they contribute several hundreds of models from mice and even from human tissue. These models are usually built from signals from somatic patch clamp. So basically you apply a stimulus, you get a response, and then you try to find a model that fits your responses. But the main problem that I see here is that neurons are more than just the soma, and the two presentations that we heard before actually confirmed that, right? And so a single patch clamp recording in the somatic region does not allow us to observe other neurites like dendrites or the axon or the axonal initial segment, which might be important and are important for the physiology of these neurons. So what can we do differently? Actually, some researchers, in particularly Itai Hay in 2011 and also others, have used multiple simultaneous patch recording in order to build these biophysical models. In this case, for example, they had 3 different patch pipettes, 1 in the soma and 2 in the apical dendrite. And they used the recordings from all these 3 regions to fit the multi-compartment model. And they were able to reproduce very complex dendritic behavior like calcium spikes. And these behaviors are actually quite important for dendritic integration and synaptic plasticity. However, I don't know if you have ever tried to do patch clamping yourself. Even with 1 pipette, that's extremely hard. And doing it with 3 pipettes simultaneously, it's very experimentally challenging. So we propose an alternative method. So instead of having multiple pipettes, we propose to use a single whole cell patch clamp recording from the soma, but to have the neurons sitting on these high density multi-electrode arrays that we develop in the lab. These are super high dense, so the pitch between electrodes is around 15 micrometers. So we can really observe the extracellular activity of neurons at a very fine scale. I would like now to kind of review what's the usual pipeline that it's used to build automatically these kind of models. So we start from the neuron, and as I said before, we have our patch pipette and we extract some data in response to some protocols. Usually step protocols are used of different amplitudes. And then from the same neuron after the experiment, we can finally reconstruct the 3D morphology. Then we need some other ingredients. From the data, instead of using the raw data, we usually extract some features that are a compressed representation of this data. And features can include the number of spikes in the response or the average interspike interval, as well as spike shape features like the amplitude of the action potential or the width of the action potential. And also we need to define the mechanisms of the neuron. And these mechanisms are conductance-based models. And we have to define which ion channels are part of which neuronal sections and how they are distributed. And then the goal of this optimization of this process is to find the parameters of these mechanisms that basically can reproduce the data that we record from. Then we put everything together and we apply the same protocols. We combine them with the morphology and the mechanism information to make a computer simulation of the neuron. And then from this simulation, we extract the same features that we extracted from the data. And we basically try to find the parameters that better fit the feature extracted from the data. In this case, we need to optimize some objective function, and this is usually done via evolutionary strategies or genetic algorithms. And this is the pipeline that BluePy Oct, which is a software developed by the Blue Brain project, uses. So basically, these objectives tell us how well these features extracted from the simulated data fit the feature extracted from the real data. And then once this optimization is done, we obtain our model, which is the best fit. However, in order to use also extracellular signals, we need to do some modifications to this pipeline. Of course, we need to extend our data acquisition strategy, also including extracellular signals. We need to also extract features from those extracellular recordings. And most importantly, we need to change the simulation in order to also simulate extracellular signals. Regarding the simulations, originally BluePy Oct support the neuron simulator, but we extended it in order to use LFPy, which is a wrapper to the neuron simulation, which also allows to compute extracellular signals. So the basic idea is that you use neuron to solve the cable equation, and this will give you all the currents that flow in and out of the neuron from all of its compartments. And then using volume conduction theory, we can sum up the contribution of this transmembrane current to compute the extracellular potential at our electrode positions. Then we need to define other features for our extracellular signals. And this is an example of the neuron sitting on top of our simulated multi-electrode array. And here I'm zooming in on the channel with the largest amplitude, and we define some shape features for all of these channels. So we have features like the peak to valley duration, the spike out width, the peak to trough ratio, and so these are defined channel-wise. We have another set of 4 features, which instead are defined with respect to the channel with the largest amplitude. And an example of this feature is the duration, or how much time there is between the negative peak in one channel and the negative peak in the channel with the maximum duration. So these kind of features also allow us to basically describe the propagation properties of this signal. And differently from the intracellular features, in this case, for each feature, we have an array of values. So we have these feature maps. So we have to use a different kind of objective function. And in this case, we use the cosine distance between the simulated feature and the experimental feature. Okay, actually, we don't have data yet, but we built some ground truth simulations in order to test this approach. So we started from this layer 5 pyramidal cell model, which was published in 2016 in the BluePy Oct paper. And then we sampled the parameter space, and we basically built 5 different new models, which we treat as ground truth, and we use them for evaluation. And then we also want to compare 3 different feature sets with the access to different sets of signals. So in the first feature set, basically this is the gold standard of building these models. So we only have access to the somatic traits, and we have 3 protocols, 3 steps with different sizes, and a total of 30 features that we want to optimize. Then we have this multiple feature set in which we have 3 different patch pipettes that can record simultaneously. In this case, we add a new protocol, which is a strong depolarizing pulse, which also allows to basically excite the dendrite in its active regime. And in this case, we have 35 features and 3 access points. And then we have this extra feature set in which again, we combine the somatic recording with extracellular features, and we have 7 extracellular features, so we have 37 features in total. I will show just some qualitative results. We're still working on doing this in a more systematic way. But in general, what you can see here is the black trace is the ground truth response of the model. And these are the responses to the 5 different test models to 3 different steps of increasing amplitudes. And in all cases, we see that basically the somatic traces are well fitted by all 3 feature sets, even because most of the features that we used are extracted from the somatic traces. But then things get interesting when we start looking at the dendritic response. And these are the responses of basically the soma and the 2 dendritic sites, which are only available to these orange signals, so the one that use multiple patch pipettes for optimization. And we can see that, in fact, the orange signal fits well the ground truth data in all of the 5 cases. The blue line instead is the somatic feature set, so the one that only has access to the somatic trace, and they can fit well these dendritic responses only in 1 case out of 5, while the extracellular one does somewhat in between, so 3 times out of 5. So in this case, and this case, it can well predict at least the amplitude and kind of the long decay of the dendritic signal. And then, so basically with this extracellular feature set, we can do better than the somatic one, but a bit worse than the multiple feature set. Then finally, I talked about extracellular signals. I just wanted to show you how they look and how the different feature sets fit these kind of signals. So in this case, we use the linear probe that basically traverse the entire neural z-axis. And these are 2 examples we can see, and these signals are actually normalized by their maximum. Otherwise, the dendritic signals would be quite low and hard to see. But we can see that in general, the green and the orange line fit very well the black one, while the blue line sometimes it's a bit worse. And also we basically summarize this by computing for each of these test models, the cosine distance between all of these ground truth signals and the signals, the basically simulated signals using the 3 different feature sets. And we find that the extracellular feature set does almost as well as the one with multiple patch pipettes, while the somatic signal is definitely worse in estimating correctly these extracellular action potentials. So finally, this is preliminary data and only in silico, but these in silico simulations can actually give us some insights also in designing new experiments. And we're still trying to solve some issues. So I showed results only using this linear probe, and ideally we would want to go to something like this, so having thousands of electrodes recorded simultaneously. The problem is that when we go from here to here, simulations start to be quite bulky, and so we're trying to speed up these simulations. Secondly, we use these layer 5 pyramidal cells from the Blue by Oct paper. However, the basically dynamics of the dendritic trees is quite limited. So we are now in the process of moving towards this Hay model in which there's a lot of richness in the dynamic of the dendrites. And we would also like to change the model to include also a detailed model of the axonal initial segment, which is well known to strongly influence the extracellular signals, and it's also involved in cellular processes like homeostatic plasticity. And then we are currently setting up the experiments, and there are some challenges that we are facing. So first of all, the neurons, they need to be sitting very close to the electrodes in order to get a large signal. So we are starting with cell cultures, which are mainly planar, and they nicely sit on top of the MEAs. And then we're moving towards 2 directions, either going to very thin slices or using organotypic slices, which are slices that are basically cultured on the chip and they become flatter over time. And then since we want to compare the experimentally recorded data, extracellular signals with the simulated one, we need to match the transfer function of these electrodes. So these electrodes are actually physical entities. They have an internal capacitance and resistance. And so we are measuring this transfer function, and we plan to apply it to the simulations to basically compare apples and apples. And finally, I would like to thank all the contributors to this ongoing project, and the ETH and EPFL for the support. And I would like to thank you all for the attention. And let's see if there are any questions.
Thank you very much, Alessio. Do you have any questions from the panelists, maybe? No, not yet.
I guess I have a question.
Oh, okay, go ahead.
So there's a paper from Christoph Koch's lab talking about high amplitude positive spikes. Are you familiar with that? And do you have any evidence of that?
You mean on the dendritic trees?
Yeah, when they recorded out in the dendritic trees, they saw
Extracellularly.
Yeah.
I mean, you do see these positive spikes, but I would say that mainly it's kind of hard to distinguish whether they are generated by the intrinsic dendritic conductances, or they are just the effect of this balancing that you see between the current sinks and sources, right?
Right.
So when you have an action potential with strong currents, like inflowing currents in the soma, you would have other like current sources distributed over the dendrites. So you usually see, and we can also see it actually here. So here I'm showing the somatic extracellular action potential, and this is a dendritic one, and you can see that it's kind of reversed, right? Yeah. So I would say that what we measure extracellular is mainly basically the contribution of this distribution of currents, but of course, especially later during the spike, or if you also kind of recruit these active conductances in the dendrites, the shape of these extracellular potentials will be also influenced by these dynamics. And so that's what we hope, that this would be enough to characterize those conductances.
Thanks.
Okay, we have to move out from this room because there's going to be next seminar session here. I want to thank all the speakers for the super interesting talks, and just wish you luck in all your research in the future, and hope to see you around at the conferences.
Thank you.
Mammals mainly, and Will Norton, expert in the aggression neurobiology, but this time in zebrafish. So you are all welcome at for the deciding action mini symposium. The goal of this mini symposium was indeed to put together like 3 speakers that will tell us about neural circuits, behaviors, and molecules, and small refined mathematical analysis of innate behaviors that govern across species, like a fundamental states of animals. So, as I said, we have 3 speakers going. So I invite all the participants to not only follow, but to prepare their question that you can slip in the Q&A message chat and challenge the speaker. I propose that the questions are going to be taken at the end of each talk. And we start with the first speaker, who's Oded, that is going to tell us about inherited memories. Oded from Tel Aviv, the Department of Neurobiology. We should all clap and congratulate you for your recent prize received, I've seen. So thanks for sharing your data with us, and the floor is yours. Go for it.
Thank you very much, Manuel. I'm just sharing my screen. Okay. Can you see my presentation? Yeah, great. Okay, so thank you very much, Manuel. And today I will talk about the possibility that the parents can affect a crucial decision in their progeny for multiple generations, or transgenerational epigenetic inheritance, which is a controversial topic in many animals. In C. elegans, in nematodes, it's more established. But before that, I want to just exemplify a situation where affecting the progeny's decisions make sense, or where transmitting information across generation makes sense. And to exemplify this, I want to start with a short clip from my favorite movie, Groundhog Day. Those of you who have seen it know, those of you who haven't seen it should probably see the movie, it's really good, that Bill Murray's character relives the same day over and over. And because it's so predictable, because he knows exactly what's going to happen, he can really acquire information in a very useful way and become an expert in what's about to happen, use it to his advantage. So let's see this short clip.
A gust of wind.
Here he robs a bank in this clip.
Dog bark.
Cue the truck. Exit Herman. Walk out into the bank. Exit Felix and stand there with a not so bright look on your face. All right, Doris, come on. Hey, fix your bra, honey. That's better. Felix! How you doing, Doris? Can I have a roll of quarters? 10, 9, 8, R, 6, 5, quarters, 3, 2,
So the situation that Bill Mary lives in Groundhog Day is similar to what we've been experiencing since Corona, where every day is the same and time lost its meaning. And it's the same also for C. elegans always, not only during Corona, because the worm's generation time is really short. It's just 3 days approximately. So the likelihood of the parent's environment being relevant to the children's environment is very high. And they live in a sort of a boom-bust way. They find an apple, for example, they eat it for a few generations, and then they will be starved for a few generations. So perhaps it makes sense to transmit the acquired information and information about the environment to the children. And while transgenerational inheritance was a controversial, or inheritance of acquired traits, that's really a dirty word that you shouldn't say, was controversial for hundreds of years. In C. elegans, it's now quite established that when you stress the worms, in different ways, they transmit small RNAs that via RNAi or RNA interference regulate gene activity in the next generations for multiple generations. So we've first shown that this happens when you challenge the worms with viruses, and also when you starve the worms, they prepare their progeny for additional rounds of starvation. When you put them in high stressful temperatures, they produce a transgenerational gene regulatory effect, and also when you expose them to pathogenic bacteria. And this really goes against the dogma of what's possible in inheritance and in evolution. And it doesn't happen through changes in DNA, it happens through this parallel pathway that transmits small RNAs across generations. And in C. elegans, the small RNAs that are transmitted between generations are not diluted because the worms have these RNA-dependent RNA polymerase that amplify the small RNA response. So there are different kinds of small RNAs, endogenous small RNAs, exogenous small RNAs, piRNAs, siRNAs, and they don't lead directly to silencing. What they do is they recruit these RNA-dependent RNA polymerases to the mRNA and lead to the amplification of much more abundant secondary small RNAs that keep the response going. And these are carried by specific proteins, argonauts, in the germline to silence genes in the next generations. And we've shown in the last couple of years that this is a tightly regulated system that typically generates responses that have a typical duration. So in most cases, when you start an RNAi response in C. elegans, it continues for 3 to 5 generations and then it stops because of this feedback process that we call the transgenerational timer, which is mediated by genes that we call MOTEC genes. MOTEC stands for Modified Transgenerational Epigenetic Kinetics. It also means sweetheart in Hebrew, that stop the response after a while. And very recently, just last month, we also showed that while the worms are genetically identical to one another, almost isogenic, there's a mechanism that intentionally creates variability between genetically identical siblings, and that the response, the epigenetic response that spreads across generations follows 3 simple rules of segregation, which are, of course, totally different from the rules of segregation of DNA, which we know since Mendel just represent, in the end, the separation of the chromosomes in meiosis. This is a different set of rules, and this inheritance is mediated by a different molecule, small RNAs. We also showed last year that if you start, and this is more related now to neuro, that if you change the pool of endogenous small RNAs in the nervous system of the worm, it creates a transgenerational response that eventually changes gene expression in the germline, and consequently also the behavior of the progeny for at least 3 generations. So the parent's brain controls the behavior of the next generation. I'm talking about chemotaxis behavior, the capacity of the nematodes to find food, which is the most important thing if you're a worm. And we found how this is happening pretty much. So this depends on one particular argonaut in the germline. It's called HRD1. And the heritable small RNAs regulate one gene, which is called SAGE2, which is totally crucial for this chemotaxis behavior. The important thing I want you to remember from this slide, because I won't get into the details because it was already published, is that the small RNA response starts in the nervous system here, but the effect goes through the germline. And it stays in the germline. So the small RNAs, the small RNA change starting in the nervous system, it leads to a change in small RNAs and mRNA in the germline, and as a result, it affects behavior. The response doesn't move back out of the germline to the nervous system. How can you affect behavior by affecting only germline gene expression? It sounds weird at first, but in fact, if you think about it, it's quite trivial even, because think of a dog. When you affect the germ cells, if you castrate a dog, it affects its behavior. And similarly, we experience all kinds of changes in puberty that eventually affect our behavior. So because the germline secretes all kinds of molecules, and it's really important for systemic responses in general. So this is just the introduction, but today I want to talk to you about new unpublished results, totally new, haven't been submitted yet, but will be soon, I hope. This is the work of three amazing PhD students, Itai, Itamar, and Yael. And they wanted to ask, what does this transgenerational regulation of germline genes enable? How can it change in the next generations neuronal functions? And can it also, in an indirect way, affect evolution? And I will explain what I mean. Okay, a little bit of background about C. elegans nematodes. These worms are hermaphrodites. So they self-fertilize. They make both sperm and egg. However, there are also males, which are more rare in the population. And the worms, the hermaphrodites, can mate with males. And this is a crucial decision, whether to mate or not, because first of all, mating has a big cost. When you mate, you risk predation, you extract a lot of energy, but most importantly, you dilute your genome in half. Or you will only contribute half of your alleles. And this is crucial for evolution because it increases variability and variation in the population significantly. So there's a big cost, but there's also a benefit if you want to mix things up. And it was shown that in a stressful situation, in an unpredicted environment, it's worth it to outcross, even in C. elegans nematodes. For example, when you co-evolve them with pathogenic bacteria in lab evolution experiments, because you need this added genomic variability or diversity to find the right solution. So it's a very crucial decision. You dilute your genome in half, but it could be worth it in unpredicted situation when you need to evolve fast. And the worms can affect their chances of mating, the hermaphrodites. Work from the Maureen Barr lab and Paul Sternberg's lab in the last couple of years has shown that while the males are rare in the population, the hermaphrodites can decide to secrete a pheromone that will attract the males and induce mating. The interesting thing is that they have a sensor, they know when to secrete it. They only secrete this pheromone when they become old. So the old worms are more attractive to the males than the young worms who are barely attractive. You can see here in the graph that you extract odors from worms. You don't even need the worm on the plate. You extract the odor from the worm, you put it on the lid of the plate, and you put the males in the middle, and they will go towards the odor or not. And if you extract odors from young mothers or that are just 1 day of adulthood, then they are not attractive at all. But as they become older, they become more and more attractive to the males. And they know that they become old and they should start attracting males because they have a way to sense the absence of sperm. When they can no longer, when the hermaphrodites can no longer produce self-sperm, they can't fertilize themselves. And this happens when they age. And at this point, it's worth it to start secreting the pheromone so the males will come and provide the sperm so that they can continue to have babies, only using this male-derived sperm. So once they sense that the sperm is gone, they start secreting the pheromone and attract the males. It's a very smart system. My hypothesis was that this would be affected. This might be affected by small RNAs and perhaps small RNA inheritance, because we know from many studies that endogenous small RNAs are crucial for regulating germline genes in the oocyte and genes in the sperm. And we know that these small RNAs can respond to stressful situations in the environment. And though maybe when there's a stress, the worms will change, and the small RNAs change the health or the well-being of the germline, and consequently would affect this pheromone secretion and mating, perhaps in a transgenerational manner. So to check this, what we did is we tried to see whether the worms become more attractive when we stress them in different ways. And we checked a variety of stresses. I won't show you all of this in the interest of time. I will just show you the most striking result, which is that when worms are grown at high temperatures, and high temperatures, I mean 25 degrees Celsius instead of 20, they become more attractive. They become more attractive, and their attraction depends on the presence of a pheromone receptor that is called SRD1 in one particular neuron type, AWA, in the male. And this receptor, we don't know what's the exact chemical structure of the pheromone, but we do know that this pheromone is the sensing of the pheromone depends on this pheromone receptor. So it seems to be mediated. Its attraction of the worms seems to be mediated by the same mechanism that was described by Maureen Barg and Paul Sternberg. As a positive control, in all our experiments, we use FOG2 mutants, which don't produce any self-sperm. So they are always super attractive, as you can see on the right of the graph. Okay. Being attractive is one thing, but mating is a different thing, because the experiments that I told you about now, there are no even hermaphrodites on the plate. There's just the odor that was extracted from the hermaphrodites in the males, which moved towards it. But we wanted to see whether they actually end up mating more with the males. So we use males. So we did a mating competition experiments between worms that were grown in 25 degrees and worms that were grown in 20 degrees. And we use that we can distinguish because they are labeled differently using a fluorophore. And we use males that have fluorescent sperm that we can track. And then we put them on the same plates with the males. We remove the males after an hour, and we see which type of worm mated more, just by looking at the fluorescent sperm. And what we can see, you can see the red is the fluorescent sperm, and the green is the worms that were grown at 25 degrees. And we see that when the worms were grown at a high temperature, they are more attractive. They not only more attractive, they also mate more with the males. So it actually leads to fertilization. The question, of course, the million-dollar question is whether this is inherited or not. This is why this is the most interesting part. And so what we did and the answer is yes. What we did are experiments where we grow the worms at 25 degrees, this slightly stressful temperatures, and then we move them to 20 degrees, and we want to see whether they are still attractive when they are at 20 degrees. So we extract the odor from the worms at the F1 generation, F2 generation, and so on, and we see that they continue to be attractive, although they are at the non-stressful temperatures for at least 3 generations, and then it stops, this transgenerational effect. And just like in the movie Groundhog Day, for the effect to be transgeneration, they have to be grown in the high temperature for a few generations. If you just grow them for 1 generation, they didn't acquire enough information. We don't know what the mechanism for that. And so for the effect to be transgenerationally transmitted, this is very important. How is this effect being inherited? Is it via the small RNA inheritance machinery, like our previous studies suggest, or a different mechanism that we still need to discover? To check this, what we did is we wanted to see whether the inheritance of the attractiveness when you grow worms in high temperatures depends on an argonaut, a protein that carries small RNAs that is called HRD1. This stands for heritable RNA deficient 1. It's one of the many argonauts that C. elegans has, and one that is necessary for inheritance of small RNAs in many different assays. The problem is, so we wanted to see whether this happens in HRD1 mutants, the inheritance. The problem is that HRD1 mutants become sterile at 25 degrees. So we couldn't do this experiment. So what we did is we created the Degron tagged HRD1, which is really an inducible, which allows us in an inducible way to deplete HRD1. We express T1, which is an enzyme that degrades proteins that are labeled with this Degron, just in the germline. And it works only when you add auxin, which is a hormone from plants. So we can deplete HRD1 whenever we want in an inducible way. And when we did this, we saw that when we deplete HRD1, we stop the inheritance. So when the worms can't inherit small RNAs anymore, they do not inherit the transgenerational attractiveness from their parents. So it is mediated by heritable small RNAs. There are additional controls that I won't get into from, with a tag, without a tag, with auxin, without auxin, and so on. Okay, to see which small RNAs are working here, we did a screen of checking whether attractiveness and inheritance of attractiveness happens in many different mutants, defective in different steps of RNA processing. And we found 4 different mutants that are prematurely attractive. They are attractive already when the worm is young, when it shouldn't secrete the pheromones. And we validated the results of this screen. And we found that all of these 4 mutants are indeed more attractive, and moreover, they mate more using this fluorescent sperm assay. However, only one of the mutants, it's a double mutant called MEG3/4, was transgenerationally more attractive. So we focused on it for the rest of the story. So if you take these worms, these double mutants, MEG3/4, I'll tell you about it in a second, they are more attractive. But also if you cross out the mutations, the wild-type descendants of these worms continue to be more attractive for at least 3 generations. Moreover, this inheritance from the mutants depends on the same Argonaut, HRD1. So in a triple mutant, MEG3/4 and HRD1 mutants, this transgenerational attractiveness doesn't happen. So it seems like the MEG3/4 pathway of small RNAs is important for this transgenerational inheritance of attractiveness. And we've shown in a previous paper from 2019 that MEG3/4 are required for transgenerational small RNA inheritance. They are very important. What they do is they are core components of the germ granules in C. elegans, where the decision of whether to silence a gene or not happens. And these are liquid-like condensates, these granules, where the mRNA, when it moves out of the nucleus, it goes through these granules and it gets selected for transgenerational silencing or not. And in the mutants, there's a disruption of small RNA inheritance and the choosing of the targets that lingers for multiple generations. Here you can see these granules around the nucleus in the worm. How do the small RNAs affect transgenerational attractiveness? We suspected that this happens because they are misregulating sperm genes, which we saw in sequencing of mRNA and small RNAs. They have a vast misregulation of sperm genes. The sperm is still functional because they are fertile. However, they have a reduced brood size. So to know whether they are defective in the sperm or the oocytes, what we did is we compared the brood size when they self-fertilize to the situation where they are fertilized by males. And when the males supply the sperm, there's a rescue of the brood size defect, which means that normally they don't produce enough functional self-sperm. And we think this is because of a transgenerational misregulation of sperm genes. And when the sperm is not there, the sensor senses the absence of sperm and starts secreting the pheromone, even when the worms are young and should, in theory, not secrete the pheromones and still have sperm. And the bottom line, and the most important thing is, can it affect evolution? Can it change the small RNA inheritance, change the structure of the hardwired genome, the DNA genome, and the mixing of genomes, the spreading of alleles in the population? Because if you will mix your genome more, then you will dramatically change the allele distribution and the structure of the population, and eventually the genomic variability that evolution can select from. So to check this, what we did is we took advantage of the fact that these wild-type descendants of MEG3/4 mutants are more attractive. And we took these worms, which are mutants, but transgenerationally more attractive, and we put them together with worms that are also wild-type, but they come from lineages that were always wild-type. So you're comparing the one with the same phenotype, with the same genotype, but a different ancestry of attractiveness. And we put them with males and let them, and we can distinguish between the two lineages because they are labeled with fluorophores. And then we just do a lab evolution experiments where we let them grow with males for multiple generations. And in every generation, film all the progeny and quantify the number of fluorescent worms versus non-fluorescent worms using an automated software that we called Worm Machine that we developed in the lab. And then we see whether the worms that descend from the attractive lineage take over the population, spread their genes more because they mate more because of this transgenerational attractiveness. And to know whether this effect depends on secretion of the pheromone, we do these huge experiments in parallel in wild-type background of males or in males that don't have the SRD1 receptor which senses the pheromone. So we can evaluate the contribution of the pheromone to the process. And we see that the worms that descend from the attractive lineage become more prevalent, more common in the population across generations, so they contribute the alleles more. So this transgenerational attractiveness helps spread their alleles in the population, and therefore could potentially affect evolution. And you can see that the number of males increased dramatically. Whenever worms cross, they make 50% males, and this is transgeneration. And when they just self-fertilize, they make less than 1% of the males. So over generations, this would create a very big effect. So just to summarize, what I hope you remember is that small RNA inheritance in C. elegans is a regulated process that has a dedicated inheritance machinery. Neurons can affect the transgenerational inheritance of traits, and stress can affect transgenerational attractiveness, therefore also the mixing of the genomes in the next generation, the spread of the alleles, and potentially the genetic composition of the population of worms. So this is just, I would like to thank our collaborators, Yael and Lilach from Tel Aviv University also, who are computational biologists that also modeled all of this to see when it will be adaptive. I didn't present it. And also Shai Shacham from Rockefeller University, who is also collaborating with us on this project. And Itai from my lab has spent the last year in his lab to do some of the experiments. And I would like to thank everyone in the lab and also the funding agencies and you for listening. Thank you very much.
Right, thank you, Oded.
Thank you.
Very nice. So, do you see the Q&A window on your Zoom?
I do, but when I press it, I don't see the questions for some reason.
That's okay. Then I can lend you the question. Here we have Safia that asks, what's the physiological relevance of disinheritance?
Right. That's a funny question about the physiological relevance. It's because I always ask this question as a joke on Twitter, which is why I think it was asked to me. But I think that by now we know that transgenerational inheritance of small RNAs affects many physiological traits in the worms. Here it's really crucial because mating, what's more physiological relevance than this, or the brood size or the amount of males in the population. This has tremendous significance. But also transgenerational inheritance of small RNAs was shown in the fact to affect aging, to affect the size of the worms, to affect their ability of the worms to stand, to survive starvation, to affect the worms' ability to survive infections of pathogenic bacteria, and also a general response to stress. So by regulating multiple genes in the germline, transgenerational inheritance of small RNAs affect many extremely important physiological traits. I hope I answered that question.
So you have a second question here from Malcolm. What is the ethological import of the 3-5 generation memory? What about longer or shorter, and whether you think other mechanisms will fill in those time spans?
Yeah, so this is a great question. So I'll separate the answer to two quick answers. First of all, the population, the duration of the response at the population level on average is 3 to 5 generations. Okay, and this is mediated by this timer that I briefly described. However, while the worms are genetically identical to one another, if they descend from the same mother, there's large variability in the maintenance of the response between different sisters. And we recently shown in the paper from last month that in some worms, the effect lasts for tens of generations. So some responses can last a lot longer than 3 to 5. The other part, the other answer is that while the response is typically short-lived, only a few generations, what I've shown you today is that even a short-lived effect can affect, in the end, indirectly, via the mixing, by increasing the mixing of genomes, the hardwired genome, and dramatically reshape the population structure. And once you have a change in the DNA, which is indirect in this case, it will be long-lasting and could potentially stay indefinitely, or these alleles can be selected. So I think that even short-lived effects can create an effect that will, in the end, have an evolutionary significance.
Thank you.
And regarding the environment of the worms, we don't know too much about their natural environment and how this plays out over there, I admit.
So before we pass to the other speaker, I have a curiosity. I'm not in the field at all, but then looking at your data was quite surprised about, let's say, relatively low variability that you see through the intergenerational process. So what I was wondering is, once these mechanisms start in a whole population, whether each individual would undergo a very parallel and very strict type of change, or whether you can have divergences in much larger or smaller populations.
Right. So first of all, I need to explain, I didn't explain this, that every dot that you saw on the graph is in fact a plate, an experiment with 100 males. Okay. So if you see 100 plates, these are 100 experiments of 100 males. So this is something I should have said. So the numbers are very big. Still, there are differences between individuals. And what we've shown in this paper from last month in Cell is that if you take one mother, then the descendants of this mother will be very similar in terms of the response of the intensity of the response. But if you compare to the descendants of another mother, then they are totally different. So right at the F1 generation, there's a variability event that creates that when you start the response, it creates a great variability between the descendants, and then it becomes quite stable and quite uniform between the individuals that decide from a particular mother.
Sounds good. Well, perfect. Thanks a lot for that, everyone. Clap. Thanks a lot for the discussion as well. And now we move on to the second speaker of today, Dayu Lin from Langone Center at NYU, that will give us a presentation entitled The Neural Circuit of Aggression, Innate Yet Flexible. Thanks a lot, Dayu, for being with us, and the floor is yours.
All right, thank you. Can you see it all right? Okay, great, thank you. Okay, so I'm going to tell you a little bit about aggression. So we're going to change the gear towards the vertebrate for a moment. So aggression, that's the main focus of our lab. It is a very essential behavior for survival, and it's pretty much universally observed across animal species, actually including worms. So the aggressive behaviors, because it's so essential and it requires so like little learning, and I show an example of that. The neural circuits of aggression was believed to be more or less developmentally hardwired. And the effort has been for a century almost trying to identify where those locus could be that's essential for generating those behaviors. And early studies with electrical stimulation find that hypothalamus is likely a key region for generating those behaviors. And in those animals, in the cats, in the early days, basically when you stimulate the hypothalamus, if you stimulate the median hypothalamus, the animal shows this defensive rage behavior. And if you stimulate the lateral hypothalamus, and it shows this predatory behaviors. And moving forward, about 10 years ago, at the time when I was still a postdoc, and identified that this tiny locus called the ventrolateral part of the ventral medial hypothalamus, short as VMHVO, being an essential region for the generation of aggression in mice. So specifically, when we activated these regions, even in the naive animals that has never experienced any fighting before, that's kind of important, showing that this is really an innate behaviors. And then it will start to attack a conspecific, as well as in this case, actually an inanimate object, which is not really its natural target. So this sort of artificially increased activity in these regions, causing its attack to just super targets. So far, what I've told you is that actually this circuit is quite hardwired. The behavior is innate. But yet, actually, aggression is extremely variable across individuals. So if we got a batch of mice from Jackson or Charles River, and we just put them simply in a resident intruder task. So the mice, they are kind of aggressive beings. So usually if a male, they are in their own territory and you put another male there, then they naturally start to attack each other. And what we found is that if you're just doing any experiment, if you feel curiosity, you can try it out in your lab. But what we find is there is a very diverse behavior. So some animals will attack quite quickly, others will just never attack. So this behavior variability, honestly, is really going to be devastating if you study any other regions like field conditioning, if there's that kind of diversity here. But for aggression, that's the reality. And the reason that it's being so flexible, we think that a part of it is because there is a lot of moderation of these costly behaviors, especially with experience, such as a losing experience and a winning experience. In fact, this effect is so strong, there are terms to describe it. The winning makes the animal more aggressive, called a winner effect, and the losing makes the animal less aggressive, called a loser effect. And actually from a decision-making point, this is actually quite important because every fight is an assessment of your fighting ability against your opponents in your same species. And if you win, that suggests that you have a good chance of winning. So in the next time, you should probably engage in those behavior more, as winning could lead to an increase of a resource, a secure resource, versus losing that says, well, you kind of suck at it. So basically try not to get in there. So the animals start to become more reluctant in engaging those behaviors. So the winning and losing really changed the behavior dramatically. Here, I'm going to show you an example here. So this is a naive animals, and it's the first time they encounter a male intruders. So you can see that the animal is extremely curious and interested in this male intruder. So it's repeatedly investigating it. And here I'm going to quickly just speed up the video a bit because otherwise we'll be spending the whole talk just watching this videos. So it takes about a whole 10 minutes for this mouse to actually start actually the first attack. So hopefully they're coming up soon. And now this is a 10 minutes in the test, and you can see this animal start its first bite of the animal, and then with some tail rattling and being more engaged in those behaviors. And then now this is really another video showing how the winner is going to react. So this animal has winned a couple times, so he knows the drill, he knows that the male comes in, and then you can see, just don't blink. You can see that it goes towards the other intruder and attacks immediately. It doesn't even investigate a bit, and it will be engaging in these fighting behaviors. So the latency to attack is significantly decreased, and also typically will spend more time on engaging those aggressive behaviors. So here is another example, which is a defeated animal. And this animal has been defeated by an aggressor introduced into the cage for a few times now. And this intruder, though, is a benign intruder. It's a not aggressive intruder, and it's not going to actually initiate attack towards this resident mouse. But you can see that the resident mouse is very afraid of it, and it was running away from it. And in the entire course of a testing, which is typically lasting 10 to 15 minutes, the resident mouse didn't initiate any aggressive behaviors. So apparently those experience of winning and losing can drastically changing the readiness to express the aggressive behaviors. And a key question we hope to address is how those experience can change the aggressive behaviors. And obviously, one of the regions we could start to, we will first focus on is the VMAT-VO. This is a critical site for the generation of aggressive behaviors. Does the winning and losing experience changing the neural circuits, especially at the VMAT-VO? So to address this question, so what we're trying to ask is whether the winning and losing experience will change the neuronal responses of the VMAT-VO cells. The VMAT-VO cells, they not only respond during aggressive encounters, but they actually also respond during social investigations. So they will respond to those aggression-provoking olfactory cues. So one thing is that in order to asking whether the response changing or not, we need to change it. We need to control the behavior. As you can see that the behavior is changing dramatically. The time they spend on investigation and attack will differ drastically after winning and losing. So we're trying to control that behavior variables to make that consistent across before and after the experience, and yet we're able to look at the responses of the cells. So we will now be sure that any changes in the response is not due to the behavior, but due to the property of the cells themselves. So in order to achieve that, student in the lab of Shali, who is really leading this project, what she decided to do is in a how to fix a paradigm. So in this case, what she did is to express GCAMP6F in the VMAT-VO and then implant an optical fiber on top of it, so which allows us to track the population responses of the VMAT-VO cells. In this case, we're actually focusing on the estrogen receptor alpha cells in this area because this population has been suggested to find to be crucial for the behavior. And then what we did is we're using this how to fix the prep. So the recording animals is running on a wheel, and then we will present this anesthetized stimulus animals to this recording animals, and for 3 days to get a stable baseline. And then in one group of animals, they were going through the repeat winning, and then in another group of animals, they were going through repeat losing. So to control the winning and the losing, we're basically giving different intruders introduced into the cage of our recording mice. And then we will probing the responses twice during this process, and in the end, we're recording the responses to this how to fix a prep again for 3 times. So what we see here is that actually before the weaning, the response to the male and the female is actually quite similar. This honestly is a surprise to me to begin with, because this area, I know it for its importance of aggression, but yet before the animal become aggressive in these naive animals, the response to the male and the female is actually quite similar. And then the animal goes through 4 wings and it goes through 12 wings. And you can see that over the course of the weaning, the male responses become increasingly going up and the female responses actually is going down. And this is just a summary from the multiple animals. We see this trend across the animals. And we can also quantify how this the changing responses as how it's related to the aggressiveness of the animals. So we find that as the animal become more and more aggressive, which reflected in the amount of time they spend on attacking, then this response become more male biased. So this is as reflected as the male to female response ratios. So and we also find that this is really dependent on weaning, not just the social interactions. As I told you earlier, some animals, they just don't attack no matter what we do. We expose them to those weak intruders repeatedly, but they just only engage in friendly interactions, but never actually engaging in a real fight. So in those animals, we call it the social interaction animals. What we find is that the response is quite consistent across the board. After 12 interactions, the response doesn't really change. And how about the defeat? So defeat, again, we see that before the defeat, the animals spend approximately the same amount of, the response to the male and the female is quite similar to each other. And then with the repeated defeat, we see a different pattern. So here what we see is a general dampening of the responses to both the males and the females. And if we look at plotting this on a time course, we see that usually during the defeat, the response change is quite dramatically decreased and then stayed at a relatively low level, which is a little bit different from the aggression. From the winning, as we see, is a gradual increase of the responses towards the male. So here, what we just summarize those data, what we find is that with the winning, the response towards the male is significantly increased, while the response towards the female is significantly decreased. With the social interaction, there's not really much change. And then with the defeat, we see the response to both male and the female are decreased. And then we can kind of look at this data from a different perspective, just looking at how the response to the male and the female compare with each other before and after those experiences. So what we find is that before the experience, the male and the female responses are actually quite similar. And after the winning, the male responses become significantly higher than the female responses. And after the social interaction or after the losing, the male-female responses remain similar to each other. So this is actually quite interesting. So if you look at this panel, only in these conditions, which is only condition the animal is becoming really aggressive, then there is a strong male bias, the responses in the VMAT-VL ER alpha cells. Okay, so now we see that the VMAT-VL cells are clearly changing its responses towards the male and the female as the animal going through those experiences. And a key question, of course, is what is the underlying mechanisms that cause those in vivo response changes? So we thought that there are three possibilities. One is that there is a change in the synaptic input. And this synaptic plasticity is probably the best known plasticity in the brain. And then the second possibility is actually the VMAT-VL cells has drastically changed its excitability. And the third possibility is that the change actually happens upstream of the VMAT-VL, and the VMAT-VL just simply inherit those changes. And these experiments are led by Dongyu, a postdoc in the lab. So, so what we do is perform the slice recording, and we're taking the slice from the animals that are going through the repeated win, lose, or social interactions, and looking at on a slice how the synaptic transmissions and the intrinsic properties of the VMAT-VL cells has changed. So here is first looking at the synaptic inputs. Well, not much to see here. Basically, what we basically observe here is after 10 days of the winning and losing, we actually surprisingly do not see a huge change in the synaptic transmission. If you squeaky eye, maybe there is a little bit of a synaptic transmission increase, EPSC increase in the winners in comparison to socials, but the losing kind of in between. So we don't think that could be the mechanisms that are really driving those huge change in the responses of the VMAT-VL cells. And the same thing for the inhibitory synaptic inputs. We really don't see a huge change here, although the winners maybe has a slightly lower iPSC frequency in comparison to the social animals. But we do start to see changes when we look at the intrinsic property of the cells. So the first thing we look at is the renal base and the resting membrane potential. So the renal base is basically the amount of current you need to inject into a cell in order to get the cell to fire an action potential. So this could be a negative value because if the cell is firing spontaneously a lot, then you need to inject some negative current to clamp it down so that it doesn't fire so much. So what we see here is that in the social and the losing animals, basically you have to on average inject some positive currents in order to get the cell to fire an action potential. But in the winning animals, because the cells are actually spontaneously active, so you don't really need to inject the currents. And in some cases, we need to inject the negative currents. And then for the resting membrane potential, we also see a significant difference. We see in the winners, the resting membrane potential is more depolarized. So the cells are sitting at a more depolarized potential, and which makes it easier to reach the threshold for an action potential. So further, when we look at this current steps, the current spike relationship, then we see a bigger difference even. So in the social animals, as we injected more currents, naturally, you will see there is more cells firing spikes, the cells are firing more spikes as we inject more currents. And then in the winning animals, we see that across those step levels, there are more spikes being fired as we inject more currents. And in losing animals, which is really intriguing, we see that the cells initially are indeed firing spikes, just like the social and winner animals. But as we inject more currents, the cell enters this depolarization block. So it fires a few spikes, but the action potential size is significantly quickly diminish, and then it enters this depolarization block, and it can no longer follow those input, the positive currents input. So here's a summarization. What we see is basically, as in the social animals, as we inject more currents, we see more spikes. And then in the winners, across all the current levels, we see an increase. So you see the maximum firing rate. Basically, after a certain level, they start to plateau. They don't fire more. But we see the maximum firing rate in the winners is higher than in the social animals. And in the defeat animals, although initially they track this firing rate changes as the social animals, but then quickly the firing rate actually decrease as we inject more currents, which is really suggesting that this defeat animals, the cells, they're in a state that's really not capable of taking the positive synaptic, excitatory synaptic inputs to drive them. They're kind of resistant to be driven. So now, knowing that the cells has those firing rate properties has changed, the next question that we hope to ask is what kind of channels really are responsible for those firing rate changes? So the first clue we look at is basically just take a close look at those action potentials. So this is an idea provided to us, advised to us by our great colleague Dick Chen, who is a great biophysicist. And then we said is, the action potential, so you can measure this maximum rate of rise. So the maximum rate of rise is how quickly the action potential can rise. And this is really reflecting how much sodium channels you have, because the more sodium channel you have, and the quicker the action potential going to rise. And the amount of sodium channel available is a function of voltage. So we need to look at the relationship between the takeoff voltage and the maximum rate of rise. So that's exactly what we did. And what we see is that in the winners, there is a clear rightward shift in this rate of rise of as a function of a takeoff. So what does it mean is that if you just take a look at this particular time point of the takeoff potential, what you see is that at a given takeoff potential voltage, there is a significantly faster rate of rise in the winners in comparison to the losers or the social animals. So that would suggest that there is a lot more sodium currents in the winners in comparison to the losers and the social animals. And then that conclusion can be further tested with the voltage clamp. So in this case, what we did is to clamp the cells on to -80, and then we will step it to different voltage and looking at the currents. So we see is that indeed, this is the sodium current. So this is largely at the -40, it reached the maximum value. And that maximum value is significantly higher in the winners in comparison to the losers and the socials. And this is just a summary across the many cells. So, but one thing is that, well, the winners has a significantly more sodium channel, which makes sense, but the losers doesn't have a significantly less sodium channels, but nevertheless, we see this depolarization block. The depolarization block is actually a reflection of the sodium channel become less and less available, as you can see the action potential becomes smaller and eventually stop firing altogether. And the depolarization block is actually critically dependent on some other properties of the sodium channels. One is the inactivation properties. So the sodium channels, they can be in the open state, closed state, and the inactive state. And the inactive state is also a function of a voltage. So what we decide to do is to probe how the voltage dependent sodium channel inactivation properties. So to do that, we basically step the voltage clamp the cells to different voltage levels, and then step it to the maximum levels that we get the maximum sodium currents. So, and then we get a ratio of basically the fraction of the sodium channels available, the sodium currents as a function of a voltage. So what you see here is basically in the losers, in the winners and the social, their function as a function of a voltage, their amount of sodium channels available and the function of voltage is quite similar. But in the losers, we see a significantly downward shift. So what does that mean? So minus 60 is where the cells sits normally, so in the resting membrane potential. And if we just focus on this particular voltage point, you see that in the resting membrane potential, there's about 80% of the sodium channels are ready to go in the winners and losers, but there's only approximately 50%. So there is a significantly less sodium channels available. So the total amount of sodium channels for the losers and the social animals are similar, but the amount that's actually ready to be opened in losers is significantly decreased in the resting membrane potential. And the other property of the sodium channel inactivation is actually it's a time-dependent recovery. So what does that mean? So basically, sodium channel, after it opens, it takes some time to slowly go back to this ready state. And that the faster it can go back, the faster it can be recruited during the next round of the action potential generation. So here we look at basically how quickly it can recover. To do that, we're using two pulses. So this pulse, we're going to activate all the sodium channels. And then after a variable amount of times, we activate again. And we're asking for the second time, what's the fraction of the sodium currents we get in comparison to the first? So the bigger it is, then it means that the more sodium channels are basically back to this ready state. And here's the winner animal. You see that with a short interval, you see there is some fraction of the sodium channel not available. And as a longer interval, then basically all the sodium channels become recovered. In the losers, we see this time-dependent recovery is become much slower, especially during the period when there's interval is short. So this is a summary here. Again, you see that the winners and the social animals, they recover very fast. Within 50 milliseconds, they more or less all recovered. But the losers, it takes much longer time. It takes about 200 milliseconds to reach the levels that the winner and the social can recover in 50 milliseconds. So which means that really they cannot fire more than 5 hertz in that sense if you are repeatedly firing those cells. Okay. So the currents that's important for the action potential generation is not just the sodium, but also there is a potassium currents and the calcium currents. So I'm going to tell you a little bit about those currents as well. So the potassium currents, the first one we look at the potassium currents, we use again voltage clamp and we block the sodium channel in this case. And we can step it to different levels and evoke the potassium currents. So the potassium currents, as they become stable, here, so this reflecting the potassium currents that's not inactivated, not transiently inactivated. So they're quite stable as we go over time. And you see that that part seems to be quite similar across the social loser and the winner animals. However, if you look at this tiny blip at the beginning of this currenting voltage clamp, you see there's appear to be some differences. And this is called A-type current. And the A-type current, it's a transiently activated and then become inactivated. So it's a very transient. And this current is important because it actually activated at a relatively low voltage, low membrane potentials. So when there's more of those currents, it kind of counteracting the sodium currents. So it makes it harder for the membranes to become more and more depolarized and preventing the action potential from generating. So to measure that current, what we did is to use a pre-pulse, and that pre-pulse is going to transiently activate this A-type current, and then the A-type current become all inactivated, and then we step to a 40 millivolts, which is where basically the A-type currents, the potassium currents is maximal, and then we can subtract this current with or without this pre-pulse, and then what we got left is this A-type current. So we see here is the winners, the A-type current is significantly decreased in comparison to the loser and social animals. So basically the winners has less of the A-type currents, which helps the action potentials to be generated. And the last, we look at the calcium currents. So for this calcium currents here, we block the potassium currents and the sodium currents, and then we step it from the -80 to different levels of the voltage. And we see that the calcium currents differs when the voltage steps are lower, but not when the voltage steps are higher. So the calcium currents are composed of two components, like generally speaking, roughly speaking, and there is a high voltage components and a low voltage components. So the high voltage components, this is a high voltage activated calcium currents, and it's also called L-type because it tend to be long lasting. It's not the transient. So in order to isolate this currents, we use the we have hold the cells at a -55, and during this voltage, the low voltage activated calcium currents are going to be transiently activated and then deactivated. So what we see here is the purely high voltage activated calcium currents, the L-type, and then we can get this L-type current, the voltage current relationship, and you see that it's actually quite similar in the social loser and the winner animals. And then using the total calcium currents minus this L-type currents, what we got is the T-type currents. And we see that the T-type currents is significantly lower in the loser animals. And again, the T-type currents, because they are activated at a low voltage, so they are active, they actually facilitate the action potential generation as well. So has less of it will make it harder for the action potential to be generated. And the T-type currents also contribute to this rebound action potentials, because when the cells are hyperpolarized, then all the T-types are ready to go. And when the membrane go back to its normal levels, then the T-type helps the action potential to be generated. So the rebound action potential also reflecting whether how much T-type currents you have. So inconsistent with the idea that in the losers, they are less T-type currents, we see that in the losers, their few cells has this rebound burst. So this is supporting that basically the T-type currents is lower in the loser animals. So to summarize, what I showed you is that the social experience is really changing the aggression level in dramatic ways. It can enhance the aggression, or winning can enhance the aggression, and the losers can completely abolish aggressive behaviors. And that change is associated with changes in the VMH-VL cell responses to the aggression provoking stimulus, particularly the male cues. And that changing responses is closely related to the changes in the VMH-VL excitabilities. So because of the VMH-VL cells, these are dedicated cells for the aggressive behaviors. So basically it can employ some very dramatic plasticities of this intrinsic properties of the cells itself to ensure that the aggressive behaviors in the losers are not going to be released. And those changes in excitabilities is due to changes in a variety of the ion channels, from the sodium channels, the potassium channels, and the calcium channels. And then now, of course, we're trying to understand the next level of questions, which is basically what is the molecular mediators from the social experience to changing those ion channel properties. And with that, I want to thank you very much. These are the two really the two persons who did all the works I just talked to you of Shali and Dongyu. They make a great team. And I want to thank our collaborators, Sumin and Nick, who has helped those who give a good advice on the slides recordings and also the fundings. So with that, I'd like to thank you all for listening. And I can take questions if there's time.
Thank you, Dayo.
Thank you.
Thanks a lot. Sure, we can have time for questions. Maybe we do as we did before with Oded, and I shoot the questions that are launched here on the Q&A chat. So you have a question from Vasil that asks, and Vasil, I'm choosing one of yours. How different on neuronal level is a social aggression compared to the predatory aggression? Do you know if the VMH cells also respond to prey?
Okay, let me see. Stimulate, how different, okay, social aggression to the predatory aggression. Do you know if the VMH also respond to prey? I don't think so. I don't think so. Actually, I think the VMH is quite dedicated for the social aggression. For example, the mice, we never really look at like mice eating crickets and whether that activated the VMH-VL cells. But when the mice are showing infanticide, which in part is a little bit of predatory-like behaviors, we do not see the VMH-VL cells become activated at all. So I think it's quite specific to the social aggression. Okay. So I see another question is either a question from Vasil. What do you observe when you stimulate the VMH-VL and no target available? Is animal attacking the air? Is it trying to find a target? It's trying to find a target. You can see that there is an increase in the movement a little bit, and but it's not going to bite the air. It's not. It has to have some reasonable target in order to release that behavior. Okay. And Steve has a question on how to fix the sniffing experiments. I assume you had to fix the sniffing experiments only allow access to volatile orders. What do you know about the role of non-volatile orders sense-divided vomeronidal systems in driving VMH-VL activities? Yeah, so the VMH-VL definitely get inputs from both the main and accessory olfactory systems. And the volatile does seem to drive it pretty well. We often see the biggest response when the animal is introduced and it was actually at a distance. So, which is supposedly will not be activating the VNO. So we have not, I guess, specifically trying to look at if we block the volatiles. We do see often, you know, when the close contact tend to activate the cells further. So I think there is a contribution of the accessory olfactory systems inputs, but we never try to deliberately separate them under these conditions. Have you looked at, okay, Kishore has a question. Have you looked into the possibility that consecutive losers have propensity into developing anxiety like behavior? Yes, we didn't deliberately look at, but they definitely show social avoidance. And in fact, the chronic social defeat has been used as a model for chronic stress and depressions. So yes, I think that there are other aspects of the behaviors will be changed as well with the chronic defeat. Although at the VMH-VL, you know, given the function of these regions, I think we are mostly looking at the aggressive behaviors and the relevance of the defeat to the changing that behavior. Okay. I think that's all the questions.
Before you close, I think I have a curiosity instead to ask you. So I was wondering whether a social hierarchy plays a role in this aggression, winning and losing. So we know that in a group of animals, often you have, among males, especially alpha males or the leader ones. And I was wondering whether this has anything or any correlative features, behaviorally speaking, with aggressivity of being a loser. Have you looked into that?
Of course. I mean, I think the social hierarchy is established by losing and winning.
Exactly.
Yes. So I think so the animals that lose a lot, of course, they're going to be in the bottom of the social hierarchy, and the ones that wins always are going to be at the top of it. I think if you take a higher hierarchy animal, basically is an animal with a lot of winning experience, and let it go through defeat just by exposing it purposefully to a very aggressive mouse. In fact, it appears that this winning experience can have some protective effect to help the animals not to kind of experience the drastic consequences of the defeat, such as developing strong social avoidance. In some animals, if you go that kind of manipulation, you can get the animals to both show aggressive behaviors and being defeated. So I think there's some protection mechanisms there. But you know, in this experiment, we didn't try to manipulate the winning and then losing and so on and so forth. But I think there's definitely interactions between the two.
Yeah, it will be curious to see whether there is any correlation between your active conductances currents and the role in social hierarchy at this point.
Yes, I would definitely think that's likely the case. Yes. Yeah. So the winning change it, and then so the cells would not undergo the changes with the defeat as much as if they're just purely defeated.
Right, right. Okay, thank you, Dayo. Thanks a lot for your contribution. And now we move on to our next speaker, Will Norton, Leicester University, who's going to tell us about the genetic basis of mirror-induced aggression in zebrafish. Thanks a lot, Will, for jungling again through the logistics, and the floor is yours.
Thank you very much for the invitation. Do you see my first slide? Yes, everything fine, thanks. So today I want to tell you about a new project we have in the lab, which we only got this data very recently, a few weeks ago. So I'll be excited to hear if anyone has any thoughts or comments about it at the end of my talk. So my lab uses zebrafish as a model organism to study aggression. And as we've already heard in the previous talk, aggression is an important adaptive behavior that animals use to establish social hierarchies, to fight for resources such as food or territory, and to protect their offspring. And what we're really interested in trying to understand is the neural circuit in the brain which controls an animal's aggressive response. So high levels of aggression can be maladaptive and lead to injury and death, and levels of this behavior have to be very carefully controlled. And we wish to understand how the brain processes stimuli in order to tailor an appropriate behavior response. So for example, there can be a stimulus approaching, it could be a conspecific zebrafish, and that will be processed within the brain to lead to this behavioral response. And on my slide here, you can see a cartoon of the adult zebrafish brain. We have anterior to the left here, we've got the forebrain, the optic tectum, which is the visual processing area of the zebrafish brain, and then the hindbrain and spinal cord. And you can imagine that if another fish approaches, we have visual input that's processed within the optic tectum. There's areas within the forebrain which can give valence to the stimulus, such as the amygdala, to tell you whether it's aversive or appetitive. And finally, the hindbrain tailors motor output to decide whether the animal should fight the stimulus or retreat. There's two ways in which you can measure aggression in zebrafish, and they're both relatively simple. You can simply place two fish in a tank and record their interaction, and both male and female fish will fight each other, and they show characteristic agonistic postures, including trying to bite each other, circling, and extending the fins so that they look larger. The second way to measure aggression is to use a mirror test, which has been used in many species, including fish species. And I have an example of this depicted on the right-hand side of my slide here. We have a wild-type zebrafish fighting its own mirror image. I'll just start it again. We place the mirror outside of the tank. And once again, we can look at these agonistic postures, which include biting against the mirror, pushing against the mirror, and moving the tail fin very rapidly. We know from a number of studies that there's good correlation of aggression levels in these two paradigms. An animal which is highly aggressive in a dyadic fight will also be highly aggressive when fighting the mirror. But there's also studies that suggest there are differences between the two types of aggression. For example, there are different hormones activated in a dyadic fight compared to the mirror, and different genes are activated as well. And so the two aren't exactly equivalent. In our lab, when starting a new project, we tend to use both of these techniques to check the aggression level of a zebrafish line. And then we favor the mirror aggression because it's easier to control the stimulus, the mirror image, which the fish won't recognize, and it also has less potential to harm the animal. So using this very simple mirror experiment, we've done a number of different types of projects. For example, we characterized the aggressive and neurochemical basis of phenotypes in zebrafish mutant lines. And we've recently carried out a drug screen in which we identified novel drugs which can reduce aggression levels. And we found that both the phosphodiesterase inhibitor sildenafil and caffeine, which is an adenosine inhibitor, very nicely reduce aggression in adult zebrafish compared to untreated animals. So in a very old study in Lob Alicru's lab in Munich, when I was first starting out doing this, we were able to identify a mutant line, fibroblast growth factor receptor 1A, which is more aggressive than wild type. And that's depicted on my slide here. It's always nice to see a really old study, which you did ages ago. So here you can see our classic mirror test setup where we have the mirror placed outside the tank and 3 sides of the aquarium are covered in a white opaque material to reduce reflections. And we can see that the mutant line on the right-hand side here spends much more time fighting the mirror compared to wild type. That was a statistically significant result. And we saw, interestingly, that over time, a wild type fish, the white bars here, reduced their aggression levels, whereas the FGF receptor 1a mutants maintain very stable aggression levels over time, as if they initiate aggression normally, but unable to reduce it over time. In this paper, we were able to show that histamine signaling in the zebrafish brain active by the histamine receptor 3 is very important for this behavior. And in a recent paper, we created a histamine receptor 3 mutant line and were able to confirm this observation. And we also identified an area of the central hypothalamus called the periventricular nucleus, which appears to be important because we saw gross changes to neuroanatomy in the hypothalamus of the FGF receptor mutant compared to wild type. In a more recent study, we characterized a second zebrafish mutant line called endothelin receptor AA. And this is a receptor for a peptide neurotransmitter called endothelin in the zebrafish brain. The mutant line was identified by Robert Kelch at the University of Bath. He had this line in his facility and he noticed that it's more aggressive than wild type. And we were able to confirm that observation using our mirror test. Although interestingly, the increase in aggression shown here is not nearly as strong as that which we saw in the FGF receptor line. However, perhaps the more interesting phenotype in this mutant is a strong reduction of social behavior. The zebrafish is a weakly social species, meaning that it shoals rather than schools. And you see a group of wild-type zebrafish will form this loose aggregate in a tank, whereby one fish might disappear for a while from the group. Sorry, I moved on. But then it will come back and join the group again. And what was very obvious when we looked at the endothelin receptor mutant is that they spread out throughout the tank and increased both the nearest neighbor distance and the inter-individual distance maximally within the area given by the tank. And we now think that these endothelin receptor AA zebrafish are constitutively dominant. They have a lower level of basal cortisol and they're aggressive and they show this reduced sociality. And we did experiments where we simply introduced one mutant into a group of wild type fish as seen on the left-hand side. And we could see that the behavior of the single more aggressive animal was enough to disrupt the social behavior in the other wild type fish. And so studying mutant lines is very interesting. It enables you to identify areas of the brain which might be important. If you look at the expression of the mutated locus, you can look at different neurotransmitter signaling pathways. But we became concerned that perhaps studying a mutant line doesn't really explain how aggression is controlled in the wild type brain. And so in a recent project, Florian Reichmann, who is a postdoc in my lab, or was a postdoc in my lab, he's now just moved to Graz to set up his own group, decided to use a classic genetic approach where we selectively bred both high aggression and low aggression zebrafish lines called HAZ for high aggression zebrafish and LAZ, LAZ for low aggression zebrafish. In this project, we started by using the AB wild type fish, which were available in our lab. And we firstly measured their aggressive behavior at an adult stage at 3 months using the mirror test. We selected the most highly aggressive and least aggressive animals and inbred them to generate an F1 generation. We then measured aggression levels at 1 month using an automatic setup and software which we developed and published. And that allowed us to phenotype a large number of animals, so over 100 animals in each generation. And at this 1 month juvenile stage, we then selected the 20 highest and 20 least aggressive animals and grew them up once more until adult stages, so 3 months onwards. And we then measured both aggression in the mirror test and anxiety using the novel tank diving test, as depicted on the right-hand side of my slide here. The novel tank diving test is one of the best used tests for adult zebrafish behavior. And you simply place the animal in a novel, tall, thin arena and measure the thick metaxis, which is the preference to spend time at the bottom of the arena. An anxious zebrafish will remain at the bottom of the tank, whereas a less anxious animal will swim up in the water column towards the top. As we can see that we carried this out for 4 generations until we had an F4, and our selective breeding worked very nicely. On the left-hand side here, we have a heat map where you can see a behavior of an F4 low aggression zebrafish, LAZ, and you can see they swim throughout the tank, whereas the high aggression zebrafish spend most of the time attacking its mirror image. Initially, in the F0 generation, these AB wild-type fish showed a widespread of aggression phenotypes or levels of aggression throughout the group, as you might expect in a group of animals. And we saw similar levels of aggression in both male and female fish in the mirror test, which fits with similar studies we've already published. In the F0 generation, we then selected the 10 most aggressive and the 10 least aggressive animals, and you can see this here. And we can see that we could already separate aggression levels by sorting out from the two extremes of the phenotype. We then inbred these animals to generate an F1, and you can see it at 1 month, there is a significant difference in behavior, but it's very slight and not terribly significant. And by 3 months, the adult mature fish do show a difference in aggression levels between HAS, so that's this orangey color, and the low aggression zebrafish in blue. We saw a similar result in the F2 generation, and then by F3 and F4, we begin to see larger differences in aggression phenotype between the low and high aggression selective lines. We stopped this experiment at the F4 generation for 3 reasons. The first reason is that we had filled up our fish room and we totally ran out of space to breed any more fish, and we were also beginning to run out of time on Florian's grant. But more importantly, we saw inbreeding depression in these animals. So when we began to in-cross them to collect embryos for the next generation, they no longer bred very well. And this is consistent with information from the literature, which says that if you inbreed the same zebrafish for a few generations, they no longer breed very well. And so we decided to use these brains from these F4 animals in RNA-seq experiments. Before we did this, we wanted to see how selected the aggression phenotype we selected for was. And we first measured anxiety, as I've already said, using the novel tank diving test. And you remember that an anxious fish will remain at the bottom of the tank. On the top, you can simply see a comparison of the anxiety levels, the time spent in the top of the tank for both HAS, the orange bar, and LAS in blue. We can see in the F1 and F2 generations, there's no difference in anxiety behavior, but by the F3 and F4 generation, there begins to be a significant change, and the high aggression zebrafish are much less anxious compared to the low aggression zebrafish, meaning they spend more time swimming at the top of the tank. Another way to look at this is to look at the correlation between aggression generation, so that's on the x-axis of all these different graphs, and the time in the top of the novel tank on the y-axis. And again, this confirms that we never saw a positive correlation between anxiety and aggression in the low aggression line, whereas in the high aggression line, we did see a significant correlation in generation F3 and F4. We also looked at other behaviors to get a better idea of how selected these phenotypes are. And we next measured locomotion in the open field test. And this is simply a large aquarium where you place a single fish inside and record its behavior from above. And by looking at this, we saw a similar distance swum for both of our selection lines. They swam at a similar speed, spent a similar amount of time immobile, and no change in their angular velocity, which gives you some idea about stereotypy, because an animal with high amount of angular velocity changes direction very often. We also measured social behavior in the shoaling test, which I showed you in the beginning of my talk, and we see a similar level of both individual distance and nearest neighbor distance for both the HAS and the LAS. And finally, we looked at interaction with a novel object which we placed in the tank. In this case, it was a falcon tube filled with modeling K, which we thought might look a little bit like a predator for the fish. And we saw a similar amount of interacting time with the model in the tank for both HAS and LAS. So there are no changes to locomotion, shoaling, or boldness towards the novel object, suggesting that the change to aggression and anxiety is relatively selective. There's a larger number of studies in the literature looking at zebrafish and other fish species, which show that aggressive animals tend to be both larger and darker. And there's a very nice recent paper from the lab of Gonzo Palavieja, where he measures the coloration of fish as they're fighting. He was able to show that more aggressive fish are also darker compared to less aggressive animals. And so we wondered whether by selecting for aggression phenotype, we also saw changes in morphometrics. And indeed, this is the case. We saw that both male and female high aggression zebrafish are longer, and they have a higher height at the nape, which is a measurement taken directly behind the operculum, the cover of the gill. And so these animals seem to be larger compared to both male and female low aggression zebrafish. We measured the areas of the fins, and we saw no difference in tail fin area, whereas the anal fin area is slightly smaller in females for both the high and low aggression line, but there's no difference in males for high and low aggression lines. We also wanted to look at dark coloration, and the way in which we did this is to take images of the fish, and we looked at the amount of pigmentation in the characteristic stripes which you see on the zebrafish's body. So the zebrafish have 3 different types of pigmentation cells called melanocytes, which give the black pigmentations, and then the xanthophores and iridophores, which give the light pigmentation in the stripes. And the different stripes on the body have names, so 1 dorsal, 2 dorsal, 1 ventral, and 2 ventral, 2V, for example. We compared the amount of pigmentation by using ImageJ to extract grayscale images. And you can see globally, if you look at the level of the whole body, we see a similar amount of pigmentation for both the male and female HAS compared to LAS. However, we do see significant differences if we look at the different stripes in isolation. For example, both male and female high aggression zebrafish have a darker 2D stripe, and that's the most dorsal stripe which you can see on the body here. Interestingly, female fish appear to have more dark, so more pigmented ventral stripes, so that's 1V and 2V, but we see similar levels of pigmentation across the 2 aggression selective lines, so it doesn't seem to be linked to aggression. In parallel, we also looked at width of the interstripe interval, so that's formed by those xanthophores and iridophores. I haven't shown the data for the sake of time here today, but what we did see is that the aggressive males tend to have thinner stripes in between these black pigmentation stripes. And we think that that makes the stripes in the males bunch together more closely. And so in summary, the aggressive animals appear to be slightly larger, and they appear to be slightly darker if you look dorsally. So in the next step, we wanted to use RNA-seq to look at differentially expressed genes between these 2 selection lines. And in order to do this, we repeated our experiment. I forgot to say that in our first measurements of aggression, we only recorded behavior for 5 minutes. But in this case, we placed single animals into our aggression setup for 1 hour. And we used a longer time frame because that gives time for gene transcription to occur. And so we might see differences in the neurogenome after we allow them to fight their mirror image for an hour. As you can see on the top line, we measured quite a large number of both high and low aggression zebrafish lines, and this is the F4 generation. And once again, we saw a significant difference between high and low aggression zebrafish compared to in a 1 hour time frame, although there is a spread of phenotypes in both groups at this point. They also swim a similar distance in the tank, and they show a same time immobile, so these are those background measurements I've already talked about. In order to do our RNA-seq experiment, we then selected the 6 highest aggression male and female zebrafish and the 6 lowest aggression male and female zebrafish, and that's depicted here. And in this case, by selecting the 2 extremes, you have a very good separation of the phenotype. On average, the low aggression zebrafish, which we use in our RNA-seq experiment, show about 3 seconds of aggression compared to over 1,000 seconds of aggression for the high aggression zebrafish. And once again, we saw no difference in the distance swum and the time immobile. And you've probably noticed that I've initially showed you that we don't see any differences in behavior between males and females when we used the mirror test. For the RNA-seq, we decided to separate the animals by sex, just in case there's some kind of hidden importance for sex in the change in genes which we see by RNA-seq. So here you can see the results of our RNA-seq experiment. And you can see from the heat maps that here we have genes which show higher expression are shown in the orange and red color, whereas the light blue and dark blue color shows down regulation of expression. And we have the male fish on the left-hand side and the female fish on the right-hand side. And you can see there are a large number of genes which are either upregulated or downregulated in males for the high and low aggression line, and a very similar profile for the female fish. In general, we saw around 500 genes were differentially expressed in the female fish, and slightly less in the male fish. It's about, I think, just under 500 genes. But the overlap between the 2 sexes is 313 genes, which are differentially expressed between these 2 selection lines. You can also plot the data using these volcano plots. And in this case, this simply shows you whether the genes we're interested in are either overexpressed or underexpressed, depending upon their position on the x-axis. So here, so this shows you fold change. A reduction in expression moves it, the dot towards the left, whereas overexpression will move it towards the right. And then we have our adjusted p-values on the y-axis. And for example, we can see that some genes were extremely significantly altered in both male and female fish. And the gene which appears to show the most significant change is called arsenite methyltransferase. It's an interesting gene. We don't understand yet its function. So we know that arsenite methyltransferase is expressed in the liver, but remember we've taken the brains of these animals for our RNA-seq experiment. It's been shown to be expressed in the brains of a number of species, but the function isn't well understood. However, we do know that polymorphisms in this gene in humans can lead to schizophrenia, and there's some suggestion that it might be important for neuroplasticity linked to learning or memory. So it's a very interesting candidate to look at in the future. We used qPCR on independent samples from the F4 generation for both HAS and LAS, and we were able to confirm the top 10 differentially expressed genes. You can see them all here. I don't propose to read all the names out because that would take a time, but I have a slide right at the end of my talk which have all of these gene names written on in case you're interested, because I don't really like seeing talks where you see these kind of codes for genes and you can't understand what they are. And once again, here's our new favorite gene, which is arsenite 3-methyltransferase. Using principal component analysis, we could see how well we were able to separate our two selection lines by looking at the number of differently expressed genes. And when we look at the 200 most highly altered genes between these two lines, we can see a perfect separation of the low aggression zebrafish in blue with the high aggression zebrafish in this red and orange color. And once again, we can see that both male and female fish show similar changes in gene expression. So in order to try and get to grips with what some of these changes in gene expression might mean, we first used David pathway analysis to look at the gene ontogeny and the cluster genes in functional domains. And the two most enriched terms that we identified were for immune pathway genes, which is very interesting because there are already studies which look at differences in the immune system and some psychiatric disorders in humans which are linked to aggression. And also the chemokine receptors, which are known to be important for some types of migratory cells. And we wonder whether perhaps the chemokine receptors could explain this change in pigmentation, which we've seen in the aggressive animals, and could explain how the stripes are clustered together more closely in the male fish. There have been other labs that have done similar experiments using a mirror to look at differences between animals which fight or don't fight the mirror. But in this case, they simply took wild type animals and didn't selectively breed them for generations. Interestingly, they saw activation of very different types of genes, principally immediate early genes, but they also saw that oxidoreductase activity is important. And you can see that this is the second cluster we identified by our David pathway analysis. So once again, that's something we'd like to dig into in more detail. Next, we used a weighted gene co-expression network analysis of our RNA-seq data. And by doing this, we can identify gene co-expression modules which are linked to the different phenotypes we can find within our fish. And by doing this, we found a number of different modules which each have a color name, and that's shown on my heat map here. And in this case, a red or a pink coloration sees a significant upregulation of expression, where the green color shows a significant downregulation of expression, and the numbers tell you the p-value. So out of these, perhaps the most interesting gene co-expression module we identified is called module magenta. And again, this provides us with an interesting list of genes which we can analyze in more detail in the future. In module magenta, we have genes which are upregulated in either male or female high aggression zebrafish, and they're downregulated in the low aggression zebrafish lines. And interestingly, the genes identified by whole genome pathway analysis are identical to the ones which we found by looking at this differential gene expression. So by using two different techniques, we identified the same genes, which gives us real confidence that they may have an important role in these phenotypes which we've seen. And once again, the top gene expression which we identified is arsenite 3-methyltransferase. So in summary, we have this new project which we're still trying to understand because we generated a lot of data. We've used selective breeding to generate two lines, which are called high and low aggression zebrafish, that differ in their levels of mirror aggression. The high aggression zebrafish are less anxious, and they're also large and darker compared to the low aggression selection line. By using RNA-seq, we identified 313 differentially expressed genes between the two lines. And the most highly differentially expressed are arsenite methyltransferase, a solute carrier family member 1 adapter protein, an ADP ribosylation factor like 4AB, and a prostaglandin receptor. And furthermore, we use David classification of genes into functional clusters, and that suggests that chemokine signaling and the immune system are really important for some of the phenotypic differences that we've seen. However, there are some important limitations to our study. The first limitation we have is that we started from a lab stock of wild-type zebrafish, which has been kept in the lab for many generations, rather than using wild fish. The reason we did this is that it's very difficult to now get wild zebrafish from India or Myanmar because of legal regulations. It's difficult to take them out of the country. And also they could potentially bring diseases into our facility, which we're slightly worried about. But that means that the stock we started for would have already been fairly genetically homogeneous. And arguably, we could have seen more changes in gene expression if we used these wild animals. The second limitation to our study is that we can't unambiguously assign genes to changes in aggression. If we wanted to do that, we should have inbred the high aggression and low aggression fish for 2 generations and then rephenotype the offspring before doing our differential gene expression. And the reason we didn't do this was firstly, the inbreeding depression, which we saw, and secondly, we were running out of time on our grant, unfortunately. As a result of that, it means we can't disentangle the genetic contribution to the changes in size, color, and behavior. However, personally, I'm not too worried about this because I believe that these phenotypes co-vary. We've only selected on aggressive interaction with the mirror. And since we know that dominant zebrafish and other dominant fish tend to be larger and show a change in color, we think that these different phenotypes are likely linked to each other anyway. But it does mean that these interesting genes we've identified, we need to think about which of these phenotypes they're contributing to. And finally, we're planning to look at the expression profiles of these genes in the brain, and particularly interested in module magenta, which gives us a really good list to look at. For example, we could generate SYN expression maps of genes activated by aggression by looking at the brain areas which express these different targets. In a recent study, so this is the histamine receptor 3 study, which Florian and I published recently, we used an immediate early marker called RPS6, it's a ribosomal protein, and we used that to map areas of the zebrafish brain which are activated by mirror aggression. And here I can show you one example, just for the sake of time. This is the wild-type zebrafish brain, and we've taken a section through the telencephalon. So this is dorsal and ventral telencephalon. And you can see that following interaction with the mirror, compared to a control animal which just swims in the tank without a mirror, we see strong activation of the preoptic area of the anterior hypothalamus, and lesser but still significant activation of the medial area of the dorsal zone of the telencephalon. So yeah, thank you. And so what we saw by doing this experiment is the activation of key nodes of the social decision-making network, which is a constellation of brain areas which are known to be important to control aggression and other social behaviors in all vertebrate brains, including zebrafish. And we saw significant activation of different areas of the forebrain, including the medial and lateral areas of the dorsal forebrain, which is the paleo-amygdala and medium pallium and hippocampus in other vertebrates, as well as the ventral forebrain, including VV and VD, so striatum and nucleus cumbus basal ganglia in the other vertebrates, and the preoptic area. And we also saw widespread activation of the hypothalamus in keeping with studies from other animals, including the central, lateral, and ventral hypothalamus and the posterior recess. Finally, we're intrigued to try and understand what type of aggression we're measuring in the mirror. And so I told you in my introduction, we know from studies from a number of labs that we see differential activation of genes depending on whether you use a dyadic fight or mirror aggression. And we also see a blunted release of some hormones such as androgens in the mirror fight. And typically people have thought that mirror aggression induces a subset of aggression, but it doesn't recreate the whole phenotype, which you see in a dyadic fight. However, we still find this a little bit hard to understand because in our selection lines, we hypothesize that the high aggression zebrafish look dominant because they're larger and darker. And so when it sees itself in the mirror, you would reason that this dominant looking animal would not fight its own mirror image, whereas the low aggression zebrafish appears subordinate if you simply look at it. So we wonder why they don't fight their own mirror image in order to set up a dominance hierarchy. And so we now wonder whether we're measuring a defensive rather than an offensive type of aggression using the mirror. Typically, people have said that there's only one type of aggression, which is offensive aggression in zebrafish. So this is just an idea we're working on. We think it would be interesting to compare in other species and see if we can really see qualitative differences in the type of aggression between these two paradigms. And finally, if we put together all the things we've learned about zebrafish aggression in recent studies, we now know that aggressive zebrafish appear to be larger and darker than subordinate fish. We know that males and females show similar behavior in the mirror test, and that aggression is used to determine dominance and social interactions, so the levels of stress differ between animals in a group and also the spacing with animals in the group. We've shown that important areas of the brain that control this behavior include the dorsal, medial, and lateral areas of the forebrain, the telencephalon, the preoptic area, and many hypothalamic areas. And we've shown that key signaling pathways include FGF signaling, histamine, endothelin, nitric oxide, and 5-HT. And what's really interesting is that many of these differentially expressed genes we found by RNA-seq aren't obvious candidates which fit within these known pathways. And so our next challenge is to try and understand where they're expressed in the brain and what their function is in controlling aggression. So thank you very much for your attention. Most of this work was done by Florian Reichmann, who was a postdoc in my lab. He's now set up his own lab in Graz. We had help from all my other lab members, and I'll be happy to take any questions if you have them.
Thank you, Will. Thanks a lot. And so the floor is open for questions. Here on the Q&A, I don't see any, but maybe I'm gonna start with one, or whether you have anyone that Yulin may make a sign as well for Oded. So in my case, so I'm not an expert on the zebrafish, but there is something I don't understand. So you showed us that like there are a group of fish that can all together swim in the same direction, led by an eventual leader. And then all of a sudden they see a mirror, and then their aggressivity behavior starts. So why is it that at a moment they are social animals, and then at another moment that they immediately switch this type of behaviors towards aggression? What are the programs that lead to that?
I think there's two things to consider here. So firstly, within the shoal, there will be microaggression events all the time, which determine the distance between different animals in the shoal and also their position in the shoal. So classically, we think that a dominant fish will be found at the leading edge of the shoal. They tend to be bolder. They're more likely to get food, for example, whereas subordinate animals tend to be pushed towards the back of the shoal. And so in that shoal of animals I showed you, they've been together for more than a week, and they've established a stable dominance hierarchy, and we would expect that the more aggressive animal had the best position within the shoal. We think in the mirror test, what you're seeing is a resetting, if you like, of the social hierarchy. So we know in dyadic fights that if you leave them for several days, you'll see a social hierarchy is formed. One animal will become dominant and patrol most of the area in the tank, and the other animal doesn't swim much and will be trapped in one of the corners. And so you remember that our mirror test is very short. We only looked for 5 minutes. And what we think we're seeing is the initiation of aggression, which is being used to set up a social hierarchy. In a dyadic fight, over time, you see a reduction of aggression, and then you see your one subordinate animals. We know in the mirror test that you see aggression levels for a longer period, more than 30 minutes, and that's because the fish lacks proper feedback from the other animal. The other animal cannot become subordinate, for example. And so we're just probably looking at initiation of aggression, which would be the very first stage towards setting up that large group of fish.
I see. Thank you.
Good.
So I have a question, Will. So usually I would think the aggression, the physical traits are essential for aggression because you need to be physically strong in order to be able to dominate the other animals. But in this particular case, since you're using the mirror test, right, you don't seem to have any selective pressure on getting the animals being bigger. But nevertheless, you get the more aggressive animals being the bigger and stronger animals. And so I wonder how you interpret this result. Do you think are the same genes that control the aggression and the physical traits are the same, or they are closely linked? Or there is still some pressures in the selection process that are gonna as you select the more aggressive animals, it's also selected the bigger animals.
Both are possible, and I can't distinguish between the two at the moment. So clearly there could be pleiotropic genes which affect coloration, size, and aggression. They're linked together. The other thing to remember is the way we designed our experiment. We grew 20 animals and then chose the 3 most aggressive. Maybe I didn't make that terribly clear when I explained our breeding program, I'm sorry. So once again, we probably selected for the dominant animals within that group. So over time, they're getting larger.
I see. Okay, there is some pressure. Okay.
It's going to be fun trying to disentangle it, I think. Whether we really can, I don't know. But
okay. Yeah. Okay, I got it. Interesting. Yeah.
So maybe here a comment for both of you, because in the end also we'll pull out this complex hypothalamic network nevertheless. So do you think, both of you, from your two different perspectives, that we can think about a conserved network as well as a genetic network for aggressive behaviors? Do you think that there could be something across species that would be like really maintained?
Would you like to comment, Dayu, or should I comment first?
I can give it a try. I mean, I think, yeah, I mean, the hypothalamus is highly conserved across all vertebrate species. So there is a VMATRIO in the zebrafish, and I will bet my money on they're doing the exact same thing. They're doing their function for aggression. And in terms of the genes, I think certain genes, they are moderating the functions of those cells, such as ion channels and homoreceptors and neuromodulatory receptors. I think they, neuropeptide receptors, they could be also conserved across the species. So, but there is certainly certain species differences, and depending on the environment and what kind of aggression and when it needs to be expressed, or whether it's regulated by, let's say, the astrocycles, or whether it's moderated by the day and night. And I think all these features are gonna have the species, both the species specific and the conserved features. Actually, there was some, I think there is the series, I forgot, Lauren O'Connell, I think, has the and Hoffman, I think they had this about the social network, and they examined a bunch of genes, and which in the social network, and it appears to be extremely conserved across species, with the conclusion that there is a lot of similarities in the modulatory mechanisms across those social network.
I absolutely agree. It was a really good answer. One of the big challenges we have is knowing how to identify different areas of the brain. So the zebrafish brain looks very different to a mouse, a rat, or indeed a human. So broadly speaking, the hypothalamus is definitely involved, but it can be really difficult to know what one small nucleus within the hypothalamus is homologous to in a rat or a mouse, and that's a real challenge. And it also comes down at some level to how you define homology, whether that's the neurotransmitter expressed, the connections it has, or indeed function. So you could argue the hypothalamus is conserved because it has the same function, and yet we have these brain areas which look different and have different names, and it's a huge challenge to understand what my periventricular nucleus in the zebrafish is and how that's similar to the rat or mouse, and that's difficult for us.
Yeah, I totally agree. I mean, it is kind of the same topic or idea we are facing in other nuclei in between mouse and fish, namely the lateral habenula or the ventral habenula in the fish, that are implicated apparently with like in aversive encoding, but yet, despite the fact that they are homologous, probably conserved, they are nevertheless accounting for different behavior, different pressure, different environmental changes, and so on. So that is reflective to any other behavioral change. So I think we are up to time and good to go. So unless there are more pressing questions, I would like to thank Will, Dayu, and Oded for their contribution in this mini symposium. Thanks a lot for the technical help and everything, and thanks for everyone attending this event. So without further ado, thank you everyone. Enjoy the rest of the day, depending on the time shift you have.
Speakers, AJ Pedagog will be our first speaker, and it will be Ignacio Obeso Martin, and then myself. So it would be a great introduction to introduce myself. AJ graduated from the University of South Alabama with a bachelor's in electrical engineering and biology, two very useful things. He wrote a senior thesis on automatic dysfunction in Parkinson's disease under the guidance of Dr. Oliver Darvin. AJ is currently working in the lab of Dr. Aaron Milsting at Rutgers. And actually, I should say, the one thing that links everyone in this session is Parkinson's disease and the clinical aspect. So, AJ, thank you.
Awesome. I just want to confirm again if everyone can see my screen. Yes or no?
Yep.
Okay, great. Awesome. So yeah, thanks for having me. Hope everyone's doing well, doing safe. I'm Ajay, and I'm going to talk to you guys about autonomic dysfunction in Parkinson's disease using machine learning. If I can, okay. Cool. So the autonomic nervous system, it has these two divisions between the sympathetic and the parasympathetic nervous system. Sympathetic has this fight or flight sort of associated with that sort of fight or flight response, whereas the parasympathetic is associated with this rest and digest response. And both of these divisions are regulated by the central autonomic network, which is this highly recurrent hierarchical structure in the central nervous system. And so within Parkinson's, a pathologist known as Heiko Brock actually found that the spread of degeneration in PD progresses from the peripheral all the way to the central via this central autonomic network. And so in stage 1, he found that degeneration first occurs in the gut and the enteric nervous system, and then there you will see corresponding symptoms of like constipation, etc. Then when Parkinson's graduates to the autonomic nervous system, you find symptoms such as orthostatic hypertension and syncope and sexual dysfunction, etc. Finally, when we get to what he calls stage 3, we get these cardinal motor symptoms that are the classic symptoms of like motor symptoms that are like slowness, rigidity of movement, etc. And then finally, as the spread of the disease worsens, it then infiltrates the higher regions of the central nervous system, like the cortex, where you start to see things like psychiatric dysfunction. And so what would be good for us is if we can catch Parkinsonian dysfunction before it gets to these cardinal motor symptoms, because by that time it's already reached significant parts of the central nervous system. It'd be great if we can catch it while it's still within the peripheral nervous system. And so one way of doing that is by looking at heart rate and heart rate variability. And so there's this prevailing notion that the autonomic control exerts itself on cardiac rhythm by increasing the time scale between heartbeats via the parasympathetic nervous system and decreasing the time scale between heartbeats using the sympathetic nervous system. And so there's this idea of antagonistic control and the linear relationship between the two divisions of the ANS. And we can measure this using by looking at RR intervals, which is the time between these R wave landmarks in the electrocardiogram wave. And I should note this is a very easily accessible thing to measure in the clinic. So now we can make two hypotheses. First, we want to know like what features of autonomic dysfunction do we actually care about with respect to Parkinsonian dysfunction? Is it like sexual dysfunction? Is it cardiovascular symptoms, etc.? Also, how does our understanding of how does this shape our understanding of autonomic control? Is there really a linear antagonistic relationship between sympathetic and parasympathetic nervous systems? And so in our study, we had 45 patients with PD who had these cardinal motor symptoms. We had 17 control subjects. Our only inclusion criteria was that they had Parkinson's, and the only exclusion criteria was that they didn't have any other sort of cardiovascular disease. And we allowed them to take whatever medication they were already prescribed. So how do we catch the autonomic nervous system in the act of regulating heart rate? The way we do that is through the tilt table experiment. What this does is it causes a barrel reflex response, because when we tilt patients to 70 degrees after some period of time in this supine position, blood flows down to their lower extremities, and the autonomic nervous system needs to tell the heart to start performing some sort of compensatory action to get blood back up to the upper extremities. And so during our tilt table experiment, we have 4 15-minute periods. In the first period, we ask something called a SCOPA questionnaire, where we ask about symptoms of general symptoms of autonomic dysfunction that occur over a very long time scale, and these are across 7 different organ systems. We then let them to kind of continue to adjust in this rather uncomfortable tilt table during the pre-tilt period. We want to minimize any anxiety because we know that emotion can modulate autonomic control. And then we have a long period of time after we tilt them. The first period of time is, the 2 periods of time are meant for patients who have orthostatic hypertension events, either very quickly after being tilted or later after being tilted. And orthostatic hypertension is the event that occurs when the autonomic nervous system cannot effectively compensate and regulate and respond to the change in blood flow after being tilted. So as I said, there's 7 different organ systems, and we sum each of the responses to each question for each organ system. We also measure blood pressure, heart rate. We collect some summary statistics, and then we classify with machine learning. So again, the scope of questionnaire, it has each question is relates to some symptom, and each response is a measure of severity from 1 to 4. And we sum across each organ system. So there are lots of different summary statistics for heart rate variability, and the first most basic ones are so-called linear metrics, and the most basic ones for these are just statistics about the distribution itself of the RR interval that we record between each heartbeat. And so there's mean, mode, a kind of central tendency metrics, and then like how wide the distribution is, which we use dispersion metrics for, like standard deviation, variance, etc. But also the electrocardiogram waveform is a periodic time series, which means we can extract oscillatory components from it. And past research has shown, as part of this antagonistic linear relationship between sympathetic and parasympathetic systems, that lower frequency oscillatory components are associated with sympathetic control, whereas high frequency is associated with parasympathetic. However, it should make sense that linear metrics don't always work, because the central network controlling heart rate is this highly recurrent structure. And so we know that these highly recurrent structures are reminiscent of complex systems and have this like chaotic nonlinear behavior. And so we need some other metrics to characterize that. There's a lot of work that's previously shown that this sort of architecture in the nervous system can be captured as a complex system. So the first is called approximate entropy. This basically measures like irregularity over time. So let's consider these 2 different distributions here, A and B, that are sequential. B is this highly regular and highly predictable series of 1 and 2, whereas A is this totally random sequence of numbers between 1 and 2. If you look at the distributions, both of them are basically the same. They have the same mean and standard deviation, but obviously one is highly irregular compared to the other. And so approximate entropy is this nonlinear metric that we can use to kind of approximate how irregular or chaotic a time series really is. Another metric we use is these self-similarity metrics that measure the fractal dimension over different time scales. And so you can see this figure, as we zoom in, we see the same motif pattern of activity over time. And so here we can see whether or not Parkinson's changes different motifs or patterns of activity across different time scales. And so just a very brief introduction to machine learning. Basically, we have a model that makes some predictions, and we try to reconcile those predictions with some observed data. So what we do is we slowly adjust over each training cycle our predictions to such that they match the observations, hopefully by the end of training. And as such, our parameters will change over the course of training. The algorithm we'll be using is called the tree bagger, and this is a quick demonstration of how it works. So what it does is it tries to divide feature space into regions that are associated with each output label. And it does this by finding regions with the highest purity of observations for a given class. So here it found that systolic blood pressure greater than 125 millimeters of mercury is associated with control patients, subjects, and then it also found that Parkinsonian patients weigh greater than 145 pounds. And it continues to iteratively scan each feature until it is able to exhaustively divide all of feature space for every single observation into a region that has a very high purity. And so then for new examples, we can generalize if a new example falls within one region of feature space, it perhaps contains that, it belongs to that class. Another algorithm we're using is principal components analysis. So we're going to be using it for classification, which is a bit different, but it has a lower computational cost. So there's a bit of incentive to that because it's a bit less expensive. So we want to validate our tree bagger because we're not sure whether the features that we extract from it are inferences that are made due to an inductive bias of the algorithm versus some underlying phenomena of the data. And so the way we do that is we just take the top features and the worst features, we train those features on several different, with several different suite of other algorithms, and we see if there's an appreciable difference between algorithms trained on the best or worst features. And there are some performance metrics that we have for machine learning algorithms. Sensitivity is basically the true, the rate at which it can classify PD patients, and specificity is the rate at which we can classify control patients. So what we found was that all the algorithms did, fairly well, but the tree bagger performed the best, and it was a lot easier for the algorithms to identify control subjects versus PD subjects. So this is likely due to the fact that I had to use some sort of generative oversampling model to balance the two classes. And so the control distribution, because it was less, it came from a more regularized generative distribution. And so it was probably a lot more difficult to recognize versus PD. We found that nonlinear features were the most important versus linear features. So this calls into question the prevailing notion that there's this antagonistic relationship between the two divisions of the nervous system. And yeah, we found that blood pressure and linear metrics were not as effective. Again, we found that the best features outperform the worst features, which indicates that the results are likely not due to an inductive bias of our tree bagger algorithm. And we found that removing these other less predictive features actually improves predictive performance. Perhaps this is due to cursive dimensionality. Perhaps this is due to the fact that nonlinear features are truly more informative of the system that we're measuring. We also found that if we do principal components analysis and we do basically a classification along a threshold from the minimum value of the distribution for each component to the maximum value, we found that we actually could find a decision boundary that separated the two classes pretty well. So this kind of calls into question, if we're using these complex machine learning algorithms for classification in a clinical context, maybe we should kind of rely on less computationally costly algorithms and more on something like PCA. Maybe PCA should be used in classification more contexts. And finally, we found that PCA, the features that we extracted from it, agreed with what we found in machine learning, and that nonlinear features are indicative of Parkinsonian dysfunction. And so I'll turn it now immediately to questions for the sake of time if anybody has any, but I'm happy to discuss more if not.
Thanks so much, AJ. Actually, I have a question here is, the argument for PCA is fine, except PCA loses the features of interest. So with your tree, you keep on there's still, you still have tangibility that these features mean heart rate or they mean whatever. While with the PCA, you've now converted into each PC component is a composite of others. So, you then have to reverse engineer it. So, there is a give and take with that. And maybe the follow-up to that is, what about overfitting?
Right.
With this, that's always going to be a question we get. So.
Yeah, that's overfitting is always a limitation for machine learning, and we try to overcome that by doing this sort of oversampling with the control subjects. But I mean, that's actually the reason why we can't publish this, is because we didn't have enough subjects. And also, you could probably say that, hey, these are not cross-validated. So, I mean, you're likely to be overfit. Our argument is that these are pretty low dimension classifications. It's one dimension actually. So you can see that examples are coming from a pretty contiguous distribution.
And just because we have a question from Kelvin Jones, how many features did you simultaneously evaluate?
I think it was something like 20 something features, because for each set of features of linear, nonlinear, et cetera, we had those features calculated for each segment of the tilt table experiment. So supine, pre-tilt, immediate, and post-tilt. So we divided them along those metrics. So the number of features we had multiplied by 4, and we found that, so here, for example, PCA found that entropy before tilting was more important, whereas we found, again, that entropy before tilting was also important. Yeah.
And unfortunately, due to time, I'm going to have to cut off, but you might be able to respond to the second half of Kelvin's question in the chat. And
Sure.
I'm now going to introduce our next speaker. Our next speaker is Ignacio Obeso. He is a cognitive neuroscience and the leading the cognitive control lab at the in Madrid, the HMCNAC. He did his PhD in UCL, a postdoc in University of Toronto, later in CNRS in Lyon, and he's back in Madrid. So, Ignacio, thank you. Oh, are you still muted? Yep, Ignacio.
Okay, now, yeah. So I will speak about dopamine role in excessive behaviors as a model of in Parkinson's disease of impulse control disorder. And dopamine has a multi-property component, which is involving effortful choices when we have to balance between cost and benefit of a reward. It also plays a role in delayed gratification, and that is the delayed reward and the value that we need to wait for our reward sometimes, and this corresponds to a ventral striatal at the level of functional studies. It also has been involved in response inhibition, so to suppress an ongoing movement, and this has been associated with caudate dopamine transmission. And all these combined cognitive processes are important to control your behavior. If one of them failed, you may produce an excessive behavior, such as the one observed here in the final of the World Cup from Zinedine Zidane, that couldn't control properly his excessive behavior. So it's a fine-grained system that needs to be all healthy to be controlled. At the level of Parkinson's disease, which is the topic of the seminar today, there is a loss of dopaminergic neurons in the substantia nigra that then expands to the nigrostriatal pathway, affecting the posterior putamen, as you can see here. And these patients need medication, which is levodopa, and some of them develop a behavioral form of addiction that turns into an excessive behavior. Some of them develop compulsive shopping or compulsive eating, gambling, or hypersexuality, which is the topic that I will cover today. So it's a complicated issue, and because the medication is so important for patients to move better, but they get up to 14% of patients, they get these non-motor side effects. So we need to really know why they develop it and know the neurobiological foundations of this problem. At the level of neurocognitive changes, we see that they have a delay discounting increase in ICD patients, in impulsive patients while on medication, so it's driven by medication. In other words, they prefer they have a higher urge to earn fast money. They want the quickest, the better, the money. So they can't wait for money. We will see more on this measure later in some of my studies. They have a deficit in learning and stopping abilities. So when they have to control themselves and learn that new behavior, they exhibit problems in stopping. And also there is a paradoxical effect that where they don't report in some studies a problem in response suppression. So they are good when they have to stop a movement, which is kind of odd and unexpected, but there are reasons to believe this is true. And what I will be covering today is that whether these excessive behaviors seen in impulse control disorders, whether are they a problem of uncontrolled actions in the sense of impulsive actions and response inhibition problems, or whether is it a problem of delay gratification? Is it really that they cannot tolerate to wait and see their reward? So we will be speaking about the theories of top-down control that from prefrontal cortex monitor control the limbic reward system, or is it a problem that the valuation system is upside down and it drives excessively the behavior? So these two systems interact nicely in a healthy brain, but may be the driving force and the explanation for excessive behaviors. So one of the studies that we are currently running in Madrid, we recruited patients with hypersexuality in Parkinson's disease. And basically we asked them to perform a stop signal task, but adding erotic stimuli before the go or the stop responses. So basically they were seeing an erotic image followed by a go trial, and sometimes they were seeing a non-erotic image followed by a go trial and a stop signal that had varying delays between go and stop. So we would obtain a response speed of inhibition, termed SSRT. And what we see at the behavior level is that the patients with impulse control disorders, while unmedicated, they see a slowness in response suppression, but when they are off medication, when they are not with the origin of the problem, which is medication, they are faster to inhibit. In other words, they stop faster after the erotic stimuli only. So this is quite specific, and it's quite a parallelism of the daily life that they experience this problem. At the neural level, we did this in the fMRI context, and what we see is that when these patients were on medication and they could successfully stop, they recruit a large network of limbic regions, including encoded BTA and cortical pre-SMA and cingulate, to successfully stop. So they need to recruit a large network to successfully stop while unmedicated. And this circuit, using a dynamic causal modeling, we see that there is a differentiation local effect in the pre-SMA between the group that has the impulsivity compared to the group that doesn't have the impulsivity. So that's one result that is quite novel and striking as a cortical area to possibly target. And also a second effect is the descending pathway from pre-SMA to CODI that is also reducing the impulsivity patients compared to the non-impulsive patients. So this is a novel result that is still in writing and submission process, but will soon come out. So, and then moving on to a second topic that I wanted to introduce you is whether these excessive behaviors is a matter of a delayed gratification problem. Is this a problem that they have a weighting mechanism that is driving impulsive and excessive behavior in ICD? So are they capable of are they capable of waiting, or are they not really capable of waiting? So this was the question that we addressed with this study. And so we again selected hypersexual patients, and we asked them to perform this task where they saw a fuzzy image of a naked woman. And they were asked to decide whether they wanted to wait to see the image in full here, waiting for a shorter time if they rejected, and they would see the image for a shorter time. But if they accept to wait, waiting times up to 9 seconds, they would then get their reward for a longer time. So it kind of mimics, again, the real life scenario to wait or not to get what they really like. And we were focused on the choice moment for the brain activity as well. But at the behavioral level, we already get some interesting results, and we go against the literature. And because the patients with ICD unmedicated are capable of waiting to get the reward. They have the lowest discounting rate. As you can see in the control group have a positive value, and they are not bothered to wait for these images, while these people are really motivated in waiting. We also computed a subjective value score of each trial, which is the weight of benefits divided by cost. And what we see here is also a larger subjective value in these patients while unmedicated. So this makes sense as well. They don't maybe have as much of a cost in the waiting times, and the reward is so high that the subjective value is as well high for this group of people. And what we see in the brain is the correlation with the medial prefrontal cortex as a group. But when we divide the two parameters of the subjective value, the delay, which means the cost of getting the reward, in the ICD group has a positive relation. So the more delay, the longer and the more cost to get the reward. This region activates more significantly in this group, whereas for the non-ICD and the controls goes the other way around, suggesting that these people will really want to wait, and they engage this region to put themselves in the waiting situation, whereas the others groups are not bothered in waiting for this. And we also find a ventral striatum activity in relation to subjective value, but in this case, the nice result goes in the rating, so in the value of the stimuli. And what they patients tell us is that what they are seeing is what they like to see. The more they like to see the image, the less this ventral striatal activity is expressed, while in the other groups, in the PD and the controls, is the opposite. The more they like it, the more this region activates. So this kind of upside down system in the ICD group may explain why they don't really value the rewarding sexual rewards, and maybe that's why they engage in these hypersexuality types of behavior. So as conclusions, what I've been telling you about, it's the role of dopamine in excessive behaviors, and specifically with hypersexual patients. We see that excessive behavior in ICD is mediated by reactive control deficits that were mainly driven by medication in the ICD group, and that may produce the uncontrolled actions. This was associated to connectivity changes from pre-SMA and striatum that were lost in the ICD group or were reduced, and that also may be a neurobiological explanation of the deficits seen in ICD patients. And regarding the delay discounting study, ICD with hypersexuality seem to be willing to wait for longer when they get an erotic reward. So this is opposed to other studies that were showing monetary rewards. But the problem with these other studies is that the money perhaps is not as appealing or arousal for these patients. And when you put them with the correct rewarding stimuli, then they are motivated to wait. They have a reason to wait, right? And finally, the results of a frontal striatal change in medial prefrontal cortex and ventral striatum that is actually upside down or reversed compared to the non-impulsive patients. And then this also may account for the excessive desire that these patients experience. So a top-down mechanism that may be impaired in these patients, implicating mainly in our studies the pre-SMA, will explain the action impulsivity that we see in the results, and this turns into an excessive approach behavior. And also this result can be combined with a evaluation system, a change in the reward limbic circuitry, where we see changes in ventral striatum and medial prefrontal cortex that may increase the evaluation system, the evaluation of rewards without consideration of the consequences. So these systems combined together may be an explanation of the excessive behavior seen in these patients. However, these studies are separate, but still are at least in the same cohort of a non-motor problem, which is hypersexuality. Then other studies may follow to explain other forms of excessive behaviors. So these studies, I would like to thank the people that helped in Spain. Also, they were done in collaboration in France, as well with the funding agents that without them, it won't be possible. Thank you for watching.
Thanks so much, Ignacio. Since I've never actually dealt with this section of PD, so prior to the disease, I accept the argument that it's on drug is where it's think, but is there a predictor prior to disease? Like, would these people have had some propensity for this kind of issue of inhibition? Like, can you predict?
We have some hints like demographic or such as being a male or younger, or tendency to addictive behavior such as smoking or drinking will put you under higher risk. But there are no objective tools to get this predisposal for ICD. So we just rely on what the patients tell to us. We don't have other way to know if this will happen. There are some eye tracking studies. One recent one that by Masoud Hussein in Oxford that has looked at this eye tracking guided by rewards, and they seem to differ between ICD and non-ICD patients. So that's promising. Yeah.
Thanks so much. And move on to the next speaker, who I'm delighted to introduce the next speaker. I've known the next speaker for 43 years, since it's me. So now, and I hope everyone can see that. So I'm going to talk about neurophysiological causes of dual tasking in people with Parkinson's, and specifically in freezing a gait, people with Parkinson's with and without freezing a gait. This work was accepted last week in experimental brain research, so hopefully you'll see it in the next few weeks. And there's a joint work with myself and Conor Fearon, who's a neurologist on it. It's a study I quite like. So we're going to investigate the impact of walking on a simple behavioral task in people with Parkinson's with and without freezing a gait. So I'm going to use FOG plus for with freezing a gait and FOG minus for without. And then I have some controls as well. Then we're going to use mobile brain imaging, EEG while walking, to investigate how walking impacts the motor processing, decision making, and the automatic processing. And it seems weird because actually I should say automatic processing, then motor and decision, but I'm going to present to you how we kind of came to this. So first thing, Parkinson's disease. Well, thankfully the other two speakers have done a nice summary for me, but Parkinson's disease is neurodegenerative, characterized by loss of dopaminergic signals in the basal ganglia. The symptoms that I'm most interested in are tremor, bradykinesis, postural disturbance, and specifically freezing a gait. There are many non-motor symptoms, which impulse control, as Ignacio just talked about. And the treatments really treat the symptoms. And so it's dopamine replacement or deep brain stimulation. But freezing a gait, it's an intermittent gait disturbance where someone's feet get frozen to the ground. Most apparent in late stage Parkinson's, and it affects 60% of patients with Parkinson's at different stages. But the reason why it's very upsetting about the freezing, but it causes falls. With or without Parkinson's, falls are the leading cause of reduction in quality of life and life expectancy in people over 65. The issue with Parkinson's and freezing a gait specifically is it's very poorly understood. There's no effective treatment. It's difficult to understand and it's heterogeneous. So, our experiment. It's a very simple experiment. It's just a target. So you see a plus plus, then you see an X, and you press a button. And they're random. So 80% of the time there's a standard, which is the plus. 20% of the time there's a cross, which is target, and that's when the person responds. There was a corridor that was part of another study, but they're just focusing on the thing in the middle. So we had three groups. People with Parkinson's with freezing a gait, people with Parkinson's without freezing a gait, and young controls. They did two conditions of that task. One where they're sitting and one when they're stepping in place. Now, I'm going to later on just use the word walking, but stepping in place for safety reasons. They're just stepping left, right, left, right. And while they're doing that, we're recording EEG. I put in one of the earlier EEG studies I did with the Fox Lab, looking at EEG while walking. And we convert the data to the current source density. So EEG while walking, mobile EEG. That's it. So here we have our little EEG cap. And in this example here, which is from my time in Albert Einstein College of Medicine, they're walking on a treadmill. So why EEG? EEG is great. It's system level snapshot. It can be used to look at attention deployment. It has temporal resolution, is what we care about. It's lightweight. So in theory, you could walk around the world with an EEG cap on, and people do for clinical reasons. And also you could do fancy online looping. So to our results. So first, let's talk about the sitting. So on the left side of the graph, we have our freezing group, FOG plus. And so the individual, that's individuals, and the dark is the group. And so what we have is the freezing group, and the middle group is the not freezing Parkinson group, have similar response times. And the younger control group are faster. Nothing really surprising there, but it's nice that there's consistency within the people with Parkinson's. what we see when they're stepping in place is something quite different. All of the people with Parkinson's with freezing a gait get slower. And maybe that's no surprise. You're going, oh, of course, you're doing 2 tasks. But in previous study with Brenda Malcolm and colleagues in Albert Einstein College, we showed actually people sometimes will get faster at simple tasks while walking. And this was the case for people without freezing a gait and controls. So they actually got faster at the task while doing 2 things because it's a simple enough task to do. So this is nice behavioral results because you have this interaction between the 2 groups. But the question is, what's happening that's causing this difference? So firstly, our EEG results, we have a standard evoked response, and I'm looking over the central parietal region. So that's our standard evoked response, which is the plus, and then the target, which is the one they're clicking to, is the red. And what I have here is the dotted line is when they responded. So that's a response time. Now, one thing we can take away here is that both the standard and target are relatively similar prior to around 200 milliseconds. And if we subtract them from each other, what we see here is what's classically called the P3 response, or the central parietal positivity. So that's for the controls. Now, for the people with freezing a gait, exact same thing. What we see is a very clean P3 decision-making response, because this is our indirect measure of decision-making, or direct. Then with the people without freezing a gait, with people with Parkinson's, we see something very similar. And then we have our young controls. Now, looking just at the subtraction waves being, we don't care was the standard versus target, we care about the difference between them. What we see is CPPs, or P3s, for the freezing groups, and the control group a bit larger, but quite similar. Below that, we have their topo plots of the distribution of activity across the scalp. And what we see is a central parietal positivity, which is commonly seen as P3. But we also note that in the people with freezing a gait, there's something happening in the frontal area. So we investigated that. Now, that frontal area is motor, not frontal as in decision-making, but motor response. And we've found for each individual something called a lateralized readiness potential, where you subtract the motor potential for the left and right hand from each other to see inhibition versus excitation. Now, for a simple single button task, there shouldn't be a very large motor response. But for the freezing of gait group, there was. So that's very interesting. So this is still just sitting, but their response time was the same as the non-freezing Parkinson group, but they still are recruiting more area. So what about stepping in place? So here we are. We have our standard and target, and then we have our subtraction wave, and we have a P3. Similarly, for the non-freezers, very nice, and for the control group. And I was delighted to see this as well, because doing moving EEG, there's a concern about noise, but I didn't do anything overly fancy with the data. I just kept classical analysis because I wanted to show you don't have to do anything too fancy to get nice looking data. And what we see here is similar looking P3s from 400 to 600 milliseconds and later. And the topos, not as clean, but still you see a nice central positivity. So that's very reassuring. But that's me looking at the relation. And now we look at motor once again, and we see once again, the freezing and gait group have a very large negative going motor potential between about 350 to when they have their button press, while the other 2 groups don't really need to recruit motor areas. So it's a larger and earlier onset of the lateralized readiness potential than when sitting. So therefore, they're recruiting more of their motor area. And therefore, we would argue that they have to recruit more resources to do something that's very simple. And that's why we believe there's a delay in the response, because they're already at their limit. And then you're asking them to step as well. But there was also another interesting finding for us, is that you see for the FOG minus group, so the middle graph, and the control group, the right-hand side, their sitting and stepping curves, their subtracting curves, track each other quite nicely. But for the freezing group, there is a separation early on. And we posit that the difference in the early evoked response suggests the loss or reallocation of early processes when dual tasking. And that would explain why there's a knock-on effect in response. It's not that we're seeing a difference in the decision-making, we're seeing a difference in their automatic processing and their motor. So, in conclusion, the behavioral data showed that the people with Parkinson's with freezing at gait had slower response times when stepping compared with sitting, while it was the opposite for the people with Parkinson's without freezing at gait and the controls. They were faster when stepping. So therefore, they were getting benefit from doing activity, while it's the opposite for the freezers. Then using EEG, we're able to drill down and say, oh, it looks like it's a motor issue because the people with freezing at gait have to recruit more motor areas, but there's also an issue with response conflict, which touches a bit on Ignacio's as well. But we did not see a difference in the decision-making. So the impact of locomotion on generating the N2 and lateralized readiness indirectly implies that these functions compete with the locomotor resources. So this is part of many ongoing studies that the highlighted people are involved with that, but all these other people have been involved in many of the other mobile brain imaging studies I've conducted over the years. And every year at the Albert Einstein College of Medicine, we would do an event for children with autism. And that's why I had access to a Cookie Monster outfit. I don't own one normally. So any questions? And, unless I ask myself. So, yes. No, and that is a sad reality. So Dave's question is, did you collect any data about the consistency of stepping? No, we were using a Wii board, which we thought was going to be really innovative and exciting. It was tedious and annoying and didn't work the way we wanted to. So a limitation of this study, and it's stated in the paper as well, is that there are two major limitations in my mind. One, I'd love to have a lot more data. And two, we didn't have their stepping. If we had their stepping, then we could look back in time at that as well. No, I didn't go down to the source space either in this case. I decided just to stay in scalp. But yes, that was hard to find. Why I used current source density as opposed to the classic ERP was, yes, we probably couldn't look at the IC components very cleanly with the data we have, but the CSD was hopefully able to separate out between the different possible sources. And that's the one thing is, one of the papers I put in from clinical neurophysiology that we had was looking at this. And if we didn't use CSD, it would look like the people with freezing at gait had a smaller P3 or CPP because there was mixing of signals. So it is actually very important to think about what the sources are and where they're coming from. But we weren't able to look at that as cleanly as we would have loved to. Thanks, Lewis. No, classically, I would argue that it probably is. I wouldn't say in that one. I would believe it's motor because the onset of lateralized readiness potential and how it was formed. I really believe it's a motor issue that we're seeing there. I would love to see more complicated tasks. I'd love to see them out in the nature, but I know John Fox and Filippo De Sanctis have now moved on to use MS, but to run even to run a study like this in people with Parkinson's, it took us a long time to publish because we were slow, but there are not many people looking at mobile brain imaging in disease groups. So, no. So, because I don't want to stress the next section, ciao Filippo. I would like to thank my three speakers, Ignacio and AJ. And also I'd like to thank Sylvia, who you don't see, but is in the background, who has been making sure that things run smoothly. And I'd like to thank everyone for coming. And so thanks so much for your interest.
You. Thanks, everyone.
Thank you, John. Thank you.
Out there. And so, hello, everybody. Welcome to the session on sensory and motor systems and physiology and behavior. My name is Kirti Krishnan. I'm an assistant professor at the University of Tennessee in Knoxville. My research interests are mouse models for neurodevelopmental disorders with a focus on psych today, along with Sylvia, who's our backend host. I'm really looking forward to this exciting session on motor behaviors. Please ask questions in the Q&A and post chats to stimulate relevant discussions. We have to abide by the time restrictions. Our speakers are aware of this, so I'll hold up the signs at the right times. And our first speaker today is Mr. Mike Mikitz. He is a doctoral candidate at the University of Tennessee, Knoxville, from my lab, actually. His talk title is Application of Human Kinematic Parameters for Measuring Motion Performance in Mice. Welcome, Mike.
All right, yeah, so let me share my screen and we'll get started. Oh, 1 second. Can everybody see? All right. Yeah, so hello and thank you to everyone who came out to see my talk. I'm very excited to be presenting some of my unpublished work on the application of human kinematic parameters for measuring motion performance in mice. So a common assay that's used to study motor learning in rodents is called the single pellet grasping assay. In the single pellet grasping assay, mice are food restricted to increase motivation to reach for a high reward food pellet. You can see here in the reach panel, the mouse or the rat will begin by lifting its paw, advancing forward, pronating its digits, and then grasping the pellet. And once the pellet is grasped, the paw then returns the pellet back to the mouth. What's interesting is this motion seems to be relatively conserved between humans during a reaching and grasping task. So if you look at the hand here shown in a non-visual reaching task for the human, it starts, lifts, advances, pronates, and then grasps the object, very similar in a fashion to the mouse. These findings, along with more, have made it a good model for studying the mechanisms of motor learning as well as execution, as research supports that there is a developmental homology between the two. Traditionally, motor efficiency in rodents can be quantified by looking at simple ballistic parameters such as velocity, angular velocity, or even just the percent of success during retrieval. Qualitatively, this can be characterized using different subjective scoring systems such as the Eskol-Wachman movement notation. An issue that occurs here is that subjective analysis of motor performance can introduce bias as well as variability in what one observer quantifies as an efficient or an inefficient motion. A common objective measurement for quantifying motor failure is needed in order for us to improve our understanding of motor learning and understand how a more efficient motion is developed over time with experience. So in humans, a common used parameter to measure motor learning and efficiency is called peaks, or also known as submovements or intermittency. So here, peaks are defined as any change in acceleration or deceleration in a trajectory are such that acceleration is equal to 0. When peaks are increased in a movement, this is typically indicative of a less efficient motion. Another parameter that is used in quantifying human parameters is something called mean squared jerk, or jerk for short, which measures the smoothness of motion. And this is taken by looking at the area under the curve of the integral of the acceleration. A study by Rand et al. Showed that Parkinson patients have increased movement duration as well as increase in intermittency, or what we would quantify as peaks, during a reaching and grasping task. So you can see they take longer, as well as if we look at acceleration, compared to human, there's a lot more fluctuation in this, suggesting that they have a less efficient motion. While these parameters have been well established in humans and primate models, we wondered if this could also be true for mice. We're interested in modeling these parameters in skilled reaching due to the wide variety of different genetic and molecular tools that can allow us to probe the underlying circuitry behind the behavior. Particularly, our lab is interested in understanding skilled reaching and grasping in a female mouse model of Rett syndrome. So in collaboration with Dr. Eric Wade, who was a previous engineer at the UTK Engineering School, we sought to answer if these known kinematic parameters in humans, such as peaks and MSJ, could also be applied to mice. We hypothesized if these patterns of motor output during a reaching and grasping task are relatively homologous between humans and mice, then we would expect that known parameters such as peaks and MSJ would be able to predict motor efficiency in mice. So we asked the questions, are there kinematic differences in successful reaching versus failed reaching in the single pellet grasping assay, specifically looking at changes in mean squared jerk and peaks over time, as well as, is there any correlation between the number of peaks or the average MSJ in terms of % success rate? If peaks or MSJ are predictive measures, then these should decrease over time with training and could build a predictive model for motor efficiency in mice. To answer these questions, we used the single pellet grasping assay with 2-month-old female wild type mice and restricted them to about 10% of their body weight and maintained this. And then we began motor training. So during the motor training, we allowed mice to reach for as many pellets as they would like by reaching through a slit in their chamber and recorded the number of successes and attempts during this process. While simultaneously recording the behavior using high frame per speed video recording. These videos were then inputted into a video analysis software called Tracker, which would then allow us to manually track the paw over time and then later on derive some of these kinematics that we're interested in. The data that I'm going to show you is an N of 428, meaning that I tracked the reaches for these mice for 428 individual reaches, and this took a while. So here I'm showing you a live demo of what we see in the Tracker motion analysis software. And this is a really cool video, right? Where you see the cyan dot where I'm tracking the paw manually in this software, and it's bringing it back to its mouth. In the right panel, you can see that there's this change in X over time where we're getting the X and Y position relative to time. And once we have this data, we can then derive kinematics such as MSJ and peaks using a custom MATLAB script that Dr. Eric Wade at Cal Poly and I designed together. Here I am showing you the average number of peaks for successful reaches in black and failed reaches in red. And all of the data is normalized to the first day of success. So you're looking at fold differences between success and fail. And what we see is that there's a significant fold increase in failed reaches in terms of peaks, and that this is consistent across training. However, as we continue with training around day 4, there is no significant change in peaks. So this suggests to us that it's possible that the failed reaches are becoming more mechanically similar to the successful reaches, meaning that efficiency overall is increasing. To further this analysis, we did a linear regression analysis to look at the % fail and the number of failed peaks during each of those reaches. So these are average per mouse on each day. And what you can see is that there was a positive correlation in that as % failure increases, the number of peaks also increases. Keep in mind that as peaks increase, this is usually indicative of a less efficient motion and suggests that mice that have higher peaks in reaching usually aren't efficient at the task. And so overall, we showed that this was correlated and that there seems to be a more efficient strategy that is developing over time. So when we look at the other parameter, MSJ, we kind of saw the opposite effect. There actually seemed to be no significant change in MSJ, whether it was a successful reach shown in black or a failed reach shown in red, also normalized to the first day of success. And if we do a linear regression analysis of this, we see that there's virtually no correlation at all with MSJ during this single pellet grasping assay. So in summary, I hope to have shown you that peaks seem to be a predictive measure for motor efficiency in mice, as we've seen that peaks and failed reaching decrease significantly with training, indicating that a more efficient reaching strategy may become developed over time as they become more similar, mechanically similar to the successful reaches. In the context of MSJ, there didn't seem to be any predictive power in terms of motor efficiency. This was surprising since peaks in MSJ are typically dependent on one another. We hypothesized that this could be due to the fact that previous studies looking at MSJ as a quantitative measure have a consistent start point. However, we do this in free-moving mice, therefore the starting position of the paw is different for each reach. My future directions right now are determining if more simplistic measurements such as peak velocity or the time to peak velocity change with training, because this has been previously reported in developmental studies in humans as well as some studies in mice. So that will be important for us to know if those are also changing consistently as well. And so I told you before that we were manually tracking these mice. And at the time when I started this, Deep Lab Cut really hadn't kicked off. However, thanks to Deep Lab Cut and Dr. Mackenzie Mathis and the Deep Lab Cut team, we're now using this approach to quantify motor behavior using automated techniques through DLC. So here you can just see a preview glimpse of where we're going with this, which is to use the automatic tracking to generate pose estimation data. With that pose estimation data, we can then input that data into our own MATLAB script and then compare that to our manual calculations, which will give us a good quality of assurance for our data. And so with that, I would like to thank my lab, my mentor, Dr. Kirti Krishnan, Billy, a postdoc in the lab, Dr. Dana Leo, a graduate student in the lab. They've all been implemental in this project, as well as 2 undergrads who helped kickstart this project. As well as our very successful collaboration with Dr. Eric Wade. It's been a journey. So, and with that, I'll open the floor for questions.
Thank you, Mike. Perhaps you may want to get off the share screen there. There we go. Okay. Yeah. So any questions for Mike? Normally I would be the one asking questions, but then that would become a lab meeting. So let's, if other panelists or anybody else have questions about this, please feel free to ask. Not much at this point. So perhaps we can come back to Mike after this part and move on to our, oh, we may have one. There we go. All right, we do have one. The question is, nice talk. What are the future directions?
Yeah, so right now we're just working on a short manuscript for submission at the Journal of Neurorehabilitation and Repair. Ultimately, what we'd like to do is look at this in a mouse model of Rett syndrome. So patients with Rett syndrome have mild to severe sensory and motor impairments. So we can use our mouse model to see if pre-symptomatic versus symptomatic changes in motor efficiency are present in our mouse model. And this could give any type of indication in terms of designing future rehabilitation strategies, as well as probing underlying circuitry later on.
That is correct. Mike's got his work cut out for him. So, all right. Okay, so we'll move on. Thank you again, Mike. That was lovely.
Thank you.
And we move on to our next speaker, who is dressed as Hermione today. So I should say that first, since it's Halloween and NMC was promoting Halloween costumes. Unfortunately, I didn't send that out too much in my Twitter accounts. So our speaker is Dr. Erica Barhorst-Cates. She is a postdoctoral research fellow at Moss Rehabilitation Research Institute. Her talk title is Action Imitation via Trajectory-Based or Posture-Based Planning. Welcome, Erica.
Thank you. Can you see my screen? Okay, great. Thanks for that intro. Thanks everyone for tuning in. So I'll be presenting some data from a recent preprint that we've submitted, and I'll share a link to that preprint at the end if you're interested in more information. So I'd like to start out with a little exercise where I'd like you to watch and memorize this action without moving. So keep your hands on your lap, but I would like you to pay attention to this movement as if you were looking in a mirror. Great, now I'd like you to try to perform that action, please. Okay, now, if you would, please think about how you might have done that. So what aspects of the movement were you paying attention to? What types of things were you imitating when you did that? And there are a couple of ways that you might have done this. The first of which is by perhaps matching the body posture. So you could have paid attention to the limb positions, how the wrist was positioned in relation to the elbow, for example. And there's a long line of evidence that this is one way that we might imitate action. So we know that movements are planned with the intent to achieve desired postures, for example. And we also know that a lot of what we know about imitation actually comes from people with limb apraxia, which is a disorder that arises after strokes primarily to the parietal lobe. And these patients have a difficulty with performing imitation and tool use gestures despite having kind of the physiological ability to move. And the way that this has been typically studied is by using static posture. So asking patients, for example, to match the limb positions of static postures, and we see a deficit in imitation ability. However, there is an alternative method of imitation that has recently been proposed by our lab that is performed by matching end effector trajectories. So rather than thinking about kind of static positional information, in this type of imitation, you might actually just think about how the end effector is moving and think about the movement of that in more of a body independent way that specifies the motion path of the hand. And the question we're trying to address here is, can people imitate using both mechanisms? Well, we hypothesize that these are useful in different situations and for different actions. For example, when learning ballet, it might be more beneficial to focus on the position of the body parts and how they're kind of configured in relation to each other. Whereas imitating an action such as a baseball throw, it might be more useful to think about the end effector trajectory. We also know that these are, or we hypothesize that these are subserved by different brain regions, and there's some evidence to support this. For example, matching postures has been associated with the inferior parietal lobe, whereas matching trajectories has been associated with the dorsal premotor cortex. However, these different imitation mechanisms have not been studied in the same paradigm. So it's actually not clear whether people can imitate using these different methods. And again, that's the question we are trying to address with this study. So if these mechanisms are different, then switching between them should be costly. So we used a sequential multitasking paradigm where participants performed a primary task that was related to either posture matching or trajectory imitation, followed by an intervening task that was meant to be congruent or incongruent with the primary task. So in the congruent case, both the primary task and the intervening task are kind of in the same processing route, or they're both related to posture matching. And so we would expect that performance on the primary task would not be impaired. There would be no switch cost here. Versus when the primary task is related to posture matching and then an intervening task is related to trajectory imitation, followed by a task querying performance on that primary task, we might expect to see a switch cost here because people have kind of switched out of processing posture matching information to trajectory and then back. And so this competition for resources might result in impaired performance. So what we did was, for our primary task, a movement imitation task where we manipulated the instructions to attend to different aspects of the movement. We had 25 neurotypical young adults who were right-handed and had full range of motion of the arms. And we attached kinematic motion trackers to the right wrist, elbow, and both shoulders to track their movements. So in our posture matching primary task condition, we instructed participants to pay attention to the position of the body parts, for example, how the elbow, shoulder, and wrist are positioned. And we gave them these instructional videos to just kind of really emphasize that we wanted them to think about and attend to that postural information. In our trajectory primary task condition, we had participants pay attention to the path that the hand takes through space. And we emphasized that using the following instruction videos. And you may have noticed that these motions are actually exactly the same. The only thing that differed between them was what we emphasized in the instructions. Our intervening tasks, which again were designed to be either congruent or incongruent with the primary task instructions, were performed after participants viewed an initial video and before they performed their imitation of that movement. So in our posture matching condition, people moved their arm to match positions presented in pictures on the screen. In trajectory, they extended their arm and traced shapes in the air. And then we included a neutral condition where they simply tapped their hand, which served as a control or baseline condition. And just to kind of clarify our procedure here, first we had participants watch and remember a movement such as this. You might notice that there's no longer those red emphasis lines. And this was combined with instructions to either imitate the postures or the trajectories, followed by one of the 3 intervening tasks, followed by instructions to imitate that primary task. So this is the dependent measure that we used, was their accuracy at performing their primary task imitation. We had 20 trials in each condition, and then participants all repeated the same procedure again after about a week's time with the other set of instructions. Our dependent variable was Procrustes distance, which is a measure of movement dissimilarity between a participant and a referent. And the higher the number, the less similar the movement. And so we modified this algorithm to allow for calculation of multidimensional space. And we used the position data from the wrist, elbow, and shoulders that we tracked to do this. So I'll get right into our results. So first, I'll show you some data of our results with participants' performance compared to the model. And I'll just say right away that there was no effect of the primary task instruction. So whether they were asked to pay attention to body postures or to trajectories, that did not affect performance. But we did see some effects of the intervening task. So for example, imitation after the neutral intervening task was less impaired than imitation after the posture matching intervening task. Because we also asked participants to match the time of the movement of the model, we also analyzed participants' movement time accuracy compared to the model. So on the y-axis here, I have movement time error. So the higher the number, the less similar the movement time was to the model. And as you can see, we did find that imitation after the neutral task, again, was better than after the other 2 intervening tasks. And interestingly, we found also that movement accuracy after the trajectory intervening task was also greater than after posture matching, suggesting that trajectory imitation may be slightly easier. So what I've shown so far is that imitation after a neutral intervening task is the most spatially and temporally accurate. Imitation timing accuracy is lowest after posture matching, but there was no effect of the primary task instruction manipulation. And again, these results were all regarding accuracy compared to the model. And so one thing that we wondered is, well, maybe imitating a model is difficult and kind of introduces error beyond what we have manipulated. So there's the need to kind of translate what the model is doing and mapping it to your own body, kind of performing that mirror transformation. So this could have introduced error that was unrelated to our manipulations. So it could suggest that our accuracy measure may have been insensitive. And so what we did in a post hoc analysis was compared each person's performance after one of our interference conditions compared to their own performance after the neutral. So this allowed us to remove the potential error that's inherent to kind of imitating another person, or in other words, to assess the effect of the intervening task kind of above and beyond the working memory demands for that individual. And so here I'll show you these results. On the y-axis, I again have Procrustes distance consistency. So a higher number indicates a greater dissimilarity between a person's performance in either the trajectory or posture matching task compared to the neutral task. And what we found was that when the primary task was to imitate trajectories and participants switched to performing a posture matching intervening task, you can see that there was a switch cost. So the performance consistency was reduced compared to when the primary task was to imitate trajectories and the intervening task was also to imitate trajectories. You can see a greater amount of error here when participants switched. But we did not observe the same effect for posture matching. Okay, so for our post hoc analyses, we found that consistency within subjects revealed a switch cost, but only when imitating trajectories and then switching to posture matching. We also found some evidence that there is this greater consistency for congruent trajectory matching tasks, which suggests again that the trajectory matching might be an easier method of imitation. And then I'll mention that we also found similar results for another analysis of performance between subjects. So we looked at how people performed, how similarly they performed with relation to each other. And if you'd like to see those results, you can see our preprint here. And then I'll just summarize to say that I present the first evidence that people can imitate using both posture matching and trajectory methods, and that switching between them is costly. We're currently exploring the neuroanatomical underpinnings of this with the stroke patient study. So with that, I'd like to acknowledge my collaborators, especially my postdoc advisors, Laurel Bucksbaum and Aaron Wong, and our research assistant, Mitchell Isaacs, our funding sources, and all of you for listening. So I'd like to open it up for questions.
Thank you. That was a lovely talk, Erica. We have a few questions in the question and answer session, so I'm going to ask you about those. The first question was, hi, Erica, great talk. Wonder if your model of imitation considers other variables for the people who imitate such movements. These variables of that model of imitation might be state of mind, state of health, different morphology, age, gender, etc.
Yeah, that's really interesting. I think if I understand the question, it's kind of like, what are the different factors that could be making you either more similar to the model or more different to the model? For example, maybe gender makes a difference, or like if they have more perceived flexibility or something like that. Yeah, we haven't really talked about that per se, but we have talked about looking into individual differences in kind of like, what is the default imitation method that people will use? Like, for example, do some people attend more to body postures and some to trajectories? And maybe how that changes across learning. So in ballet, for example, do you start by matching postures and then moving to trajectories, or vice versa? But yeah, I think that's a really great question, and we would love to continue thinking about that.
Right. Moving on to another question. This is about the theoretical background. Are you sure that using two coding schemes leads to switching costs and not to parallel processing advantages?
No, I'm not sure about that. I think we kind of, I guess we look at it as we had to start somewhere, and we hypothesize that these are kind of dependent on different neuroanatomical regions. And so theoretically, we would expect there to be greater cognitive resources to kind of switch between them. But yeah, it's possible that it could be kind of parallel processing. You know, maybe people are paying attention to both things rather than like one or the other. But I think with our stroke study, we might kind of start to address that. For example, if a person has a lesion that has broken one of those mechanisms and we're able to get them to imitate accurately using the other, it might suggest, I guess it still doesn't quite answer whether one of them's not being used if the other is being used, but yeah, I'll have to think about that.
Another question, perhaps this will be our last question, and we'll come back to others if we have time at the end. It is, do you see baseline preferences in which tracking strategy neurologically intact participants prefer? So which tracking strategy would they prefer?
Yeah, I think, again, like I mentioned earlier, that is something that we're interested in pursuing, is what do most people prefer one versus the other, or are there individual differences? I guess we would say from our movement time data that perhaps people might prefer trajectory matching because it seems to result in slightly better performance, at least in terms of timing. But those results were related to the intervening task, not to the primary task of paying attention to the end effector trajectory of the model. So yeah, I think the answer is we don't really know, but we're very interested in finding out.
Great, thank you. You're welcome to answer the rest of the other questions in there in the chat when you get a chance. Thank you, Erica, for a lovely talk. And so now we move on to our last speaker for the session today, Dr. David Huberdeau. He is currently a postdoctoral scientist at Yale and a Kavli Fellow. He will be presenting his doctoral work from the Krakauer Lab. His title is Dissecting Learning and Generalization of a Continuous Motor Skill Using a Customized Video Game. Welcome, David.
Yeah, thank you. And thank you for the introduction. So yeah, today I'll be talking about how people learn continuous movement skills. And so what is a continuous movement skill? And I think a good example is Serena Williams serving. And so you can see in these videos that Serena Williams has this the serve that's basically a continuous movement that's quite stereotyped. And if it weren't for the fact that there's different people in the background of these videos, you might think that they're the same instance. And now Serena Williams wasn't born knowing how to serve. This is something that she learned over a lot of practice and a lot of experience. And so I think that that's kind of the essence of what I'm talking about with continuous movement skills, is that they're continuous and took a long time to learn and quite stereotyped. And so how might people learn these types of behaviors, these continuous movement skills? And one theory is what's called the action chunking theory. And the idea here is, let's say that there's, for example, 4 movements, A, B, C, and D, that for a particular behavior are required to be executed sort of in that order. So early in learning, you might select each of these movements individually. But with a lot of practice, the theory goes that this sequence of movements coalesces into what's called a chunk, where the action selection occurs at the level of the chunk instead of at the level of the individual movements themselves. And so I think a good example of this is, say, playing piano, where you have to press individual keys in a discrete way, but with a lot of practice, that becomes somewhat automatic. And so the question is, do continuous movements, like a tennis serve, kind of fall under this model? Or is this model applicable to these continuous movement skills? And to answer this question, I think we need to actually move away from tasks that require a behavior that is fundamentally the selection of discrete actions. In other words, we need a new kind of task that actually requires continuous movements. And so to do this, we partnered with a video game company that was started at Hopkins. And they built us a game on an Apple iPad that requires participants to steer this little cartoon character along a winding and narrow path by tilting the tablet. And so I have a few examples of what that looks like. And so here, this is a participant on day 1. And you can see that they're prone to falling off. You know, they don't necessarily have this skill down yet. But if we fast forward to their 5th day of training, we'll see that they're a little bit better at this continuous movement skill. So that gives you an idea, on one hand, of what learning looks like, but also just how kind of new and sort of fun this task is. And from this task, the kind of data that we get and the things that we can look at are the trajectory of the little character in the game. And so you see examples of this for one participant on the left, where we can plot the actual trajectory of the character in the game on the track. And we can look at this across days of training. And a couple of things that you notice are that the distance that the participants got on each trial on average increases across training. And another thing is that the variability of their movements seems to reduce, so they become more stereotyped. And then other data that we get from this game are the tablet tilt directions. And so this is just the angular direction that they were tilting the tablet. And you can see, concordant with the car paths, that the duration that they stay on the track seems to increase, as well as the variability of their movements seems to decrease with training. And so we'll be looking at the distance traveled and the movement kinematics in terms of movement variability. And linking this back to the chunking theory, the kind of key manipulation that we're going to do in order to test that theory is introduce what we call a probe track, which is just the mirror image of the track that they were training on. And the critical test or assessment is to look at how behavior changes or doesn't when they encounter this probe track. And so the way this worked is that participants trained exclusively on this training track for up to 10 days. And then they were introduced this probe track for a continuous sequence of trials only on their sort of last day of training or once during their training. And so the critical thing we're looking at is to see how their performance is affected in these probes. And just to be clear, under the chunking model, we would expect that their performance would degrade because the probe track requires an entirely different sequence of actions. And so we're going to look at what happens during these probes. And if we look at the distance traveled measure, there's a couple of things to point out. So the first is that across days, performance improves. And so you can see that by looking at the window that I've labeled here as the pre-probe, which is trials that were done on the training track just before the probe was introduced. And so you can see that across days, their performance on average is increasing. Another thing to point out is that during the probes, their performance transiently drops, but then recovers to about 90% of where they were before that probe was introduced. And that's true of days 3 and onward. And so you might say, well, look, the behavior is a little bit worse. But I think the critical point is to point out that the behavior isn't at baseline. So the probe doesn't induce this catastrophic decrement in their performance according to the distance traveled. And so I would argue that this actually violates the chunking theory because the probe track requires an entirely different set of actions. And we can look at the behavior in terms of the kinematics. And so what we've done here is we've taken just a segment of the track, the first turn, and looked at only those trajectories that made it through at least that first turn. And what you can see is that the trajectories, their variability seems to come down across learning, so from days 1 to 10, and then seems to be also low during the probe. So that top panel of individual trajectories is just a single participant as an example, but we can get a more concrete measure of the variability by doing PCA, principal component analysis, across those trajectories, and then get a measure of the variability over the first couple of principal components, and then plot that across training and during the probes. And so on the bottom left, you can see that variability as it changes over practice, and then during the probes. And critically, the variability didn't really change during the probes. And so there wasn't really evidence of the variability somehow regressing to some previous state of learning. And so I think this is quite strong evidence against the chunking model. However, we have this potential slight discrepancy between our performance measure that's based on the distance traveled and the performance measure based on these kinematics. And we'd like to explore this and see what's going on here. So I think the one key thing to keep in mind is that participants can fail on any trial, even if it's the training track, the one that they've practiced for 10 days. And that's true, of course, also of the probe, but we wanted to check if the way that failures are occurring is different or the same between training and the probe. And so to do this, we introduced a new kind of analysis where we look at the action that individuals are taking across different states of the trajectory. So we defined a state space that included different locations on the track and actually also different car heading directions. And we defined what we call as a policy, what we call a policy, which is defined as the average tilt direction at each state among trajectories that were successful. And from this policy, we can define what I call a policy deviation, which is the absolute angular distance between an individual trajectory's action at a given state and the policy at that state. And the way that we can use this to assess how failures are occurring is to look at the policy deviation of all trajectories that fell off, aligned to the point at which they fell off. And so in this plot on the far right, the darker curve is showing the policy deviation of failure trajectories that are aligned at 0 to the point where they fell off, and then successes that are within that neighborhood as a comparison. And you can see that the way that trajectories fail is that they have this sort of momentary lapse right at the end before they fall off. And so they seem to be quite consistent with the successes up until just before they fall off when they have this lapse. And so we can ask if people are failing in the same or different ways during the probe as they were during the training or the pre-probe. And it turns out that we really have no evidence of the trajectories failing in different ways. In other words, they fail the same way, whether they're on the training track or the probe track. However, what was different is the proportion of trajectories for which a failure occurred, for which a lapse occurred. And so during the probes, their proportion of having one of these momentary lapses just happened to increase. And so this potentially could happen because they're somehow distracted. Something is going on, but what's not going on is that their skill, their movement ability, the ability to make these continuous movements seems to be consistent during the probes. And so as an alternative to the chunking theory, we are calling this a sort of learning a flexible policy, where a policy is defined as a map between the state, say of the individual in their world, to actions that would be required at that state. And so linking this back to Serena Williams serving, she goes through these different states of her body and the world around her. And at each state, there's basically an action that is sort of best for that particular contingency. And so, we're suggesting that this data is actually inconsistent with the chunking model. And instead, it seems consistent with individuals learning some flexible control policy that's partly defined, within a state space. And it seems like lapses are responsible for failures, and that probe tracks induce more lapses. They don't induce a decline in skill. And so I would like to thank my PhD advisors, John Krakauer and Adrian Haith from the BLAM lab, as well as the Kata project, composed, at least initially of Omar Ahmad, Kat McNally, and Pramit Roy, who built the video game for us. And of course, I'd like to thank my current funders through Yale, the Kavli Foundation, Center for Research Computing, and the Stabilization Foundation.
Thank you for that lovely talk. I have a few questions, but one of the first questions I was thinking about was when you were showing these tracks, what are the age of your participants? I have a 7-year-old who does Wii when she does this, and it's fun and great to watch her try to navigate this environment. So I was curious about the age of your participants.
Right, they were young adults, basically college students, graduate students. Yeah.
Who need money.
Right, right. Yeah, exactly. Who are susceptible to my persuasion.
Right, great. So we have one question in the Q&A. So, hi David, great talk. Did people train on a single track or on multiple ones? Do you think the people would learn differently? Sorry, go ahead, yeah.
Sorry, real quick. The first question, the answer to the first question, they trained on only one track. So it was the same track all throughout training until they received that probe, which was just the mirror image.
Do you think that people would learn differently if you were to offer them a set of tracks?
Yeah, good question. I think that's a fantastic question. I think potentially, and I would say that potentially they would learn differently in the sense of perhaps doing even better during the probes, maybe because they have had this experience of seeing multiple different tracks. And so when they get this new thing, it doesn't sort of throw them off as much.
Right, yes. Another question is, do you think you would have found the results inconsistent with chunking if you had reordered, maybe leave the first turn out, the training turns, rather than inverting them during the probe?
Wait, sorry, there's something I didn't understand. Is this
Carly Sombrick?
Okay. Oh, reordered them. I see. Right. Yeah, no, that's a good point. Yeah, that's a really good point. If you had just reordered the turns, but they were otherwise identical turns, then the chunking model, I think, would expect you to have actually better performance, potentially, in as much as if you can imagine a task that, say, has 2 chunks or something. So maybe you would say, like, actions ABC is one chunk, and the next 3 is another chunk, but then you flip them. Under some instances of the chunking model, I think you would expect that putting them in different orders might retain the sort of improvement within a kind of sub-chunk. So yeah, it could get complicated, and it might not be entirely inconsistent. Yeah.
I'm sorry, you do have more questions in here, but we're almost going to get kicked out of here in a minute or so. So I just want to thank everybody for a lovely talk, wonderful discussion, and I just wanted to finish by saying we see some of these things in our mice model, actually. So I was really excited to see your the way human performs and mice perform may have some similarities here. So renewed faith in model systems. Great, thank you again. That was a wonderful session, and I hope you all enjoyed the Neuromatch Academy series, and hope to see you all again soon.
And the organizational principles that govern the specific cell layering or the calcium dynamics in the mural glial cells across development, all the way to the consequences of retinal waves and motion detection in the superior colliculus. All 3 talks in this session are interactive. So the talk format is 5 to 6 minutes of overview and then 10 minutes of questions. So I encourage all of you to keep the discussion alive and ask the speakers as many questions as possible. The first talk is by Xinshin Ge. She is currently a postdoc in Massimo Scanziani's lab at UCSF. though she would be presenting her work from her grad school work from Michael Crer's lab at Yale. And she's going to be talking about retinal waves and how they prime visual motion detection by simulating future optical flow. The stage is yours, Shinshin.
Thank you. Okay, can you see my slides? Okay, thanks for the introduction. Hello, everyone. My name is Xinxin. As Lena mentioned, I'll be presenting my PhD work from the Career Lab at Yale. And before I start, I would like to acknowledge the great team that enables this work, especially Kathy, who's heavily involved in a lot of experiments I'll be talking about. I would also like to thank my PhD advisor, Mike, for his great mentorship and support. So, So the main question that we are interested in is, during development, how does the visual system prepares itself for the future incoming stimuli without knowing what it is going to see? And to investigate this question, we use the mouse visual system as our model. So here's just a brief background. Neonatal mice don't open their eyes until P13. And actually, before P9 or P10, their visual system does not receive any visual inputs. Yet from previous studies, we know that there is a lot of spontaneous retinal activity, which is wave-like, that exists during this window without the input of sensory inputs. And here I'm just showing you examples of the readout of the retinal activity by imaging the spontaneous activity in the retinal ganglion cell axons in the superior colliculus. And so for today, what I'm going to focus on is to try to understand the main function, or the function of the spatial temporal features of the retinal waves in visual system development. So what we found is that actually the retinal waves are not random. In fact, during a transient window, the majority of the waves propagate from the temporal to nasal direction in the retina, corresponding to forward to backward in visual space. The wave direction flow pattern actually is also not homogeneous on the superior colliculus, but rather exhibiting this specific pattern. Interestingly, what we found is that the wave optic flow pattern matches very well with the optic flow the wave flow pattern matches very well with optic flow pattern from the animal's self-forward motion after eye opening, which is probably the most likely optic flow inputs after animal visual system matures. And in contrast, the wave flow does not correspond very well to optic flow from rotatory motion. So we were very interested in the origin as well as the function of the directional bias retinal waves that mimics the future optic flow. So using pharmacological approach, we found that GABA-A is required for wave directionality. So here I'm showing you example movies before and after the gabazine injection, which is a GABA-A blocker. And you can see that the wave directionality is disrupted with the gabazine. In addition, we also found that the starburst submarine cells, which provide asymmetric GABAergic inhibition, which further confer direction selectivity in the adult retinal circuits, are also important for wave directionality. So in a transgenic animal where the asymmetric inhibition from starburst submarine cells along the temporal nasal axis is disrupted, we found the disrupted wave directionality. So these results suggest that the directional retinal waves are mediated by asymmetric GABAergic inhibition from the starburst submarine cells. And so last, I would like to talk about our efforts to understand the function of the directionality of the waves. And to answer this question, we inject gabazine chronically from P7 to P12 to disrupt the wave directionality during this time window. We then measure the visual response properties of individual cells in the superior colliculus right after eye opening. And here you can see that individual cells respond pretty well to the drifting grading stimulus in both the saline injected control or the gabazine injected group. However, we found that there was a significant reduction of the selectivity or the tuning strength of the cells preferring temporal direction in the gabazine injected group compared to the saline. And this is not the case for other directions. And here is just a summary showing you that for all the cells that prefer different directions, only the temporal direction preferring cells were significantly disrupted in the gabazine injected group. So overall, I've showed you that during development, we found a transient window where the wave directionality exhibits this temporal to nasal bias. And this is mediated by asymmetric inhibition from the starburst amacrine cells. Interestingly, we found that directional bias waves resemble this future optic flow from animals' forward self-motion. And the wave directionality also sets up or helps the development of motion detection in the downstream visual areas, such as the superior colliculus. And with all these results, we think our results suggest a very smart and efficient way of the sensory system development, which is that the developing sensory system primes itself for the most likely visual inputs by generating spontaneous activities that mimic the important features of future sensory inputs. And with that, I would like to thank everyone for your attention and also be happy to take any questions.
Thank you, Shinshin. That was really great. Let's see if we have any questions. While we wait for questions from the audience, I actually have a question regarding how basically you have the direction selective retinal ganglion cells as well that provide input to both the DLGN and the superior colliculus. So how is their activity, like their wave featured activities, different compared to the non-direction selective retinal ganglion cells, and also whether the changes in the wave directions due to one cell type versus the other has any influence on this motion detection later on in life?
Yeah, that's a very good question. So I don't have the direct results here, but I can tell you that we also imaged other retinal ganglion cells that so like specific types of retinal ganglion cells that does not prefer this temporal to nasal direction. And it turns out that those retinal ganglion cells also exhibit this temporal to nasal wave directionality. And also in our experiments, when we express the GCAM, we label all the retinal ganglion cells, including the non-direction selective ones. So we think that the majority of the retinal ganglion cells are participating in the spontaneous retinal waves in the same fashion.
Very nice. We have a question from Daniel Gardner, and he asks, do you have any sense that waves at other developmental times are priming for other stimulus classes?
Yeah, this is very interesting. So the previous studies, for example, has already showed that the amount of retinal waves or the frequency of the retinal waves, specifically during this P0 to P9 window where the waves are mediated by acetylcholine transmission, are important for example, the refinement of retinotopy in the superior colliculus or the thalamus, as well as the eye-specific segregation. So the input from both eyes to the superior colliculus or the thalamus. And beyond that, I think there hasn't been specific experiments testing like certain features of the retinal waves in terms of the their response, in terms of their function in further development. But because the waves show this very complex patterns and changes a lot during development, we suspect that it may also be important for such as setting up the receptive fields in downstream areas or like the orientation selectivity or other properties.
Yeah. So I know at least in the primary visual cortex, both orientation selectivity and direction selectivity is developmental. So early on, you don't have much direction selectivity, and then it just gets strengthened as the animal matures. And so how does that work in the superior colliculus?
Right, so in the superior colliculus, we found that there was already a lot of direction selective cells right after eye opening. There may be some further refinement of the tuning strengths, and maybe a little bit of the preferred direction or orientation, but we already observed a large proportion of cells that respond to the directional motion or orientations. And I guess in comparison to the visual cortex, we also found a lot of visual cortex. We also found a lot of cells in the visual cortex that are direction selective or orientation selective right after eye opening. But there was, as you mentioned, also a developmental refinement after eye opening.
Right, right. I'm a bit curious about your gabazine experiments. So those were injected in the superior colliculus, right? Not in the retina?
Those are injected in the retina.
Oh, okay, in the retina.
Yeah, yeah.
So it is disrupting the waves in the retina itself.
Yeah, yeah, yeah.
Do you have any, I guess, clues about if you disrupt the local activity within the superior colliculus, how would that, during this developmental time window, how does that affect the optic flow essentially, or the direction flow? Is it purely inherited from the retina, or are there some local circuits that emerge as a result of this?
Right, that's a very good point. Previous studies from the Korea lab have shown that probably up till eye opening, the superior colliculus activity is completely inherited, or like majority of the activity is inherited from retinal ganglion cells. We haven't done anything to specifically disrupt the activity in the superior colliculus, but actually I can imagine that if we mismatch the posterior, the postsynaptic activity versus the presynaptic activity in the superior colliculus, we may create some like different disruption to, for instance, for setting up the receptive fields or retinotopic refinement.
Cool. We actually have 1 more minute left, so I'm going to take the liberty to ask one more question.
Yeah, yeah,
and
sure.
That's about looming stimuli. So you know how there's been a lot of focus on, predatory type of looming stimulation that are encoded in the superior colliculus, and I feel like these waves, retinal waves, could also be a contributing factor in the emergence of such looming stimuli evoked behaviors. So what are your thoughts on that?
Yeah, I think this is certainly a possibility. We haven't specifically examined the responses to the looming stimulus in the superior colliculus, but I would imagine that potentially with this global pattern of retinal waves, it may to some extent help the responses, but we don't know. I think this is purely just my guess right now.
All right. Well, that was very fascinating. Thank you,
Thank
Shin-Shin,
you.
again. Next, we have Pui Shi Lee. She is a PhD student in Dr. Liane Godino's lab, and she at the Technical University of Munich. And she's going to be talking about the formation and maintenance of horizontal cell mosaics in the zebrafish retina. The stage is yours, Pui Shi.
Can you give me 1 second? Can you see my screen, or it's not visible? Okay. So basically, as Lena mentioned, I'm going to talk to you about the formation and maintenance of horizontal cells mosaics in the zebrafish retina. So first of all, maybe most of us here know more or less about the general architecture of zebrafish retina, or in general vertebrate retina. We have the retinal ganglion cells that projects to the brain for visual processing. We have the photoreceptors that detect the light, and we also have interneurons such as horizontal cells, bipolar cells, amacrine cells, and we also have glial cells called Muller glia. So basically, during development, the retina is essentially an apical basal oriented element, which then gradually transform into a structure with characteristic cellular pattern. So basically, at about 30 hours post-fertilization, there are still apical basally oriented elements. And then at about 3 days, you can see the layer structure with all the different layers of cells. And however, if you actually slice through one of these layers, what you can find is the somata of these cells. And many previous studies have shown that these arrangements are non-random. So this is an example of how the zebrafish cone mosaics looks like. The 4 different colors indicate the 4 different cone types that zebrafish has. And you can clearly see that they are sort of non-random. And a lot of studies has been looking at how the layers are formed, but very little is known about how the mosaics are formed during development. So this is what I'm focusing on. So what is a retinal mosaics? It is non-random arrangements of cell bodies of individual neuronal subtypes. And it is first quantitatively analyzed by Wassel and Riemann in 1978. Functionally, it is proposed that it is important to ensure that all parts of the visual field have access to a full set of processing elements, so that you can get parallel processing of visual stimuli from all receptive field. And there's also a theoretical work that showed essentially that photoreceptors mosaics is designed to optimize visual performance. Regular arrangements can also help in developmental wiring of connection between cell types. So mosaics are independent of each other. So each mosaics of different type of neurons are independent to each other, and mosaics and dendritic tiling can be independent of each other. So one is about the arrangement of the somata, and the other is the arrangement of the dendrites. So basically, I'm looking at horizontal cells. So a brief introduction of what horizontal cells is inhibitory GABAergic neurons, and it is shown to be important in contrast enhancement and the generation of antagonistic centers around receptive field. And in zebrafish, horizontal cells can be classified into 4 distinct morphological subtypes with unique connectivity to photoreceptors. So I'm going to focus on just one subtype, the H1 subtype. This is how a typical H1 cells look like on fast view. So this is how the photoreceptor look at the horizontal cells. It has an axons, and the dendritic tips actually terminates in a rosetous flower shape. And it can be labeled genetically in a specific manner. And it is shown to be connected to red, green, and blue cone. So first of all, to ask the question of whether there is a mosaic, there is a H1 mosaics, I basically dissect the retina out of the animals, and I flatten it to make a flat mount. So we know that by 3 days there's layers. So I'm asking whether at 3 days I can see a regular pattern. This is a representative image of how the retina look like, and you can more or less see that there is some sort of arrangement here. And I basically do automated segmentation and then analyze using spatial statistic analysis. There are 3 methods that I use. The first one is nearest neighbor regularity index. It's only concerning about the nearest neighbor. The Voronoi domain regularity index, basically it is assessing how the whole retina space is spaced out by different cells. And that's also a density recovery profile. It is more about looking at the global arrangement. So using all three methods, comparing to random simulations, I basically see that H1 mosaics is established as early as 3 days, because for all of the index that I show, it is higher than, it is always higher than the simulation case. And how does then this regular arrangement arise during development? This is so this is when I turn into in vivo imaging of mosaics formation. So it has never been done before because of technical issue, and yeah, so most of the studies is done more theoretically. And yes, so as I mentioned before, horizontal cells are generated starting at 2 days and forming a layer by 3 days. So I did a time-lapse movie from 46 hours post-fertilization to 72 hours post-fertilization, and this is a representative image. So what you can see here is that there are a lot of cells coming, and basically it follows a developmental wave of how the retina is formed. So basically from preliminary results, I can say that mosaics formation could be divided into at least 3 phases. That's 1 active proliferation, migration, and refinement stage. There are a lot of possible mechanisms as to what could be underlying neuronal mosaics formation, and I'm only going to focus on tangential cell-cell interaction, mediating tangential migration and peripheral translocation. So in the migration phase, if you look at this movie, you can sort of see that maybe I can play this again. So you can see that there are 2 cells that touch each other, and then the cells kind of move away from space into a more empty space. So basically here you can see that cells move away from crowded space, and it kind of sample the space to find where the maximum distance to neighboring cells is. And also in the refinement stage, this is a 2 hours movie of one part of the retina, and basically I do automated segmentation and Bayesian tracking. So what you can see here is the velocity of all the cells at all time point, and you can see that they have quite low velocity at this stage. And when I plotted the distance from the first time point, you can see that cells are still sort of deviated from their initial time point, but it always have this jiggle. So this poses that these cells are basically sampling the space and then find kind of where it wants to be at the end. And this can also be looked at by the membrane labeling. And you can see all the protrusion that just keep coming up and go. So all this information implies that there might be some cell-cell interaction mediating mosaics formation. So as a summary, I told you that H1 mosaics is established as early as 3 days post-fertilization, and I have established an imaging paradigm for in vivo visualization of mosaics formation, and it follows a developmental wave. And it can be divided into at least 3 phases: proliferation, migration, and refinement phase. And H1 cells are sort of actively sensing the space during migration and refinement phase. And yeah, the future direction will be to perform like sort of ablation studies to further test this hypothesis. And for that, I would like to thank you all for your attention. I'm happy to take any questions.
Thank you, Puishi. That was really cool. Especially the fact that you're able to image across development and look at those cell-cell interaction dynamics across all these developmental stages. So while we wait for some questions, I have a question about at what stage do you see these cell-cell interactions? I mean, is it only after the proliferation phase when all the cells have already been born and proliferated, and then you start seeing these cell-cell interactions that push the cells away to their final destinations? Or do they start interacting with each other even while they are migrating and proliferating to find their optimal positions, even before new cells come in, essentially?
Yes, so basically, I didn't show it here, but I have preliminary results that basically show if I look at the division angles of neighboring progenitor, that don't sort of space out from each other. So it means that the progenitor doesn't know that needs to stay away from the other cells. And also, if you look at this movie, you can see that there are a few cells that sort of clump together. So it means that, yeah, basically at this phase, there's no more division at this part of the retina. So yes, the different phase.
Sounds good. We have a question from Yunlu Ju, and he says, do you think contact inhibition, CIL, could be a potential mechanism for their migration?
Yes. So there have been theoretical works that show contact inhibition could lead to a regular pattern. And yeah, one of the problems here is that we doesn't know what kind of cell surface molecules are expressed in this type of cells. So we have no way, and at least at the moment, no way to perform a sort of loss of function study to look at this. But it can definitely be a mechanism.
Related to that question, actually, I was wondering if these cells have, in addition to just contacting each other, do they signal to each other as well in terms of activity and that guides where they would migrate to, or is it purely a structural based interaction?
I haven't looked at the activity of these cells. And yes, it could be a very interesting direction to go. Definitely.
Okay, sounds good. Let's see if we have some more questions. If the audience has questions, please feel free to post them in the chat for Puishi. I actually was wondering also about, you said you haven't done it, but do you have any hypotheses on what kind of cell-cell interactions might take place in terms of whether they're cell-cell adhesion molecules that interact with each other, or is it some other type of signaling mechanism?
So basically, cell-cell adhesion molecule, for example, cadherin could be possible. And also because horizontal cells does form gap junction with each other at a later stage. So that might also be one of the mechanisms where you have connexins that basically bind to each other or repel each other.
And this is different, and these are only cell body tilings, right? This is different from the dendritic, like you mentioned, mechanisms
Yes.
that would probably have other kinds of DSCAM-like molecules or something like that that probably govern those mechanisms.
Yeah.
All right, cool. Well, thank you so much, Puishi, for that very insightful talk. We are going to move to our final presentation by Cyril Eleftheru. I hope I pronounced that right. He is a postdoc in Dr. Butir Sagdullaev's lab at the Berg Neurological Institute at Weill Cornell. And he's going to be talking about clusters of Mueller glia and how they display sporadic radial bursts of activity after eye opening. Take it away, Cyril.
Is my screen shared already or not?
No, not yet.
Okay, where's the button for screen sharing? Ah, here we go. And I can do, okay, so wait a second. I'm gonna do the beginning and then I can do oh, okay, there we go. This one. Okay, is it playing? Can you guys see? Is it the full presentation or is it just
Now it is the full presentation. Yeah.
Okay, it's not my notes or anything.
No, you're good. Good to go.
I'm good. Okay, all right. So as this is the interactive type of presentation, I wanted to do my best to be brief and to present a data set that's somehow somewhat immature. The data still requires a lot of analysis and input, and I'm hoping to get a bit of that from the participants today. So first of all, as you all know, and as Pishri introduced earlier, retinal visual processing arises from 5 classes of neurons: the photodetect receptors that detect light, the bipolar cells which separate the on and off signals, the retinal ganglion cells that digitize visual signals and project to the brain, horizontal cells which were discussed just now, and amacrine cells that perform the lateral processing. And this is the image that we see very often and that we're taught in textbooks. But one thing that's missing is the neuroglia and vascular cells. So there's 3 different vascular plexi within the retina that, especially in the mammalian retina, that give nutrients, oxygen to the retina, and these are brought over by the Mueller glia. So there's astrocytes, which exist in the nerve fiber layer and provide support to retinal ganglion cell axons, and also by Mueller glia, which are the transverse glia which hold the retina together, provide metabolic support throughout the retina, and also very importantly recycle glutamate, which is the main excitatory neurotransmitter of the CNS and also in the retina. Now, in our lab, we're interested in studying the dynamics of these glial and vascular cells, as well as the neurons that perform the neural processing. So we generated this mouse line, which expresses GCAM6F in all types of cells in the retina. And we can image at different layers within the retina and then record the spontaneous and light-driven calcium dynamics. So here you see some retinal ganglion cells responding to a pulse of light, as well as the vascular cells. So here, some endothelial cells and pericytes, which are spontaneously fluorescing during the recording. But what I want to talk about today is the Mueller cells. We found that when we record at low magnification for extended periods of time, so 10 minutes, so I was taking 10-minute recordings of the ganglion cell layer, we can find spontaneous activity of spontaneously fluorescing Mueller glia. And I performed these same recordings in a number of different ages. So at P10 during stage 2 developmental waves, at P15 after eye opening, at P20 in the barely mature visual system, and at P60 in an adult stage where the system is fully mature. And what I found is that the frequency of these spontaneous events really increases significantly after eye opening. So at P10, there's almost none of these, but at P15, 20, and 60, you get a lot more. And the duration of these events seems to be always the same or similar in the same ballpark, same as the distance traveled by these clusters of glia. So with our microscope, we can actually investigate within the layer of the retina. So we can look at the ganglion cell layer at the same time as the inner plexiform layer, the inner nuclear layer, outer plexiform layer. We can really image as the objective is moving very, very fast, and we're imaging these very slow calcium transients. And what we found is that most of the signal is much stronger, deeper within the retina. So at the inner nuclear layer and outer plexiform layer. And we also modulated these events with pharmacological agents. So as you may know, CNQX blocks inotropic glutamate receptors and also the off signals from the photoreceptors to the bipolar cells. And these were able to completely abolish the retinal waves. TBOA, on the other hand, is a blocker of the glutamate recycling transport on the Mueller glia. And when we block those, we flood the retina with glutamate, and we have a significant increase in these waves, which hints at an origin, at the glutamatergic origin for these spontaneous waves. Maybe as the retina is processing too much data, there's too much glutamate, we don't know. But what we were able to really model them by adding webane, which is the potassium ATPase blocker pump, which really keeps the neurons and glia at a specific intracellular concentration of ions. And there we have very, very strong signals. And I'll show you examples in both the ganglion cell layer and in the nuclear layer. These events are a lot more frequent as the retina becomes completely unphysiological, but it's that same kind of radial coordinated form that we observe, and we can really observe them much more frequently in this state, which is really gives us a much closer look at what's happening, and maybe we can try and other types of pharmacological systems to modulate them somehow. Let me accelerate that a little bit for you guys. You can see there's a lot more of these things happen as the retina becomes a lot more unphysiological. Now, as I mentioned in the intro, there's still a lot of work to do. I need to improve my analysis pipeline. I need to look at different pharmacological interactions. I need to, I've got a lot of light responses, experiments that I've performed, and I need to analyze them. We have established a line of retinal degeneration, and I want to look at what glia are doing as the retina deteriorates. And we also want to set up in vivo to look at what the cortical glia are doing and the cortical vascular cells are doing in these different mice lines. Also, we're open to collaborations, and also if you're looking for a job, we've got some money. And also we were interested in people that are passionate about the retina and its function. And I'd like to thank my team before asking questions. Thank you very much for your time and your attention.
Thank you, Cyril. That was great. And as Cyril mentioned, they have job openings, so apply. As we wait for, we have a question from Yunlu, and he says, in blind mice with no visual input from birth, do you still get spontaneous glial calcium waves? If so, how are they different?
So I've established the mouse line, but I've not actually performed any of the experiments. I'm aging the mice. So now they're about a year old, so they're severely degenerated. We've done some optokinetic reflex tests. So we know that they're fairly blind. And I'm going to start doing these experiments soon, but I'm very, very excited to see what's happening inside these retinas.
Yeah. I was wondering if there is any type of synchrony between the muloglial cells that you image from and the other cells in the retina. Are they signaling to each other in a very coordinated fashion? And also whether the synchrony is there early on during development and sort of becomes asynchronous, eventually similar to cortical circuits in a way.
So during development, so what I'm not showing here is that at P10 we have obviously very strong spontaneous waves of activity in the ganglion cell layer. And there is strong coordination between this and the vascular cells, which is another project I'm working on. But the mulloglia appear not to really have any function at this stage. However, one thing that I've observed, and again, I'm not sure exactly how to quantify it. I'm trying different computational approaches, but I'm not sure how to quantify it or how to describe it. But there does seem to be a lot of synchrony between the vascular cells and the glia. So when we see these waves of activity, let's see if I can trigger it as well here. We kind of see a bit of a flash first on the vascular branch. Maybe this is not the best example, as I have somewhere you can really see, activity being started either in neurons, then, that triggers a response in the vascular cells as they, they kind of give blood or in nutrients in vivo. This is obviously taken in ex vivo. And then we get that glial activity. So this is something also that I need to quantify.
Yeah. So in your manipulation experiments where you did the Wabain experiment and you see this increased frequency of the spontaneous activity, I was wondering if in those manipulations you see the changes in the blood vessel dynamics like you just mentioned.
Well, possibly. No, it looks like here there's not much going on with the blood vessels. It feels like these glial events are happening kind of on their own, but the Wabain situation is very unphysiological. We're basically dysregulating everything, which completely dysregulating the balance of ionic potentials within each cell in the retina. So it's very hard to kind of make patterns from that. But yeah, just looking at this particular video, it seems like the vascular cells are not doing much. It's mostly the neurons and the glia. If anything, the vascular cells are becoming a lot more dim. I don't know if you can see it's a lot darker. Whereas all the neurons kind of lit up, I think, as a lot of calcium comes into the cell.
Right. So if you manipulate, or do you plan to, let's say, manipulate these dynamics using, let's say, some mutant mice or the drugs that you have injected, and look at what happens to retinal function later on in life? How do these early events produce adult function? Sort of similar to what Xinshin was describing in terms of how the retinal waves guide later optic flow properties.
Well, one thing that I want to try, so Mueller cells are generally 14 microns across in diameter, but these things are about 50, 60, these kind of rosettes. They involve multiple clusters of glia. And one thing that I really want to try to do, first pharmacologically, and then maybe with different type of light responses, is to decoordinate them. So to basically stop the spread, just generate activity, but that is completely either highly coordinated or but it doesn't spread in that fashion. And I think that's because they're connected by gap junctions. And obviously gap junctions are very important in visual function. So connexin 36, for example, is heavily involved in the on bipolar cell to, in the rod to on bipolar cell pathway. And we got to identify which type of connection it is connected these clusters. And if we can dysregulate them, then how will that actually impact visual function is a question I'm really interested in.
Very cool. Well, thank you. I think that is just on time. So thank you, everyone. I'm going to thank all the three speakers again for their very, very wonderful presentations. It was really very exciting listening to all of you. And thank you to our audience for listening and posting their questions. And have a good day, everybody.
Thanks.
Thank you. Bye.
Bye-bye.
Audience. Just a reminder that these are interactive sessions, which means that the speakers have 5 minutes for their presentations, and then 10 minutes will be reserved for audience questions. So I would highly recommend and request that the audience members ask questions as and when they come up. And please also indicate in your questions whether you'd like to ask them to the panelists directly or whether you'd like me to ask them on your behalf. Thank you. And our first speaker is Samantha Cohen from Cornell University. She's a senior in biomedical engineering, and she is currently an undergraduate researcher in the neural engineering lab with Dr. Maram Bixen. And she is going to be talking about the contributions of skin preparation on static impedance, typically measured prior to administration of electroconvulsive therapy. Take it away, Samantha.
All right, thank you. Here we go. All right. Hello, I'm Samantha. I am a senior at Cornell University. I'm researching in the neural engineering lab at City College. I'd like to thank everyone for coming, for our hosts, and for NeuroMatch for organizing this conference. And I will be speaking to you today about the importance of skin preparation in electroconvulsive therapy. So electroconvulsive therapy, or ECT, has long been a treatment for mental health disorders. The safety and efficacy of ECT has much improved over the last century, making it an effective treatment for depressive disorders when pharmacological therapies have failed. Most patients undergoing ECT treatment see relief in their depression, typically after several ECT sessions and sometimes with the addition of medications. ECT works by applying electrical pulses to the scalp via electrodes that carry current to the brain. The goal is for the current to induce a seizure in the brain, thus charges increase during treatment until a seizure threshold is reached. The brain's adaptation to the seizure is responsible for ECT's antidepressant effects. Although it is not fully understood how ECT improves depression, it is thought to promote the activation of dopamine-responsive neurons. During the administration of ECT, current density is much greater in the area surrounding the electrodes than in the rest of the brain. This is because current is entering the body via the electrodes. During the setup of ECT, the impedance of the electrode is measured with currents much smaller than those of the treatment. Impedance prior to treatment is called static impedance. And both the contact area and the quality of the skin-electrode interface influence static impedance. As such, they are important in the setup of treatment. In the present study, we varied the conditions under which adhesive hydrogel electrodes were prepared for ECT. During the administration of ECT, there are many different ways to prepare the skin for electrode placement. Particular factors of importance include the contact area between the skin and the electrode, the adhesion between the skin and the electrode, and the cleaning solution for the skin. Preparations vary in conductive gels used and types of cleaning solutions. We were interested in how these conditions impact static impedance. After varying electrode preparation under 6 conditions, we then measured the static impedance across electrodes every minute until static impedance stabilized. So here we can see an image from our experiments. And of course, I was not applying ECT to myself. I just want to reiterate that these measurements take place prior to typical treatment. So the impedance under 6 conditions was measured for 3 healthy subjects. With many different conditions, different combinations of prep, we selected 6 that would best reveal the role of each method. So During our experiment, we had saline skin preparation with pre-tack application, and pre-tack is just a common conductive gel used during ECT. And then we had alcohol skin cleaning with pre-tack. And then we had the normal saline skin preparation with pre-tack, but we covered half of the electrode so that only 50% of the contact area was touching the skin. Then we had alcohol skin cleaning only with no conductive gel. And then we prepared our own solution to mimic the role of pre-tack, which was used with a polymer PPPK90. And we had two conditions, one with just PPPK90 and one with KCl, which is just a salt. And those were both prepared with saline skin preparation. So for each subject, there were at least 3 trials for each condition. And this is accounted for in the error bar seen on the graph. 2 commercial ECT machines were used to measure static impedance, the Thymatron and the MECTA. So focusing in on one exemplary subject, we can see that all conditions with an adhesive substance had comparable impedances. We also see the decreasing surface area increases static impedance, and that the concentration of ions had no significant effect on impedance. These results point to the idea that variations in static impedance are largely driven by the quantity, so electrode coverage, and quality from surface adhesion of the skin electrode interface. Not always when we believe there is full coverage is there also full contact. So this is seen in the center image here with poor contact. So optimizing the preparation of the skin will enable the current calculated to best induce a seizure for each patient to reach the brain. Thank you all for listening to my talk, and I welcome any questions.
Yes.
Oh.
Is it? Oh, okay. Yeah, so I guess my first question was about the electrodes you use. So I saw it was like 2 on like the front. So yeah, I guess could you go back to how you did the analysis for those like electrode data?
Sure. Let me share my screen again. Sorry. Okay, sorry. So we recorded the static impedance for each condition for every minute for a minimum of 17 minutes or until the static impedance stabilized, because it would often jump back and forth a little bit. And so it decreases over time, as we can see. And then we took the averages of each condition for each subject, and that is what we have plotted over here.
Okay, yeah, cool.
So I had a question. Is there a minimum static impedance that you'd like, or is there an optimal range that you're looking for?
There's not an optimal range. Every subject will have very different static impedances because there are a lot of factors biologically that influence it as well. From like just the basic properties of your own skin conductivity to like whatever, if you have like makeup on your skin or more oils than another person. So a lot of people will vary within subjects, but you tend to have similar static impedance within individuals across different time points.
But from the perspective of the ECT machine, does it perform better with some subjects than others because of this?
Yeah, so actually when I was the subject, I would tend to go like above the safety measurements of the machine. So it'll not actually apply current if your static impedance is too high, because then it becomes dangerous. So that was about 5,000 ohms, but most people don't have that problem.
And did you try anything? Were you only allowed to try pre-approved protocols for this? Did you try something that something that you thought of might be a good adhesive or might be a good electrode material?
So we selected PVP K90 because it's very common in a lot of products. It's actually common in hair products and some food products.
I see.
So it's readily available, and yeah, that's why we chose that. It's accepted by the FDA.
So we have a question from Maria Stott. If you would like to come on live and ask the question, that would be great, Marius. You can unmute yourself and ask the question whenever ready. That doesn't seem to be happening, so I'll just ask the question for him. I may have missed this, but did you also test if increased impedance had some effect on therapy?
No, so this is just static impedance. It doesn't have anything to do with the actual therapy of ECT. So this is all like the setup process. So we're actually analyzing static impedance in relation to dynamic impedance, which is what you see during treatment, and that will be discussed later, but that's not part of the study.
Got it. And where do you want to take the study next?
So it'd be really interesting to look more between the variability of subjects, like what makes each person so variable. Like, for example, I always had a lot higher impedance than the other two subjects. So I think it'd be really interesting to learn what specifically makes each person have their static impedances that they do. So that way you can improve your models and you'd be able to calculate the best currents that would have the most efficacious treatments for ECT.
Right. If there are no more questions from the audience, it doesn't seem like it. Let's thank Samantha. Thank you for the wonderful talk and for answering our questions. And let's move on to the next speaker. The next speaker is Afreen Jalil from Johns Hopkins University, my alma mater. And she's a research intern with Dr. William Anderson at the Institute for Computational Medicine at Johns Hopkins University. And she is currently a senior in the combined BS-MSc program in biomedical engineering at JHU. And her talk is on the application of image analysis methods for quantifying spatiotemporal neurodynamics. Take it away, Afreen.
Okay, thanks. Okay. So, yeah, as Manu introduced, I study at Johns Hopkins University and studying biomedical engineering. So my research is centered around methods for analyzing data from microelectrode arrays. So I worked specifically on applying methodology from the field of image analysis to recognize patterns in cortical responses recorded by microelectrodes. So microelectrode arrays are used to measure electric potential data from the brain cortical surface. And recent advances in microfabrication have enabled the ability to record from neurons in higher dimensions and resolution with surface and penetrating microelectrode arrays. And you may have heard about Elon Musk's new, relatively new Neuralink company, where they're doing a lot of technological advances in our ability to develop microelectrode arrays. So with these advances in technology, we can develop methods to analyze patterns in the data that is collected. For example, some of the application of this methodology that we specifically are interested in is, for example, if you have electric neuromodulation at a local level to a particular region of the cortex, and you have a certain set of pre-stimulation responses and post-stimulation responses recorded by these local field potential data. This way can develop ways to analyze patterns in these cortical responses. So one of the methods that we can use is histogram analysis. So this is a method used in image analysis where you can basically identify the spatial distribution of values based on the frequency of a particular value in this distribution. And as you can see in these two figures, these were all 4x4 arrays that were then interpolated to have 100 by 100 points. And so the values that are similar have a higher correlation, whereas profiles that are not as similar have a lower correlation value, which is basically showing that this is a way to compare the spatial distribution of potential values. Another method that we looked into is the radial profile, where you can parameterize the distribution of values as a function of angle from a midpoint. So you can have a reference point in the center. You could also place a reference point in a corner. And as a function of this angle theta, you can, for example, take the sum of potential values in that particular direction. And from this, we can identify current sources and sinks from the maxima and minima of these radial profiles. For example, here you can see a maxima or current source at around 90 degrees and current sink around 270 degrees. And you can also detect the direction of wave propagation over time by looking at differences in the radial profile and thereby the differences in the location of current sources and sinks over time. So this was simulated data where we simulated the location and the number of current sources and sinks. So here's one current source, one current sink. Here's two current sources and two current sinks, and likewise the same here. And so you can see that you can identify that profiles with a similar location of current sources and sinks show a similar peak in the location of the radial profile. But if it's different, then you'll have a different location for the peak and minima. But then the question was asked, how do we differentiate between having one current source and one current sink versus two current sources and two current sinks? So you can do that by using a radial profile of the Fourier transform magnitude of the 2D array of data. And so you can see here that you can identify one peak and then a second peak when you have one current source and one current sink, whereas you can identify two consecutive peaks and then another two consecutive peaks if you're doing two current sources and two current sinks. So this is a way to distinguish the number and the location of current sources and sinks. So the application that we used to identify one way in which this methodology could be used is we looked into a data set of pre-stimulation and post-stimulation responses. So this was data from a patient who was undergoing a procedure for epilepsy. So basically they measured the sampled cortical response 100 milliseconds before and after a stimulation pulse, and there were 67 trials. So the way we define state dependence is the probability that pre-stimulation states were correlated given the post-stimulation states were correlated. So basically this means that the state that happened before the stimulation pulse would lead reliably to a given post-stimulation state. So this was a way of identifying event-related responses that were consistent in this data set. So states were considered correlated if the radial profile correlation is greater than 0.9, and they were considered state dependent if this probability here was greater than 0.7. And so we identified certain critical responses that were state dependent. And we also found that from doing a permutation test where we shuffled the order of pre-stimulation states but kept the post-stimulation states constant in order, we found that pre-stimulation context does affect the cortical response. And this is shown here in this histogram. You have a lot less number of states defined as context dependent when the association between pre-stimulation and post-stimulation states is randomized. Whereas in the original data set, there were a greater number of states that were defined as state dependent. And these two figures here show correlation correlation heat maps, correlating pre-stimulation states with each other and post-stimulation states with each other. And you can see that there are groups of responses that are similar to each other, whereas others are very different. So it's not, so basically what this analysis is saying that the response to stimulation is not random, but more likely to be context dependent. And yeah, one of the applicability of what we're doing is, for example, if we want to develop real-time feedback control to optimize neuromodulation therapies. Yeah, that's the end of my presentation. Do you have any questions?
Thank you, Afreen. That was great. That was a lot packed into 5 minutes. So I don't see any questions from the attendees yet, but I have many questions, two of which I'll ask probably. So first thing is, you talked about spatiotemporal responses. How would you be able to use the same techniques in order to examine responses changing in time as opposed to changing across space? Any thoughts on that?
Yeah. Yeah, my family is in the background. Yeah, so in terms of temporal responses, that came back to the idea of, for example, using for example, wavefront propagation. So if we can detect differences in the degrees and we're sampling at a certain frequency, you can, for example, be able to use this to tell both the direction of the wavefront propagation, as well as if you have like time sequence data, you can use that to develop a speed for propagation as well.
Yeah, that makes a lot of sense. Could you go to your cortical data slide, please? If you don't mind. Also, slide? Yes. Hold on, I just wanted to let the attendees know that they can ask questions whenever. We are here to answer questions. That's the point of the interactive talks. So I have a question about this, which was, so you see that the pre-stimulation context affects cortical response. However, is there something you can do to dial it down a little bit further in order to examine how it affects the cortical response, or over what time scales or something like that? What would you be able to do that?
Yeah, I think that would definitely be a great area for future work. to specifically pin down how it is affecting the response. Yeah, our current methodology is it's I guess more like a classification of responses, like grouping responses that are similar. But I think, yeah, that would, given like more data sets, I think that would be a great direction to take this research in the future.
And you talked about neural feedback, and that's very exciting to me personally, because that's part of what I do as well. So how would you use this in order to, can you give me an example of what might be an exciting future work related to neural feedback from this?
Yeah, so yeah, one idea would be, for example, I mean, yeah, there's a lot of movements towards like precision care medicine, right? Like tuning models or feedback towards the specific needs of patients. So I guess one like workflow, I guess you could see in terms of how neurofeedback, how this would integrate into neurofeedback is, for example, if you can measure these like pre-stimulation and post-stimulation responses and correlate that with like associate like a particular pre-stimulation and post-stimulation response with this is like a better outcome, like an optimal effect versus this is like a worse outcome, or like it has side effects. And then like programming a stimulation device that so that it recognizes computationally that pre-stimulation response, and then like the pre-stimulation state, and then initiates a stimulation based off recognizing the pre-stimulation state that is associated with a good response, or avoiding the pre-stimulation state that would generate a worse response or side effects.
That makes a lot of sense. I feel like you should talk more with the other two panelists who are here, who is working on a particular type of stimulation. All right. Seeing no other questions from the attendees, thank you so much, Afreen. That was
I have
great.
a quick question.
Oh,
yeah.
Yes,
sorry.
Please.
Can you talk a little bit about how you go from the microelectrode data to the images?
Yeah. So, oh. Yeah, so here we have the like 4x4 microelectrode array. And so basically we have like the locations for each of these electrodes, and then we use 2D cubic spline interpolation to interpolate those points to a 100 by 100 array of values. And yeah, the rendering of these images was done using like Python libraries for image generation. But basically, like an image is just at its core a 2D array with intensity values. And so yeah, we use interpolation to generate this like 100 by 100 array from the given number of channels.
Thank you.
All right. Thank you so much, Afreen. Perfect timing. Let's move on to our third speaker for the evening. And it is Kaliza Canela. I'm hoping I'm pronouncing your name correctly. Also an intern working with Dr. Maren Bixson at the City College of New York in the Neural Engineering Department. And Kaliza will be talking about the correlation between static and dynamic impedance. So in our first talk, we heard a lot about static impedance. Now we'll be talking about that in comparison with dynamic impedance during ECT. Take it away, Kaliza.
Hi. Let's, wait a minute. There we go. Hey, thank you guys for the opportunity to speak about this. I will be talking specifically about the correlation between static and dynamic impedance during electroconvulsive therapy. So electroconvulsive therapy is decades old. It started in 1932, and it is currently cleared by the FDA as treatment for severe drug-resistant depression. And what it does is it uses a high-intensity current to elicit a motor seizure response in the patient. This high-intensity current is passed through 2 electrodes that are located on the head. I will specifically be speaking about the right unilateral placement and the bilateral placement, which are pictured below. So there are a lot of parameters involved in ECT, but 2 specifically are determined by the clinician. So the electrode placement is determined ahead of time. The pulse width of the current is also determined ahead of time. And the pulse width can be either 0.3 milliseconds, which is referred to as ultra brief, or 1.5 milliseconds, which is referred to as brief. When the machine is turned on, after the electrodes are placed on the patient, a measurement called static impedance is taken, and that is taken at a current of 1 milliamp. During an actual treatment, when the therapeutic current is applied to the patient at either 800 or 900 milliamps, the impedance of the head or the resistance of the head to the current being passed is called dynamic impedance, and that's also measured. The charge is determined by an initial titration session. So because ECT has been around for so long, there have been many publications over the past probably like 90, 80 years or so. And so certain things have been established. Among those are that dynamic impedance is always significantly lower than static impedance. So dynamic impedance ranges in the hundreds ohms range, and static impedance goes from several hundred ohms to sometimes many thousand ohms. There hasn't quite been a relationship between static and dynamic impedance that has been established, but it also bears mentioning that static and dynamic impedance vary widely from one person to the next, and so for all of the data for a single person between sessions, their static and dynamic impedance can also vary. What the other 2 relationships that have been discussed in the literature are the weak reverse relationship between charge and dynamic impedance, and the weak direct relationship between age and charge. So when we were looking at this data, we had a lot of, or when we were looking at ECT, we had a lot of questions about how everything was related, and the relationship that emerged from the raw data from Sackheim et al. 2008 that was done at the Department of Biological Psychiatry at Columbia University was that there was this emerging relationship between static and dynamic impedance. And so what we have in front of us are just the raw data for the, actually not the raw data, but the average data for these 90 patients where we took their average static impedance, average dynamic impedance, and then you can see the full range of what the actual session by session impedances were through the error bars. So as you can see, the static impedance varies widely and the dynamic impedance varies a little bit more narrowly. And what we found was a statistically significant correlation, sometimes weaker, sometimes very strong. And we also looked at this data session by session. So we took each session as an individual point, as opposed to like the average of all the data for each person. And we looked at the relationship again. And again, you could see that there was sometimes a weaker relationship, but sometimes a fairly moderately strong relationship. We also looked at the data and its distribution among different subsets within the entire data group. So what we looked at was we compared RUL to BL, we compared females to males, and we found that RUL tends to have higher impedance both in static and dynamic, and females tend to have a higher impedance when compared to males. Then we also looked at all the other parameters, and we found fairly weak relationships in the things that had been highlighted by some of the past literature. And I don't think there's very much to say about that, so I'm going to move on. We were then curious to see if this is just like a fluke, right? Like, are these 90 patients was there something about the fact that it was single center, like Sackheim did all of the procedures, was there something about that was unique, or could this be seen in other subsets? So we looked at the the data set from Sackheim et al. 2009, which the practitioners were different and the procedures varied slightly. And they were also done at 3 different locations. And you can see that even when the relationship is weak, it's still statistically significant. And I guess that's pretty much it. I guess the only thing I have to say now is that it's interesting that this relationship hasn't been pointed out in the past, considering how much literature there is, how many publications there are when it comes to ECT. And I guess that's everything. Thank you guys for your time and your attention. I appreciate it.
Thank you so much, Kalesa.
Should I leave the discussion points up, or does it not
Yeah,
matter?
I mean, it does not matter. If you feel like that would contribute, please do leave it up. So I don't see any questions from the attendees. So I will, I have actually a lot of questions. So what is the implication, in your opinion, about the relationship between the static and dynamic impedance? Like, in practical terms, what does that, how does knowing that help us?
That's a really good question, because I've definitely thought about it a lot. I think the lack of variety within the data that is specific to dynamic impedance tells us that there could possibly be a stronger practical application for that. It also tells us that there are certain things about our physiology and our head anatomy that are sort of like, that are irrefutable, right? Like, if someone has consistently high static impedance, it doesn't mean that that really applies to anybody else. And when you're looking at how tissue variability changes, and, you know, because the tissue dynamics changes when the current is applied. So there are certain things that just can't be refuted, like, you know, the amount of fat tissue you have, the amount of skin tissue you have, and how that affects the impedance also will tell us how the current is shunted in the head, and whether or not that can actually elicit not just a motor seizure, but a whole brain seizure that will give you the response you want for the therapeutic advantages of ECT. But I hope that answered your question.
Yeah, it does. And it's, you know, it's definitely an open question, right? Like, because this was theorized, now there is good evidence for it, and hopefully people will use that. So, and also kudos for replication, like having that same type of relationship in multiple different sites. That's actually really great. But I actually had a question about something that is probably not your direct work, but the age-charge relationship. Why do you think that is slightly positive? And so as you get older, it takes a slightly larger amount of charge to elicit a seizure. Is that correct?
Yeah, the skin becomes more resistive. And
okay.
It also has to do with the way that the fat content near the skin changes as you age. And that's been largely discussed and proven by other papers as well. But the difference is so slight, in my opinion, that it didn't see it so strongly in this data, even though it was so consistent. So part of what allowed us to see the static versus dynamic relationship was the fact that the procedure was so consistent throughout, because it was the same person who was doing it and following everything to a T. And it allowed this to come through where normally it sort of didn't come through as strongly.
Makes sense. And so, but what about the variance within a session? And do you have any, have you observed any consistent changes in the static and dynamic impedance I assume that the static impedance measurement is also taken between each application of ECT, correct? And so is there any consistent trends in temporally across the session?
We looked at that, and actually, like, if you look at multiple stimulations within a single session, so that person is, let's say it's the titration session where they're having more than one stimulation. Sometimes, like 45% of the time, the static impedance drops dramatically on the second one, but sometimes it increases in like a lot more than I would assume. And sometimes it stays the same. So there's not really like a consistent relationship there. Although we did look at it, we assumed, we went in with the like assumption that it would drop, and we didn't see that. So that's why it's not on here.
If people will start sweating, is that?
I don't know. I don't know what it is. I don't know physiologically like what happens, but it doesn't seem to be consistent with everybody.
All right. I'm still not seeing any questions from the attendees, so I have no more questions. Does any of the other panelists have? I guess, Samantha, you are in the same lab, right? So if not, then I would like to thank all the panelists for a wonderful presentation. And I wanted to say something else. Oh yeah, I also wanted to thank Nicola for minding the back end and keeping the conversation flowing. Thank you so much, Nicola. And see you all at some point, and have a wonderful day, morning, evening, wherever you are. Bye.
Thank you, guys. Bye.
Thank you.
Thank you.